Good morning, and welcome all to the Young Investigator Neurology Career Development Workshop, hosted by Texas Children's Hospital and Baylor College of Medicine, Kennedy Krieger Institute and Johns Hopkins School of Medicine, Scripps Research Florida, and Bridge the Gap Syngap Education and Research Foundation. I am Charles Bergamo. I chair the board of Bridge the Gap, and I'm also a parent of an 18-year-old daughter, Gabriella, with Syngap 1. I look forward to today's conference and the upcoming days as we have a loaded lineup, and we'll hear from a broad range of professionals, together with parents and caregivers, about living with Syngap. But first, a quick word about Bridge the Gap and our organizational mission, which brings individuals such as myself and Monica to you today. Our mission is to advocate for those who cannot advocate for themselves, to educate our fellow caregivers, the medical community, and the public about Syngap, and relevant for today's conference, to support you in your research efforts. We also recognize Syngap knows no borders, and we're here to support the global community and to push forward toward a better understanding of Syngap, a better plan of care and treatment for those with Syngap, and most importantly, a better life for individuals such as my daughter and Monica's son Beckett, and those who love and care for those individuals with Syngap. Before I turn this over to Monica Weldon, president of Bridge the Gap, I'd like to give a heartfelt thanks to all the individuals behind the scenes who have helped make this event happen, especially in light of all the changes foisted upon us by COVID. Without individuals such as Leslie Ruthven, our conference planning chair, Dr. Connie Smith-Hicks, Dr. Jimmy Holder, and Dr. Gavin Rumbaugh, all of whom were instrumental in putting this together. Also, Nathan Foreman, Connor Murphy, Drew Kirkbridge, and conference co-chairs, and my fellow board members. We all applaud your continued work and research under these unique times and circumstances, as well as the professionals here at Dudley Media who have been kind and understanding in working with us to help us understand the medium that I have never worked in before, namely sitting here in a TV studio and the surrounding setting. As many of you know, these important events do not take place without sponsors, and I'd be remiss if I did not take a moment to offer a heartfelt thanks to all of our sponsors. We also wish to thank the National Institute of Health and Neurological Disorders and Stroke, supported by a NIH NINDS grant awarded Johns Hopkins School of Medicine, Kennedy Krieger Institute, Baylor College of Medicine, Texas Children's Hospital, and Scripps Research Florida. And now to kick off day one, I'm pleased to introduce Monica Weldon, president of Bridge the Gap. Monica is the founder and driving force behind Bridge the Gap. I'm always amazed at her work ethic and her ability to move the needle as it relates to Syngap and our organization's mission. Monica will also set out some housekeeping for the day and, working double duty as usual, kick us off with the first presentation of the day. It is my pleasure to introduce Monica Weldon.
Thank you, Chuck. This has been a great pleasure being here today and being with you all. A couple of housekeeping reminders. Today we're going to be using an app called Slido. And this will be used to ask our presenters questions during the question and answer session. And we will be putting up a slide. I also would like to remind everyone that we will be having a Young Investigator lunch and dinner where we're going to have families present so that we can just have a casual conversation so that we can learn a little bit more about your work and also a little bit more about the families and how they live and cope with living with Syngap. A real quick reminder is that today we have 2 breakout sessions this afternoon. We have breakout session B for Basic Scientist. So if you signed up for the Basic Scientist breakout, that will be held here in the studio for live stream. If you signed up for the Clinician Session A, that is going to be an outside link that you can access on Day 1 Hub. And remember to come back for the live stream here today at the end of those breakout sessions so that we can close out and then you can give us your feedback comments in the end of the conference survey today. Well, I wanted to go in, and many of you know me, many of you do not, but I wanted to share with you today my personal story. And how this all started. And I would like to introduce you to the journey in which all began this. And this is my son, Beckett, and this is Beckett's story. So I thank you again for all joining us today. And I hope that it really makes an impact in your work and that you can learn all of the things around building your lab and getting some advice and some knowledge around the experts, but also get to know the families, which I wish we could all be here together in person. And I'm sure the next time we meet, we will be. So thank you for being here. And I just want to go into my own personal story. Today, I'd like to introduce you to Beckett. Beckett's journey has been incredibly I would say daunting, but also gratifying, because without this little boy, I don't think that we would really be here, and it has changed my life, and I'm hoping it has impacted the lives of the families up to now to where we have that future hope because of all you in the audience. It all started, well, we'll go with what Syngap is. Syngap is an intellectually related disorder, a neurological developmental disorder, and it creates the protein Syngap. It's the most abundant protein in the brain. It controls brain development, and it's responsible for brain plasticity. It also controls dendritic growth. And the people that have mutations only make half the protein, so they're really only developing at kind of half speed. And that's why we're here today. What we know about the protein is incredibly important, but we also know that there are so many more things that we can learn moving forward in the future. Some common symptoms that we have come across as we've moved through time learning about this disorder is that they have many patients have intellectual disability. They have varying degrees of epilepsy, autism spectrum disorders, ADHD, impulsivity, sensory processing disorders, mood disorders, sleep disturbances, and hypotonia. And the list could go on. And I'm hoping as time goes by, we can help treat some of these symptoms that our children suffer from. Well, Beckett's name actually means slow-moving stream, and this was the beginning of my journey. He is a twin. He's a fraternal twin, and his sister's name is Piper. And I noticed at 4 months old that he was not progressing the same as her. And these are some of the pictures when he was 6 months old, 5 to 6 months old, where he could not sit up, and he had to wear a little helmet. We went through about 4 years of just constant testing and therapies. We went through MRIs and CAT scans. We went through the basic genetic testing, which all came back normal. But I knew in my heart there had to be something else. He lacked motor control. He had pronated feet. His muscles were weak. He had strabismus, or the lazy eye. He had digestive tract issues. When he was an infant, he had reflux, acid reflux for many years, and constipation. Sensory issues. He couldn't eat certain foods. He was very, sensitive to temperatures and motion and flashing lights. I knew that something was wrong. He ended up sitting up at 10 months. He crawled at 16 months and walked at 22 months, all with the help of PT, OT, play therapy, and food therapy when we began all of that at 8 months old. Then we went through 19 specialists until we finally reached Texas Children's Genetics. We had gone through all the stops. A lot of people really don't know kind of what happened behind the scenes, but I thought I'd throw a picture up of the notes I took. This is the actual book that when I got the phone call with Beckett's diagnosis, I jotted down everything that the doctor told me, and it really honestly didn't cover even really half a page. All they knew that it caused was global delay. There were some autism spectrum disorders. It causes intellectual disability. They told me it was a fluke because neither myself or Beckett's dad had the mutation. And it was all caused because of a little bitty amino acid that changed from a C to a T that caused a codon stop, which prevented that gene from making the protein he needed to develop properly. Well, to get that test, they only gave me a 20% chance to find anything because the whole exome testing then was experimental. And I knew that insurance wasn't going to pay for it, but I had to know, and it was worth the money. So I went and took out a $15,000 loan. Now, it probably wasn't my husband's favorite thing to see, but and it wasn't my favorite thing to do, but I had to know it was worth every dime to go through that. Now, of course, as you know today, genetic testing is the cost is driving down. But I kind of wonder if I had never taken out that loan, where we would be today. So he was the first one diagnosed at Texas Children's in 2012. He was one of 6 recorded Syngap 1 patients in the world at that time. That diagnosis sent me on a journey I never expected. I think I cried all the way home that day, and I processed for about two days, and then really just had to bite the bullet and do something. And I finally went to a neurologist who listened to me, and we got a 22-hour EEG at the age of 5, and he was diagnosed with absence and atypical frontal lobe seizures. He had 15 seizures in a 6-hour span, and they were all at night, and probably one of the reasons why he did not sleep. Currently, we have him controlled under Lamictal, and which is a glutamate inhibitor uptake seizure medication. And at the time before we had his seizures treated, he was actually kicked out of 4 different daycares, so it was a real struggle for us to try to find help for him. After we finally got the help we needed, and we scooped up Dr. Jimmy Holder at Texas Children's, he took us on, and he's been with us ever since. And him, and then also Dr. Gavin Rumbaugh, who was the first basic scientist I reached out to, changed the whole path in which what got us here today. And Beckett, as we progressed, tracked his symptoms. And now that he's on certain types of medicines that now treat his symptoms, he is able to progress in a way that we thought he never would. And I'm very thankful for that. As we go through, we are still involved in trying to mix the medications and progress with therapies. And he's making gains in memory and cognition. He's sleeping. His behavior is so much more manageable than it was before. And he attends regular daycare, which I'd never thought would happen in my life. And he is actually functionally playing and learning with children. Now, cognitively, he might be 5, but I would take that over any day with his suffering that he had before with behavior and seizures and the sensory issues that he had. So as we move forward, I want you to think about some of these questions as you move through this time, and we hope you can join us for our international conference tomorrow. And then most importantly is we hope you can join us Thursday afternoon for our patient-focused drug development meeting, where we will have the FDA in listening mode and many other different therapy companies and scientists available. So that is open to the public and free. So please, it's not too late to register. But as you think through what you're learning today, what is a parent supposed to do when your child is diagnosed with an ultra-rare disease? You just want to have an answer. What do you do to get an answer? There are very few resources, and when you see a child suffering every single day, and as you all know, those of you who are parents, you would probably walk through glass and hot coals, hell or high water, to help their child live the very best quality of life they can. You know that you have dreams for your child, and you also want to find somebody who gets it. You want your own tribe. And so as we move forward, we have our tribe, we have our global tribe, and we aren't stopping till we can find a treatment. And that's just what you do as a parent. You find every way you can. So what you do, you roll up your sleeves, you take, you hold no bars, you get rid of the fear, and you'd never take no for an answer. And you look for other people like you. And so this journey for me is what started Bridge the Gap and started the group. And I am happy for the progress that the people in the community have made in creating their own local communities to continue the work and just do the very best they can, because that's what parents do. So as we take action, I want you to understand and hope that you will look at a new model in how kind of science is done. And don't be scared to talk to the families. The families are there to help you however they can. And we hope to bridge the gap to the future. And we, want to do this, and our organization does this through many programs that we have. And the foregoing path that we continue are to continue to build relationships and share our goals. And we do this by determining our priorities. And we try to be a broad-based organization. And our three solid foundation vision that we have for our organization is awareness, advocacy, and accelerating to new treatments. Our key to our successes are our families, our community partners, our research scientists, and clinical experts, and the medical centers that we have academic affiliations. And the heart of everything we do is our families. So when you are working behind the bench, just remember that there is a face that is really rooting you on behind the scenes, because we want you to succeed so that you can help our children succeed. So as you look at the progress that we've made through the times and our programs and incentives, we are really trying to build up an educated community through our different programs, such as our family meetups and our registry and our conferences like we have today, and continuing to spearhead groups of physicians and teams of physicians that will focus on Syngap 1. Our centers of excellence, we hope to be one day clinical sites for clinical trials, and also the supporting clinical programs that we have there to learn more about what this protein does in the brain. So we have a list here. So if you can look, we are working on a few more centers right now. We have them pending, and we are very excited about the growth that we have. And we have several studies that we support and recruit for, and that we have provided seed grant money to get preliminary data so that we can get bigger grants at our centers. And Our hope is just to bring everyone together for our international conference, which I mentioned earlier, which is happening tomorrow, and having the experts come together to learn more about the Syngap protein, and not just really understanding how it's going to help our patients, but understanding how it even overlaps into other neurological disorders and possibly helping millions more in the world with these types of conditions. We also want you to make you aware that we do have the largest natural history study that's been ongoing. We are 4 years old, and we have data that is free and available to researchers throughout that are working on Syngap 1. We are preparing new surveys and targeted surveys for the community. And we hope that this will help support your bench science and put a target with the translational component of science there and combining the human data with the animal model data. We do have currently many registrants. Data that we currently collect are the socio-demographic data, medical and diagnostic data, treatment and disease progression, management of care and quality of life. We are working currently on adding more data as well to that. Current research laboratories are there. We are starting a new program soon with another university, so we will be adding to the list shortly. And we have already published out of the database, which is hugely important and shows signs of progress. So we are very proud of the accomplishments that we've had over time to help support our scientists in the work that they're doing. Never give up, never stop. I think if you find a balance and find your passion, then we're going to all work together to continue this journey that is going to improve the lives of others. Like the slide says, it says, dare to dream, you can change the world. I wanted to thank everyone today. I just want you to think outside. Don't think that there is a box. Think that there is no box and there are no limits, because where there's a will, there's a way. And you cannot take failure as failure. Failure is a lesson, and failure is always good information that will teach you what not to do and maybe put you in another direction that's going to get you the answers that you need. And I guess that's the teacher coming out in me. But I want to thank everybody today. I want to thank you for listening to my story. And please reach out with any questions. My door is always open. Thank you. And what brings us next to our next in our program is I'd like to introduce our first young investigator presenter. I want you to remember, though, we have the Slido available. And please pull out your app and get your app ready to throw out some questions for the short question answer. And we're going to listen to our first Syngap Young Investigator presentation. I'd like to introduce Amina Smith-Fine. She is a graduate student at the University of Miami. And I'd love you to listen to what she has to say, and go ahead and take it away, Amina.
Hi, I'm Amina Smith-Fine, and I'm a neurologist working in the Moser Center for Leukodystrophies at Kennedy Krieger Institute. Today, I'll give you an overview of what we've learned from a natural history study of the disease LBSL, which is a leukodystrophy that features progressive ataxia with onset usually in childhood. The approach we've used to study LBSL can be applied to related rare diseases that affect motor development, including Syngap1. An exciting aspect of this project is that we're using wearable sensors to get information about walking and balance during tele-research visits at patients' homes. And this is especially relevant in an era where the ability to conduct research remotely is essential. So leukoencephalopathy with brainstem and spinal cord involvement and lactate elevation, or LBSL, is an autosomal recessive disorder caused by mutations in DARS2. This encodes an enzyme that's critical for translation of respiratory chain complex proteins. The MRI pattern in LBSL is very distinctive and features white matter changes in the cerebrum, cerebellum, and brainstem and spinal cord tracts. And then there's also usually an increased lactate peak on MR spectroscopy. Clinically, patients experience progressive ataxia, spasticity, and dorsal column dysfunction, and the onset is usually in infancy or childhood, although occasionally not until adulthood. Earlier age of onset has been associated with earlier loss of independent walking and wheelchair dependence. And of course, these are the outcomes that we would hope to intervene on and avoid in this disorder. So our objective is to establish clinically meaningful quantitative outcome measures for clinical trials in LBSL. Our approach is to obtain information about gait and balance using wearable sensors and an established clinical ataxia rating scale. We perform assessments during virtual visits at participants' homes, as I mentioned earlier, and this is to increase participation and decrease the burden of travel for these families. We are also doing testing in age and sex matched healthy volunteers. Today I'll be showing baseline data, but we repeat the assessments every 6 months over 5 years. So we chose the Opal system of accelerometers, as this is a non-invasive, relatively inexpensive technology for recording movement in a real-world context. The system has been used to measure disease-specific characteristics and other neurodiseases, including MS, Parkinson's disease, and cerebral palsy. And similar metrics of gait and sway have been reported in other ataxic disorders. The Opal system consists of 3 watch-size battery-based accelerometers that submit a signal to a central sensor through low-power radio frequency waves. And the sensors are just placed on the feet and the waist for a brief series of tests. So for the virtual visits at home, we send an Opal's kit, a web camera that's remote controlled, a laptop, and an Opal device manual that's in simple layperson language to explain the tests and the equipment setup. This is just a picture of one of our participating patients and family. As I mentioned, we also used as a measure the scale for assessment and rating of ataxia, or the SARA. So this is a brief and sensitive tool for clinically rating ataxia. It's been used in SCA and Friedreich's ataxia previously, and has shown excellent test, retest, and inter-rater reliability in these disorders. The SARA was able to show progression in SCA after a year, but it did not capture all ataxia symptoms reliably early in the disease. However, wearable accelerometer measurement of gait ataxia was shown to be superior to the SARA in the early stages of SCA. So we're exploring the use of both of these measures to assess disease severity and progression in LBSL. In terms of our clinical characteristics of our cohort in LBSL, this is based on a sample of 8 patients. Our median age was 10, with a range from 5 to 23 years of age. Basically, we had a minority of patients that were female. The mean age of symptom onset was about 5 years of age, and generally patients always presented with ataxia and fine motor delay. 3/4 of patients experience an episodic deterioration in the disorder, meaning if there was a fever, a viral illness, a cold, they had a temporary regression in motor symptoms, but then a later recovery after a week or so. And then in terms of overall gross motor function as measured by the GMFCS, the scores were low. So on a scale from 1 to 5, with 1 being mild and 5 being the most severe impairment, most patients only had a rating of 1 or 2. And then the manual ability classification scale, or the MAX, also were in the mild range of 1 to 2. The SARA ataxia scale is rated from 0 to 40, mild to severe ataxia, and you can see that the mean was only 8, so patients were mildly affected, mild to moderate. Okay, so this is a video of a young woman, 10 years old, with LBSL, performing a walk test. So this is a 22-foot walkway. Participants perform 2 trials at a time, and information about the gait cycle is collected. So first of all, what we found in terms of the stride length during the walking test is that the stride length, and again, the stride length is the forward distance traveled by the foot during a gait cycle. This is the first initial contact to the next initial contact of the same foot. And a short stride length is a compensation for unsteady gait. So as hypothesized for patients with ataxia, our LBSL group had a shorter stride length compared to their age-matched controls. This difference was not statistically significant in our small group of participants. And then next, it appeared the time that it took to complete a gait cycle was slightly longer for the LBSL cohort. And finally, the LBSL group had a slower stride velocity compared to the control group. And per the literature, a difference of this degree of about 0.1 meters per second is actually clinically meaningful, although we did not see a statistically significant difference. The hallmark of ataxia, of course, is variability, which can be estimated by the lateral step variability. So when considering 3 consecutive foot placements made by the left or right foot, the LSV is defined as the variability of perpendicular deviations of the middle foot placement from the line connecting the first and third. And this basically indicates a veering or stumbling gait, and larger deviations might also indicate a wider base to preserve stability during the walking. So our LBSL group showed a greater lateral step variability versus controls by about 2 centimeters, which was statistically significant. Next, we examined circumduction and elevation at mid-swing. And so this is basically the distance that the foot travels outward during forward swing. So we saw that the LBSL cohort exhibited a slightly greater circumduction value compared to controls. And in terms of the elevation at the mid-swing, there was also a statistically significant increase in this value in LBSL patients. We next considered turning during the walk trials and found that the LBSL participants took slightly longer to turn with a smaller turn angle, and that resulted in a slower turn velocity in degrees per second versus controls. So together, these findings indicate balance and stability during the turns for these patients. Next, to move on to talking about standing balance, our remote balance testing consisted of 4 postural sway conditions in which we challenged participants by variably removing visual input and or narrowing the base of support. So 4 conditions. They performed 2 trials of each condition, and their stance width is standardized with a foot plate. Sway is measured from the lumbar or waist sensor, and there are 10 key variables that are collected by the sensors in 3 different planes.
10 seconds left.
So
Done.
to demonstrate, these clips represent LBSL patient participant performance on the balance test. Above are plots mapping this girl's sway path compared to an age-matched healthy volunteer. And basically this is looking for them top down. So you can see that across all conditions, there is a greater sway for the LBSL participant, but especially in conditions when her eyes are closed, and it's greatest in conditions when her eyes are closed, in the condition where her eyes are closed, also with a narrow base of support. And I'll just demonstrate again each of these.
Almost done. Less than 10 seconds left. 10 seconds left.
Okay, so here are the sway plots for the control and LBSL groups, and below we've quantified the sway area for the groups. So as in our representative patient, we just saw the sway path is greatest and the feet apart, eyes closed, and the feet together, eyes closed conditions. And then to further evaluate the impact of removing visual input on sway, a Romberg quotient was calculated for each participant. This is the sway area with the eyes closed divided by the sway area with the eyes open. And you can see that the average Romberg quotient was significantly increased for LBSL subjects, which reflects proprioceptive contributions of the dorsal column medial lemniscus pathway fibers in the spinal cord. So to us, this meant that the myelopathy that's seen in LBSL may be an important contributor to their overall motor disability. Lastly, these plots are demonstrating that the SARA ataxia score is strongly correlated with the lateral step variability during walking in the first plot and the sway area during standing. This supports the idea that the wearables are capturing variables that are clinically meaningful and do represent ataxia. So in summary, through virtual testing at home, we found that LBSL participants walk and turn slower, have shorter stride lengths, and show more gait variability compared with healthy volunteers. The step variability and sway area measured by the wearables is strongly correlated with the clinical ataxia scale, the SARA, which we think is likely related to sensory or cerebellar ataxia. And then finally, removing visual input has a greater impact on the sway area than narrowing the stand space, suggesting that the myelopathy or the dorsal column dysfunction is really an important contributor to instability while standing. And so again, this is demonstrating the power of these wearables in a specific leukodystrophy, but this is an approach we do believe can also be applied easily to related rare diseases affecting motor development, such as Syngap 1. All right, and thanks to everyone who participated in this research. Certainly, of course, number one is our LBSL participants and families, and who also gave permission to show the recordings today. Ali Fatimi is the principal investigator and my mentor who has worked with me on this project, as well as Amy Bastian, who directs our motion analysis lab at Kennedy Krieger. Thanks to Constance Smith-Hicks, who gave the invitation to participate in the Young Investigators Research Day, and to the rest of our amazing team. Thank you very much, and I'll take questions live.
Hi, Amina. How are you? Thanks for being with us today. Great presentation.
Thank you. Good morning.
Good morning. I'm going to field some questions from the audience to you, and we actually have a question. But really quick, I want the audience to log into Slido and get your phone out and bring up the app. And if you have a question, I will field this to Amina, and so that she can answer. And I do have one question already for you, Amina. Dr. Jimmy says, perhaps I missed this, but have you recorded gait in the same individuals over time to demonstrate reproducibility?
Great. Thank you for the question. So I really only briefly mentioned this at the beginning of the talk. So I'm actually going to answer a couple of questions regarding repeating the gait measures. So we were first pretty interested in test-retest reliability. We know ataxia, the hallmark of ataxia is variability. These patients fluctuate. Certainly we expect, day to day in some cases that might happen. So what we did at the beginning of the study was to do all of the walking and the standing balance tests actually on 2 sessions on separate days. So day 1 and day 2, we would send it to the home and have them walk. So, and I didn't show that data, but we did have, especially for the walking, the test-retest reliability was high. So, basically inter-rater correlations were, I believe, mostly 0.9 and above. Yeah, right around 0.9 for the walking, closer to 0.7, 0.8 for the standing balance. So some more variability there. And then sort of I only showed baseline data today, but over 5 years, we actually are repeating the same walking and standing balance measures in the same patients every 6 months over a course of 5 years. And so, with and the thought is that possibly we would be able to see some evidence of progression, and we'd be using some modeling to determine that at the end of at least a couple of years.
Okay, awesome. We have, and then he has a follow-up question. Jimmy asked, what is the age range for your population?
Yes. So, and yeah, again, that was just sort of briefly in one of the tables at the beginning, but so our median age range was 10. So this is a pediatric population, and our age range was from 5 years to 23 years. So we've had, so for younger kids, that developmentally and cognitively are age appropriate. We've tried it, and a 3-year-old, I think, is the youngest age, but still a lot of instruction is needed. And we basically found that 5 is that lower age limit for kids being able to follow the instructions without any prompting to get what we feel is a really adequate trial.
Awesome. Okay, and we have another question that came in. Dr. Julia Dahlman asks, is there any evidence that there are peripheral proprioceptive deficits might also contribute to balance issues in this group of patients?
Yeah, that's also a really good question. And, that is something that is honestly it is difficult to assess remotely. So, the study that I mentioned, these are all virtual visits where we're sending equipment to the patients' homes. Most of them are also patients at Kennedy Krieger, so we've been able to do at least one physical exam, and then we have documented neurologic exams from their home neurologists. So we have some information about if they have like a tuning fork assessment, and we look at vibration and proprioception, et cetera. We have some other ways that we can kind of, or even some of them have had nerve conduction studies. So there are some ways to get at that question, but it's challenging to do all that remotely. I think it would involve the clinical piece to bear it out more completely.
All right. We have a couple of questions. Actually, I had a question just as an observer. In the Syngap group, because I know this is a different group, when you looked at the MRIs, because typically a Syngap, looking at some of the data, the MRIs, like my son's, for instance, they said it was non-remarkable or no significant changes in his MRI. So did y'all notice any correlation in the MRI imaging, points or something spectacular that, you could mark on the MRIs? Because that's something that we haven't actually focused on in the Syngap or a study that I know of, looking at the MRIs and comparing them, because most of them come out, from what we've reported in the patient community, normal.
Yeah, absolutely. That's a good question. And, certainly in the field of leukodystrophies, we're very interested in, is there any way we can look at the brain regions involved, the extent of changes in those brain regions, and try, somehow to link that to disease severity or to understand more about prognosis. That's been done really nicely, actually, in another leukodystrophy called ALD, adrenoleukodystrophy. And in fact, looking at brain regions, at the symmetry, at the distribution helps a lot in terms of understanding when somebody crosses from mild disease to severe disease and they need a treatment such as bone marrow transplantation, which in that case is in the right patient at the right time can be curative. So that's sort of the thing that we all hope for. And there's an MRI score that we're trying to develop in LBSL that accounts for the areas that were involved or that are involved most frequently. Right now, we've tried to develop that score and look at ataxia, and there's not a clear relationship, unfortunately. So we're still making that attempt. We think that part of the issue is that we need more specific scoring of this, of what's happening in the spinal cord.
I see.
So yeah, but it's a challenge. And in the literature, it seems like there's an unclear relationship. So.
Okay. No, thank you for that question. This was absolutely awesome. And I want to thank you for being with us. This has been incredibly insightful. Also, if the audience has any more questions, because we're running up a little bit on time, thank you so much for Amina for being with us, and we hope you can continue throughout the day and the week to be with us. And for the audience, again, if you have any questions, we do have a feedback survey. That in that survey, if you have any additional questions, we can field those questions back to the presenter. So leave those in there, and we will make sure that the presenter gets those as well. So thank you so much, and thank you so much for a great presentation. It was incredibly insightful.
You're
thank
welcome.
you.
Appreciate being able to participate.
And now we're going to pass it over to Chuck to introduce our next presenter.
Okay, so now we're off to our next presentation of the day. And I'd like to introduce, and hope you'll forgive me in pronouncing his last name, Thomas Vassier. He is a PhD recipient from the University of Lyon in France, and he's staff scientist at Scripps Research. And he'll be, his video will deal with structural and functional sensory motor alterations in a model of Syngap1 related brain disorders. So I'm looking forward to this presentation. And here we go. Off to Thomas.
What if you could feel this texture? This texture may give you pleasure or make you uneasy. However, by actively seeking the surface and exploring further through touch and other senses, you may be able to form a more coherent picture. Make a decision about how to interact with it. So we operate in the world through those active interactions with our environment, which are key for our daily lives and also our survival. What are the key mechanisms in our brain that enable this interaction between sensation and cognition during this active feedback loop? And most importantly, can a disruption between this active sensing and perception underlies cognitive impairment, like the one observed in neurodevelopmental disorder, such as the one associated with Syngap1 haploinsufficiency? This is the burning question that major contributor for this project, Jesse, Sheldon, Gavin, and I, along with others, set out to address. So we were especially interested in this context of the role of Syngap1, this postsynaptic protein that has been associated with a lot of major disorders, which include schizophrenia, epilepsy, and intellectual disability. But beyond its role at a synaptic level, we hypothesize that Syngap1 impacts circuit assembly and disrupt active sensory processes and perception, which are essential for normal cognition. So the main idea, if you like, is there's no proper cognition without proper perception and active sensing. And out of all the senses, we're really focused on touch, as it is one of really a fundamental process that occurs only on. And this was really striking in the case of Syngap1, because thanks to a study published by Sheldon and the Syngap1 patient registry, we had insight into the fact that touch-related sensory processing defects really were prevalent, as they constitute 35% of the sensory processing deficits that were described. So, in our case, we're especially interested in a mouse that can be used as a model for Syngap1 haploinsufficiency. And past research from the lab have shown that behavioral deficits such as social interaction, learning, and memory are impaired, and there's a risk of anxiety and risk-taking behavior, which showed that this mouse model has good face validity. So our hypothesis was really that Syngap1 mutation impacts circuit assembly, disrupting active sensing. But how do we go about studying this in the mouse system? So unlike us, mouse really probe their environment with their whiskers, and that's really what we use to as a surrogate for touch in the animal model. So I'm going to walk you through pieces of data that we gathered over the year and that really get to this idea by looking at first what happens when a stimulus is triggered and the response in a part of the brain, the somatosensory cortex, which is really fundamental to process this type of information. And now from there, we built a broader picture of the global circuits within the brain and identify key brain regions which we found are critical for the good active sensing that happens during more naturalistic tasks like pole presentation. Now this can impact the mouse behavior as a whole, exploring more complex environments. So initially what we found when we performed this type of piezo stimulation was that part of the brain where a reduced sensory evoked brain activity. And that was quite interesting and very surprising because other parts of the brain in Syngap and haploinsufficient mice have shown the opposite behavior, where in the mutant mice, what you would see is an hyper activity of the brain. So this suggested to us that these brains are way more complex and complexly organized. So we looked at the morphology and the structure at this one brain region especially, and throughout the development. And what we found was there's a decreased complexity in layer 2-3 and a strange dynamic in the layer 5, where the neuron seems to mature much faster in the mutant mice. So if you like, that's the type of trajectory throughout the development in terms of length and complexity of those neurons. And this really stemmed to us this idea that some early disruption may have like a critical impact on how those systems and circuits are built, not only at the place of the somatosensory cortex, but also more globally. So in order to address this, we focused not only on the structure, but also on the circuit as a whole. And in order to probe the circuit across the brain, what we did is we used a rabies virus strategy, where you can inject a viral vector inside a specific brain area, and then it's going to spread across the brain and enable you to identify all the inputs which are received by the region of interest. And when we looked at the wild type and mutant mice, we didn't find any gross differences. And you can see an example of this type of quantification where most of the brain regions were found to be similar, except for one specific connectivity between the motor cortex and the somatosensory cortex. And this was confirmed through optogenetic experiments, where we could confirm this selective outside connectivity between those two brain areas. This now really led us to be like, okay, there's two key brain regions that seems to be disrupted within the circuit, especially S1, the somatosensory system, and M1, the motor system, which are essential for more naturalistic behavior, like active sensing when a mouse is presented with a pole and the way it explores it. And here you have an example of the type of behavior that occurs in the mouse, where you can see that the wild-type mice are exploring the pole, touching, and the het mouse mutants do also to some extent. However, their exploration is much shorter. The interaction with the pole are much shorter. And you can see a quantification of this in the conventional knockout lines, where you can see this reduction. However, when we did the cortical knockout, you can see that this seems to be like cortical specific, and when we rescued Syngap in the cortex, this disappeared. So what happens now at the level of the circuits functionally? So now the idea was to be able to probe the brain and assess its function through different areas of electrodes across multiple brain areas. And while the mouse is performing the same task, now record the brain activity in the somatosensory cortex. And you can see that for each touch event, there's activity which are associated with S1. So now if we look at a quantification of that in the wild type, you can see the representation of different brain regions, like the thalamus for the upper panel, S1 for the middle panel, and motor for the bottom panel. And what you observe is consistent with what's been published with the literature. After the stimulus or like touch onset is presented as represented by the dashed line, you can see an increase in activity of S1 and an increase in activity of the thalamus as well, along with the motor cortex. Interestingly, however, the pattern is very different, with a striking reduction in the somatosensory cortex and also a very different dynamic in terms of brain activity prior and after the touch onset. However, when we looked at the whisking phenotype, although there is differences, they don't seem to be as drastic as with touch. So we really think this type of behavior, and we have additional data suggesting this, where the behavior during whisking is also normal, really relates to this disruption in the active sensing process itself, where you need to have sensory information and feedback occurring through this circuits that I was describing. So now we can hypothesize that this type of disruption will have a drastic impact on more naturalistic behavior, which are required for the mouse to perform, like textual discrimination. And what you can see is that there is indeed an impaired textual discrimination when two textures are presented. Then a wild-type animal is able to discriminate between the new object that is presented to it. However, a mutant cannot perform this type of behavior. So to us, taken all together, this piece of information really left us with this idea that Singaporean mutation not only impact the circuit assembly, but also disrupt active sensing, which are required for cognitive processes. With that, I'd like to thank the National Institute of Health, my collaborator in the Miller and Rumble Lab, as well as our collaborator in Sweden, Dennis Medis and Daniel Firth, and all the Syngap families for the insight they provide. Thank you very much for your attention. Enjoy the meeting.
Hi, Thomas. It's so good to see you. Thank you so much for your presentation. It was awesome. I wanted to remind everyone that for questions, you can go to Slido, and we're going to put that up again. So if you're just now coming in for any questions, and go ahead and put that in. With your camera, you can do the little QR code, and you can put in your questions, and we will field those to the good doctor over here. I'm so excited. This is great work. And we're going to see about some questions that come in. And now, while we're waiting, Thomas, is this, just out of curiosity, do you notice a change over time with the sensory perception in mice after you've introduced them to a certain like a touch? And let's say it's, like even in our children, they get desensitized after so much. Do the mice do the same thing?
Hi, Monica. Thanks for the question. And that's actually something that we're exploring at different levels. So all the initial studies were, the initial phenotype were observed on a very short time frame, like 30 seconds, which were initial to the given condition of mice. So you place the mice in a specific condition, and then they're in the presence of the pole, and we observed the phenotype that were described over 30 seconds. But now, like for the brain recordings, what we have is like much longer period of behavior where they can have this type of active sensing, and we are trying to dissect those dynamics that you're actually referring to on a much longer time scale. I mean, still brief because it's in the order of like 30 minutes. However, it seems, and we have like, what I should mention too is we have altered condition because we have this idea of like multisensory dimension. So we have phase where they're in the dark and the pole is present. So you have, sensory input from the visual system as well, or touch, and we are trying to dissect those aspects. And also for sound, so we have condition with the sound that's present and they can like sense through touch. And we're in the current process of exploring this exact question. So I don't have a definitive answer yet, but that's something we're actively working on.
Okay, awesome, awesome. And now we have some questions flooding in for you, Thomas. Our next question is from Julia, and she asks, what do you think underlies the really specific altered connectivity between motor and somatic sensory cortex, and what is known development of this conductivity? Or connectivity, sorry.
So for this, what we think is happening, and a lot of this stems from our previous work, where what we've observed is the morphology of those neurons and the development of those neurons themselves is having the different kinetics depending on specific brain region. And why we think this specific connectivities are altered is probably a combination of the program of those neurons that have this like specific altered trajectory in terms of their morphological feature, and a balance between the brain that's trying to keep balance and homeostasis. So we think that's conceptually that's the concept where some of those neurons are going to develop much faster, they're going to have like more activity, and the brain is going to try to compensate for those functional alteration early in development, and try to compensate for it in a structural fashion that will then lead to altered connectivity later on. So that's kind of the leading hypothesis. That's also something that we are trying to explore more. We have not performed any of those studies early on. Mm-hmm. That's something also that we are really interested to move our research program towards.
Okay, awesome. And we have another question coming in from Jimmy. He asked, the tactile sensory data you present could underlie patient sensory seeking, but do you think that it also underlies sensory avoidance? This is also seen in Syngap 1.
So I think for the touch phenotype that we've observed, it's almost like a lack of registration that the object is there. Because if you see like the dynamic of the whisker when the mouse interacts with the pole, it's very specific, it's long, there's like some level of bending. I mean, for an analogy of what you would see with an object with your hands, right? You would take your keys, and then maybe you'll try to like rub them, touch them more. But in the case of Syngap 1, it's almost like they're tapping on it, right? Without really like trying to explore it and register it. So to really get at Jimmy's question, I mean, in this specific instance for touch, I don't think it's either like seeking or avoidance, maybe just, like a misregistration of the nature of the object, or like misrepresentation. And that's true with some of the behavioral data as well, where there's, this incapacity to be able to discriminate between two different objects. But we have, other, I think there's other things as well that are present from other lines of research in the lab, where it's lack of avoidance. And I think that's something that was seen in some of the patients as well, right, where like registration for pain, for instance, is not really present, or the threshold is different. So again, like maybe not registering the information coming in, the sensory information coming in.
Okay, awesome. And we're getting a full of questions here, and we've got another question for you. Dr. Kadam asks, how is the surround inhibition for the barrels altered in Syngap 1?
So we have not explored this yet. We really think that some of the phenotype is driven by cortical excitatory neuron, And again, this stems from previous work and other type of data that we have in the laboratory, where we have the capacity of really looking at the mutation on those excitatory neurons and not on the inhibitory neurons. And it seems like most of the phenotype is driven by those excitatory neurons. So if you have, to give you a more concrete example, if you have an excitatory neurons that has a mutation, then we observe the phenotype. And if you have like an excitatory neuron that doesn't have the mutation and an inhibitory neuron that do have the mutation, we don't observe the phenotype. So there's like some level of rescue, and this seems to point out to a specific excitatory mechanism.
Okay, and we have one last question for you. And this question is, was there anything that surprised you about the sensory processing findings in your research that you weren't expecting?
So, I mean, I think like this ties up to Jimmy's question in terms of it could go both ways, right? You can have like more seeking, or you can have like a complete avoidance. And I think what was surprising in a way was the fact that I think it made our thinking evolve where towards this lack of registration, and not so much like, oh, maybe there's going to be more seeking, of texture, or like because to try to compensate for some of those things. So whereas now I feel we are like maybe towards more some kind of not necessarily avoidance, but really something more complex about the touch, because one could think, oh, if it's avoidance, there's going to be no touch. But there is touch. It's just like the way the exploration happens is different. So I think this brings, a more nuanced feature of this type of sensory exploration.
I see. Okay, that's interesting. And I have one last question for you. Do you notice anything different in how, do you look at when the brain is developing and you're looking at these neurons, do you notice anything different in comparison to the mice? Let's say the litter mates, and this is just my own curiosity, I guess, asking, do the neurons develop differently in different areas? And do you see a variance in their sensory touch if they're litter mates having the same, genetic makeup? But do you notice a variance in how those neurons develop in maybe different parts of the brain that possibly interfere, make that spectrum of touch?
So, I mean, the way we're looking at the data is more like on a population basis, right? So we pull a lot of subjects together to be able to have this. And I think some of the question, like your question is very well taken and could be addressed, but we would need like much more mice and trying to see if there is like a spectrum in performance, because maybe some mice with a mutation have a different capacity or like different type of behavior, and then we can like potentially tie this back up to either like functional connectivity or structural connectivity. We don't really have the power to do this. However, what I can tell you is for some of our data, what we've observed at the population level is we have like more variability. So the average of the population is different, but the variance is also greater in some instance with Syngap that, would tie it up to this idea of the spectrum. I mean, there's also other, interesting aspect of maybe one day part of the spectrum like for even a given individual, maybe one day the performance would be one way and another day the performance would be a different way. So those are like very exciting questions, but we don't really have a detailed insight on that.
How you describe it is almost exactly like our children. So no, I so appreciate you being with us. This is so fascinating, and the entire team, you and Gavin and everybody at Scripps are doing such an amazing job. Thank you for being with us today. And for the audience, if you have more questions that did not get to get them in, please make sure that you ask them in your feedback survey, and we will funnel them to the appropriate person. Thank you so much, Thomas, and I can't wait to see you guys in person soon.
Thanks, Monica.
Thank you.
Thank you.
All right, our next presenter is going to be Brennan Sullivan, and Brennan is going to be presenting on, and you're going to have to forgive me, Brennan, I'm not sure I can pronounce this, epileptogenesis in Syngap 1 encephalopathy. So we're looking forward to that presentation. Take it away, Brennan.
Hello, everyone. My name is Brennan Sullivan. I'm from the Kadam Epilepsy Research Lab here at Kennedy Krieger Institute in the Johns Hopkins School of Medicine Department of Neurology. I'd like to thank everyone for tuning into my talk. Today, I'll be presenting some unpublished data and talking about some ongoing experiments here in the Kadam lab. And the title of my talk today is An Early Forecast for the Syngap 1 Storm. This is a large cohort study published by Dr. Ingrid Scheffer and the group in Australia, where they describe the median age at seizure onset for patients with Syngap 1 mutations to be 2 years, with a range of 4 months to 7 years old. So with this cohort data, we know that seizures are occurring early in life in patients with Syngap 1 mutations. So as a preclinical researcher, the question is, well, do mice with Syngap 1 haploinsufficiency, do they also have early life seizures? And previously, all seizure data from this mouse has come from its adult age. So here I show in a P7 HETs, so these are haploinsufficient P7 mice. This is the mouse originated by Dr. Hugenair, the original one, and the one that the Kadam group we had previously published this year. These are early life seizures. They occur on the 7th day of life in these pups. I use a tethered subdermal scalp EEG recording for 2 hours. And here on those 1 minute traces on the left, you can see there's spikes, there's poly spikes and clustering of spikes, as well as seizures. Over here on the right, you can see a P7 wild type 1 hour trace, as well as a P7 het mouse trace. And this is a het mouse with a lot of seizures over 1 hour. When this is quantified for these P7 hets that were the same mice are recorded at P7 and then again at P12 for 2 hours each, you can see that they're ictal events per hour. I want to highlight that the females here are in pink and males are in blue. Currently, we have not seen a significant difference between males and females with their ictal events per hour. However, there does seem to be a group that at P7, they have a very high seizure burden. And there's another group of Syngap heps that have low seizure burden at P7 or even no seizures at P7. So with this being known that there were these seizures, and some of these seizures are short duration or other sub-seizure ictal-like activity, what we wanted to look at was there a behavioral phenotype that was different from the hets to the wild types? And so what we did was we just looked at a behavior on video with a score blind to genotype, age, sex, and seizure score of the hets, so blind to all those things, and they scored the behavior just on video of flexor spasms or jerks, which is denoted as 1, waddling, lordotic stretching is 2, or a behavioral seizure is 3. So when you look at this for the flexor spasms or jerks, there was a significant increase in flexor spasms or jerks in the het compared to the wild type. And then there was no differences between wild type and het for either of the other behaviors. However, when we took this, what we knew was scored but not by seeing the EEG or the true seizure burden, just the behavior, this behavior score. And we overlay that to what we did know from the EEG of the true seizure burden. What we could see was that of all of the seizures, less than 50% had any behavioral score at all. So not only are these seizures subclinical, but they require EEG, as the majority of them were electrographic only. So this is, again, this is P7 Syngap 1 HETs. So a study I would like to highlight, this is from Takao Hinch's group at Harvard in collaboration with Francis Jensen. What they showed here was using a chemoconvulsant-induced seizures with PTZ. It is a GABA-A antagonist. The idea is that the GABA-A antagonist decreases inhibition in the brain at high doses, and it induces seizures. So they induced seizures with a GABA-A antagonist. These seizures disrupted the plasticity of the auditory cortex during its critical period, where the auditory cortex undergoes refinement and the unsilencing of synapses in an experience-dependent manner. One of the things that they show that these early life seizures have a disproportionate impact on critical periods of plasticity, but also that AMPA receptor antagonists rescue these deficits in this acute seizure model. So with this being known, what I wanted to do was look at Syngap 1 HETs at P7 and see if I could stop seizures. So this is an EEG recording for 1 hour. These black lines are seizures. And you can see at 1 hour, I gave a loading dose of phenobarbital. Phenobarbital is a positive allosteric modulator of GABA receptors. It's the idea that it's going to increase inhibition. And when this occurred, it had little to no effect on seizures and HETs, and maybe even aggravated seizures in the second hour. I'm blind to genotype during my experiments, and so I wanted to show that there's even some, very few, but some knockouts live to P7, and I recorded from them. And when I do that after 1 hour of phenobarbital, when I gave it to them, they actually were aggravated by this loading dose of phenobarbital. So at the Kadam lab, we study these chloride cotransporters and early life seizures. So we've been interested in a chloride extruder, KCC2. And what KCC2 is known to do is extrude chloride, and this allows for the GABA-A receptor to be inhibitory. So because I gave phenobarbital, and there was no inhibition in the brain during EEGs, phenobarbital was almost aggravating these seizures. I then wanted to look at KCC2 to see if this was my suspect. And when I did some probing of total protein or its phosphorylation profiles, I did not see any data thus far that KCC2 is decreased in the cortex or hippocampus of P7 Syngap 1 heterozygous mice. However, another way that I've been able to probe altered inhibition in the brain is, this is a paper that's currently under review from our group. It is a preprint. I used 2 phosphomutant mice. This is the S940A mouse and the T1007A mouse. They have mutations on KCC2 that don't allow for these sites to be phosphorylated, and they've been shown to increase and decrease KCC2 function, respectively. These mice were generated originally by Dr. Stephen Moss at Tufts University. So I gave this GABA-A receptor antagonist quite a low dose compared to what is normally used for chemoconvulsant-induced seizures. I gave 20 mg per kg, and what this did is this showed that in the S940A mutants, this KCC2 hypofunction mutant, that they went into status epilepticus. So I tried to repeat these experiments by giving PTZ 20 mg per kg to a Syngap 1 het after 1 hour. And interestingly, the seizure rates went down. This was surprising. This was not what I was expecting. And what I think this may show or suggest is that early in life, there may be high GABAergic activity in Syngap 1 pups. So after this PTZ experiment, I was a little confused, and so I went and started looking at the literature for what was known about PTZ and how its relationship to neurodevelopmental disorders. And so PTZ was used as a cough suppressant and actually was used as an ECT therapy early on at high doses. So the cough suppressant was low. They gave high doses of the cough suppressant. They found it induced seizures, and they were giving this drug to induce seizures in patients instead of ECT back in the 1930s, I believe, and it lost FDA approval for its cough suppressant action, I believe, in 1980. So this drug is not new by any means, and it is actually being used in low doses. So low doses are to be given to children or adults and adolescents, 13 to 35 years, with Down syndrome. And the primary outcome of this study is safety and tolerability, but they're also using cognitive, functional, and behavioral paradigms to understand how this drug affects this patient population. And they're also using quantitative EEG and auditory event-related potentials to measure this drug's function. However, this clinical trial is based off of some preclinical work out of Stanford that looked at low dose, or identified that low dose PTZ rescued cognitive benefits in Down syndrome in 2007. And then they also showed that using quantitative EEG, the Down syndrome mice had high gamma power on 24-hour video EEG, and that PTZ reduced this to wild type levels. And this is interesting because if you're familiar with our work, earlier this year we published in Biological Psychiatry that these mice, the Syngap1 mice on 24-hour EEG, also had high gamma. They had high gamma during sleep. You can see here, this is gamma power plotted. Yellow is wake, non-REM is blue. Gamma stays high during non-REM in adult Syngap1 mice. And then when we gave an AMPA receptor antagonist, this reduced gamma power and allowed gamma power to be modulated from wake to sleep back to wild type levels. So since we initially showed this in the adult mice, we're now interested in younger ages. And so here is a P24 experiment that is a juvenile age of a wild type, which has this, again, this high gamma power during wake, low gamma power during sleep, and modulation over 24 hours on video EEG. This het mice has no modulation from, or very little modulation from wake to sleep, as you can see in this expanded transition from wake to sleep here. And then when we give this AMPA receptor antagonist at low doses, you can see we can rescue that to wild type levels. So this is at P24. So this is juvenile ages with a head cap experiment showing what we previously have described in the adult Syngap1 mice. However, since sleep structure changes over time and over age in mice, we ask the question, is this high gamma during sleep dependent upon sleep structure? Or if we were to normalize the amount of time it was awake before, the mouse was awake before it went to sleep, would we get a robust response from this gamma? So using a telemetry system, we recorded during 24 hours, and we started off with 6 hours of sleep deprivation, and then the mice wild type or Syngap hets would then sleep. And what you can see here is that when we controlled for this duration of wake, that we got a robust response in this biomarker of gamma power, so 35 to 50 hertz on video EEG, and that the rate of change from wake to sleep for gamma was opposite in hets, and that the overall power is higher in hets for gamma. But importantly, the slow wave activity, or this delta power, 0.5 to 4 hertz, was not different. So even though we gave the sleep deprivation, the slow wave activity after the sleep deprivation was not different. However, the gamma power was. So in summary, these Syngap1 pups have early life seizures, and they are predominantly subclinical. And then some of the ongoing experiments have identified that maybe these Syngap pups have an increased GABAergic tone, and this actually may underlie some of these early life seizures. I've shown this through the phenobarbital aggravating seizures and low dose PTZ actually decreasing seizures in Syngap1 hets. And then further, the recently reported biomarker of high gamma, so 35 to 50 hertz, which we had shown in our paper published this year, is is present at juvenile ages at P24 in these Syngap1 mice. I would like to acknowledge from the Kadam Lab, obviously Dr. Shilpa Kadam made all of this possible. Pavel Kipnis was a great lab mate, and Simon Emanuel, who's a lab alumni and collaborator who's at the Chang Lab at UCSF, and also Dr. Richard Hugener's lab, specifically Dr. Haraki and Dr. Hong, who allowed me to start my own experiments by generously giving us a portion of their Syngap1 colony. That obviously expedited my timeline to getting new results, and so thank you to them. Also, I would like to thank all Syngap1 stakeholders and the organizers of this virtual conference for young investigators for allowing me to speak, and I look forward to the discussion and questions in the future. So thank you all.
Hey everybody, thank you, Brennan. And just to remind the audience, I'd like you to bring up your Slido and go ahead and log in to submit your questions. And we will wait for just a second for you to get that app up and send us some questions. And we're going to go ahead and welcome Brennan. Thank you so much. That was an incredible presentation and exciting new research. And congratulations on your publication. That was out. And we're going to have a couple of questions for you. And we actually already have one that's coming in. And this is from Dr. Jimmy. And he asks, this is given the impact of early life seizures on plasticity, have you considered trying to give another anti-epileptic to quell seizures, then measure the phenotypes later?
Right, so I think that some of the data I showed today shows what I was doing to get to that, right? So I was looking at phenobarbital, which is, I kind of talked about its mechanism in action, and it's a very intense, like it was a loading dose of phenobarbital. So those are some of the data I showed today showed that I was kind of investigating some of these anti-epileptic drugs acutely while on EEG at a young age. We are interested in looking at how can we stop seizures over a long duration of time. And then when we're looking at those anti-epileptic drugs, what are the doses that we want to give so that we can really piece out the idea that they're anti-seizure, but there's always going to be side effects of the anti-epileptic. So we're interested in that kind of interaction going forward. But right now we're trying to look at the acute effects of the drugs because that really is what sets us up for their efficacy over development. So that's the first thing we're looking at. And we're trying to get a good understanding of that. And then what types of drugs do we want to look at? And is it very similar mechanisms of action, or do we want to look at very different mechanism of action? And then we're trying to tie that to the acute EEG now. So definitely something we're thinking about. And I think it's kind of one I'd like to describe why I was doing those experiments, and they're kind of positioned to that question. However, we have to figure out more things, I think, that's happening at the really young ages before we start the chronic long-term experiments. And then the next thing I will say, which I think is what is the functional outcome in development for the Syngap1 mouse, right? Is it seizures? Is it behavior? Or is it sensory processing? Is it a mixture of all three? Those are things that we're looking at, and meetings like this help to guide that thought process.
Absolutely. And we have a question coming in from Dr. Connie, and she asked, have you looked at earlier ages, and is there time when the EEG is normal?
Earlier ages than P7?
Yeah.
On the EEG? Yeah, so I've recorded from P5 animals before. They were not Syngap pets. So, but what I will say is that I do this, the breeding of mice, and the way this works practically is that they can, have a litter at a time of day that's probably plus or minus 12 hours. So is there a time earlier? No, I haven't gone earlier than P7. And that's really we're guiding by what P7 means developmentally, at least this is kind of like a thought process. But what I will say is that in that graph that I showed with at P7, male, female seizure burdens early on in life, is that there are P7 hets who don't have seizures, right? And so that's kind of, I think when we put this up, this data up online so that, it was kind of open for everyone to look at. And what we wrote in there is we think that what this means is, even though we have the Syngap1 het pup, is that if it's not having seizure burdens over 2 hours, well, when I do these repeated recordings and I can control for a lot of variables in mouse research, it says that we need longer, early in life, we need longer EEGs, right? And that would be beneficial for seizure identification because even in the same exact mutation mouse, right? So this is when we look at genophenotype broadly considered, even in the same exact mutation, right? When I go to do my experiments for seizures, I could have someone that's having tons of seizures and obvious, interictal spikes. And then you can also have that same, phenotype may not be present at P7, but when I go to P12, now that mouse may have tons of seizures. And so they almost look two functionally different days. It's almost two functionally different, EEG kind of analysis or score that you'd give for that one day.
Oh, wow, that's incredible. And I think if you had, we are at 10, and I think we could talk about this all day, Brennan, because this is just fascinating because what you're describing is exactly similar to what we experienced in the Syngap community. It seems like progressing along when they're younger, not finding any seizures, and then they're worse, a couple of years later. So I think this is hugely relevant and great work on what you're doing. And for the audience, if you have more questions, remember, just go ahead and put those in the feedback comments, and we will field those to Brennan if you have them, and we'll just continue the discussion as we move along. And thank you so much for presenting today. Fascinating work and great work, and this is just phenomenal progress that you've made, and you should be very proud, you and your team. So thanks so much.
Definitely a team effort, and thank you for having me.
Yes, thank you. Okay, we're going to go ahead and let Chuck introduce our next panelist.
Okay, so our next presenter is, and once again, I apologize, Serini, ahead of time if I'm mispronouncing your name, is Serini Sumathapala. She is a graduate student from the University of Miami. And I've wanted to say this all day long. I don't think I've ever thought I would say the words mutant and zebrafish in the same sentence. But she is going to give us a presentation on modeling sensory-induced hyperactivity in Syngap 1AB mutant zebrafish. Take it away, Serini.
Good afternoon, everyone. I'm Serini from Julia Dahlman's lab. Today, I will be presenting my work on sensory-induced hyperactivity associated with Syngap 1AB zebrafish models. A human brain has billions of neurons, and many of them fire at a given time. In order to maintain normal brain activity, it is important to have excitatory and inhibitory balance. If excitatory inputs exceed inhibition, one might experience epilepsy-like phenotypes. If the inhibition is increased compared to excitation, those individuals might experience sensory deprivation. This is a schematic representation of an excitatory synapse. So many of these proteins that function at this postsynaptic density are known to be associated with ASD and ID. One such protein is Syngap. So when the neuron is at rest, Syngap inhibits these small GDPases, such as RAS. But when the neuron is depolarized, there will be an influx of calcium ions, which would activate CAMP kinase 2. CAMP kinase 2 will in turn phosphorylate Syngap1, so that is the activated version of Syngap. So activated Syngap can no longer inhibit these more GDPases. As a result, we get increased ERT signaling, which in turn lead to increased translation. And also we get AMPA receptor traffic into the membrane. So this part is very important for synaptic modulation associated with learning and memory. During the last decade, a growing number of loss of function mutations in Syngap1 have been discovered to cause ASD, and ID, and these mutations span throughout the protein. With that, we ask the question, how do Syngap1 mutations impact nervous system development and behavior? In our lab, we use zebrafish to study to answer these questions. So why zebrafish? Zebrafish have sequence similarity and conserve genes with humans. They also have ex utero fertilization and optical clarity, allowing us to study early developmental processes. We can also use comparatively inexpensive gene editing techniques such as CRISPR-Cas9. Given their high fecundity and small body size, and also highly conserved stereotypic behaviors, make them a good model to carry out these high throughput behavioral assays. With the available tools, now we can do whole brain activity mapping to identify brain regions associated with different behaviors. So throughout this talk, I will be referring to Syngap1A and Syngap1B. That is because zebrafish have two Syngap1 genes. That is because Stelios went an additional genome duplication event, thereby retaining 2 copies of Syngap1 gene. When we look at the proteins in both Syngap1A and B, they have the same domain structure as we can see in mammals. So we use CRISPR-Cas9 genome editing to create these loss of function mutants, and these red bars represent where we find the mutations. A lot of work has been done in the past few years trying to identify different mammalian isoforms and their respective functions. So this work inspired me to look at what are the zebrafish isoforms and annotate them. So the way that I did was I obtained zebrafish protein sequences from NCBI. Those included both published and predicted sequences. And then in order to identify unique and common exons, I used UCSC zebrafish genome browser to test whether they are expressed in zebrafish. I further tested them against using expressed sequence libraries such as ESTs and TSAs. After that, I was able to identify 5 different Syngap1A isoforms and 7 different Syngap1B isoforms. Similar to mammalian isoforms, both these genes show variations in their C and N termini, and their core exonic regions remain somewhat intact. Interestingly, I was also able to identify a Syngap1B isoform, which is this orange one, which has over 95% conservation with well-studied mammalian alpha-1 isoform. So this isoform also had a putative PDZ domain, suggesting that this might be the isoform that is enriched at the excitatory synapses of the brain. So just like with the mammalian models, after coming up with this figure, it made me wonder how differentially expressed these isoforms are, where are they expressed, and what are the functions that they serve, because we have 2 different genes here. Okay, so trying to understand what are the phenotypes associated with the models that I have. So since human Syngap1 variants experience sensory-induced hyperactivity, I wanted to see, using zebrafish, whether they also exhibit similar kind of phenotype. So in order to do that, we placed Syngap1AP heterozygous larvae and wild-type larvae in a 96 well plate, and then we placed them in a behavioral chamber with a camera to capture larval behavior. So first, we gave them, we dark adapted them before exposing them to 5 minutes of lights on and 5 minutes of lights off. So this is what the output would look like. So to your left-hand side, you will see wild-type larval activity, and to your right-hand side, you will see heterozygous Syngap 1AB larval activity. So this is a speed video at 16x. And also you will be able to see two different distinct phases where there is less activity and then there is increased activity. So yeah. Sorry about that, that freezes for a bit. See, this is low activity levels and this is high activity levels. Okay, so then we quantified this activity that we see. So in these graphs, always blue data is coming from wild type, red is data coming from Syngap 1AB heterozygous larvae. So y-axis, it's always time, and then we graphed mean distance moved by a larvae during light on periods and then during light off periods. So one feature that I would like to point out that, so these three are coming from three independent trials, but you can see during dark phases in all three trials, the activity levels are much higher in heterozygous larvae. However, during lights on period, it varies a lot, the activity varies a lot. We think this might be due to a batch effect. Given these observations, we are currently working on identifying possible levels at which Syngap1 function in sensory motor circuits in zebrafish. In order to help address that, we are working on whole brain activity mapping to identify the regions that are associated with these visual stimuli and whether there will be any differences in brain regions or activity levels in Syngap1 larvae compared to wild type. Okay, given that ERK pathway is downstream of Syngap1, I would expect to see increased phosphorylated ERK in heterozygous mutant compared to wild type larvae. So, so this is what it's going to look like. So we will stand for p-ERK activity after exposing them to a stimulus, and then we also would stand for total ERK, and then we will normalize p-ERK with 2-ERK, and to get the significantly regions that are significantly different when, like, in the presence of a stimulus. To further test loss of functions in GAP1 models in terms of their learning and memory, this is another experiment that I'm working on. So this is a habituation assay based on Owen Randler's work, which uses a dark flash as a stimulus. So here, this graph shows how habituation occurs in wild-type larvae. So from this assay, I would expect Syngap1 heterozygous larvae to show reduced habituation or no habituation due to their altered synaptic modulation circuits. So to summarize, Syngap1AB zebrafish models of AHD-ID will help understand roles of Syngap1 in development of the neural circuits, which in turn will help us address the mechanism underlying ASD-ID linked behavior. So with that, I would like to thank Dolman Lab members and zebrafish facility manager, Ricardo, for his support. And I would like to thank the funding agencies, NIH, NICHD, and University of Miami Department of Biology for all their funds. Thank you for your attention, and if you have any questions, I'll take them. Thank you.
Thank you, Serini. Did I say that right? Is it Serena? Serini. Okay, awesome. Well, before I continue introducing you, I would like to remind the audience that if you go to the Slido on your app, and you can use your camera to use the QR code and hop right on in and then send your questions to our panelist, Serini. Thank you so much. This has been an incredibly fascinating presentation. And of course, you know, as I'm watching and you're talking about the light and hyperactivity, and I'm just going to throw this in while we're waiting for some questions. In my observation, is that just thinking of my own experiences, even with my own son, he runs from light, and he will tell us, you know, light, turn it off. So, you know, of course, I'm thinking in my head, what kind of connection is this? Because, you know, I have to say they're kind of hyperactive at times. So, but no, thank you so much. Real quick question coming. Was there anything really surprising about anything that you found in this particular study that you did? That just kind of threw you?
Yeah, so it was interesting that different batches have different effects. So this effect, you know, it was consistent throughout like dark phases, but it was not consistent throughout like light phases. So it is just surprising to me, like why that would be, because is it a batch effect or is the circuitry? So that's a mystery there, but that was interesting to like really see.
Okay, so what do you think the correlation is between the light and the hyperactivity? What would your theory around that be?
So the thing is like, we're still trying to figure out what is the real connection between, like, you know, because light is, sensory input. What we're seeing is like, you know, increased movement. So we're trying to link, like, what are the middle circuitry that is impacted by this? So is it just, circuitry associated with the movement, or is it just sensory, integration centers, like, for example, to sensory to motor? So that is, we're actually stuck there because I can't honestly tell you, like, that is our question. We're trying to figure out, like, what exactly that is happening there.
Another question. Of course, you're doing the sensory type study. Do you think there is any relationship to seizures? Because, many of the patients that we see with Syngap 1 actually respond to the flashing lights, which induce the seizure activity. Have you looked at that component, or have you possibly gone down that path to look at that kind of scenario?
So we are trying to do it, but I have not looked at it at the moment.
Okay, awesome. Well, we have another question for you. We're loaded full of questions. Jimmy asks, the activity assay seems amendable to high throughput screen. Has the activity assay been used by others for drug screens?
Yes.
Okay. Can you expound on that? Do you know in comparisons what drug screens?
So it's not have been used for like Syngap 1 related ones, but for other drug screening, they have been used because that's one advantage of having, tiny animals in this 96 well plate, because it's always like high throughput. So for epilepsy studies, and then, a lot of drug screens, they use this method, but not for Syngap in particular.
Okay, awesome. Well, is there anything else that you would like to add about maybe next steps and what you're going to be? Well, I mean, I don't want you to tell your trade secrets, but but something that, a path down in which this might take you and towards therapies kind of the next step.
Yes, so you will hear from David James from our lab how he's studying, Syngap 1 and gut motility and stuff. So the newest finding from this presentation is that we were able to go in and look at different isoforms. So apparently we were able to find a isoform that is pretty similar to human and mouse one. So I would like to see where these, we have a lot of isoforms based on the database. So we would like to see where these are differentially expressed, whether they have, in zebrafish, we have 2 genes to begin with. So from these 2 genes, we have so many different isoforms. So there must be a reason why we have so many isoforms. So it would be really interesting, find out what acts in where, what contributes to what function in the future.
It definitely is fascinating. Well, this is just an incredible presentation and very enlightening. I would have never thought to put together hyperactivity and light. That's just phenomenal. So thank you so much for all the great work you're doing. And any other questions? Yes. Well, and I hope to see you in person soon. We've got to get out of this COVID stuff. But thank you so much. This was great research, and I'm looking forward to what you're going to have in the next couple of years, and you can present it to us, and we'll have questions fielded to you that come through later, and we will be in touch and looking forward to it. Thanks.
Thanks, Monica. Thanks for giving me this chance. Yes.
Thanks. Okay, guys, we are going to move in to our next panelist, and I'm going to hand that over to Chuck.
Okay, well, I am pleased to introduce David James, and he's going to give us a presentation on the gut-brain axis, modeling gastrointestinal distress in genetic forms of autism spectrum disorder. So with that, we're going to be going to David's presentation, followed by the Q&A. And let's take it away, David.
Hi, everyone. Thanks for listening in. My name is David James. I'm a graduate student in Juliet Dahlman's lab at the University of Miami. I'm going to be talking about some of my recent work on modeling GI distress in autism. So I think autism spectrum disorder is probably best understood through its diagnostic criteria. Disturbances in communication, restricted or repetitive behaviors, intellectual disability. But in addition to these behavioral issues, there's a whole slew of comorbidities or co-occurring symptoms, including the one that I focus on, gastrointestinal or GI distress. GI distress occurs anywhere from 50 to 75% of the time in individuals with autism, and can be really variable in its intensity. Anything from something as mundane as nausea or upset stomach to something as severe as cyclical vomiting, recurring cases of diarrhea or constipation. So I look at GI distress through the lens of 2 ASD-related genes, SHANK3 and Syngap1. I think that using genetically defined forms of autism offers a really good foundation for research that's aimed at answering questions about possible mechanisms for some of these GI phenotypes that we're seeing. So before I get into some of my data, I do want to give a little bit of background information. I think this is probably a figure that everyone's very familiar with. We're certainly all familiar that we have a brain, and that it's comprised of many billions of neurons, which collectively have many trillions of synapses. And at these synapses, there's a collection of players that impact form and function. Again, the 2 that we focus on are Syngap1 and SHANK3. These are postsynaptic density proteins involved in anchoring other players in place and making sure that the receiving side of the synapse is functioning the way it should. I think traditionally, and certainly by way of the majority of the research, our understanding of these genes is under the context or within the context of the CNS. But we have this secondary brain, as it's kind of frequently termed, in our gut of the entire nervous system. And it functions off a lot of these same players and can actually function autonomously without any input from the brain. And then on top of that, recent work has shown that a lot of ASD genes, the the expression levels of these genes is actually upregulated in gut epithelial tissue, so non-neural tissue. So Sauer's paper in 2019 shows upregulation of these genes, including SHANK3 and SYNGAP1, within gut epithelium of mice. So there is in all probability roles for some of these genes that are non-neural. So we use zebrafish as our animal model of choice. I think in addition to kind of all the typical benefits that animal models provide researchers, zebrafish have the nice benefit of being transparent for the first couple of weeks of development, which makes studying things like the gastrointestinal tract really easy. So you can see in this magnification in the bottom half of the picture with the adult swimming around up top and magnified image on the bottom, the gastrointestinal tract doing its thing, going through peristaltic muscular contractions, really rhythmically moving food through its digestive tract. This large black mass is called the swim bladder, helps the fish maintain buoyancy. So I'm broadly interested in the gut-brain axis, how these bidirectional interactions between the gut and the brain might be responsible for some of these GI phenotypes that we're seeing. Specifically today, I'm going to be talking about how mutations in these two genes, SHANK3 and SYNGAP1, might impact GI function. So one of the earlier things that we saw was a change in the rate and the strength of these peristaltic contractions. Peristalsis, again, is rhythmic muscular contractions that moves digested food matter through the GI tract. So we were able to write a program that was able to record and measure these contractions. And what we found was differences in both SHANK3 and SYNGAP1 mutants as compared to wild-type zebrafish, but for different reasons. So you can see the SHANK3 mutant, the rate and the strength of these peristaltic contractions is reduced as compared to the wild-type. But the SYNGAP fish has a more regular rate of peristalsis, but the strength of the peristalsis itself is actually reduced. So again, SHANK3 is really the only fish showing a change in rate of peristalsis. And the SYNGAP fish don't show any significant change as compared to wild type until we look at digestive transit time, or the amount of time it takes these fish to move food through their digestive tract. So in order to get at that, we fed these fish little fluorescent microspheres and tracked how long it took them to pass these spheres through their digestive tract. So on the left, you can see a typical wild-type fish. Again, really robust, rhythmic muscular contractions, peristalsis moving these beads through the digestive tract. On the right, you have an example of a SHANK3 fish. I unfortunately don't have any videos of a SYNGAP fish. But what you see is a drastic reduction in the rate at which these beads are moving through the digestive tract. So in dividing the digestive tract of these fish into 5 distinct regions, dark blue being the pharynx or the throat, orange being the intestinal bulb or the stomach, cyan being upper intestine, yellow being lower intestine, and green being expelled, we were able to track and record how long it took these varying genetic backgrounds to pass these beads. So for these graphs, what you can see is varying colors corresponding to the region of the GI tract. Each color has 3 different columns for the 3 genetic backgrounds, wild type, SHANK3, and SYNGAP. And then you have 4 different graphs representing the 4 different time points that we looked at: 3 hours, 6 hours, 12, and 24. So you can see in the 3-hour period, all the fish, irrespective of their genetic background, the majority of the beads exist somewhere in between the intestinal bulb and the upper intestine. If you carry this timetable out to 12 hours, you can see that only a few of the wild type fish had any beads remaining in the intestinal bulb or the stomach, whereas the majority of them were actually already expelled. But the 2 mutant models, the SHANK3 and the SYNGAP fish, were having a much harder time moving the beads from their stomach into their upper and lower intestine and eventually expelling them. So the rate at which these 2 mutants, the SHANK3 and SYNGAP mutants, were able to pass these beads was drastically reduced as compared to wild type. So we kind of wanted to get at the question of why these mutations might be impacting GI transit time through some potential physiological experiments. And one of the most approachable ones that we had on hand was just to do some dissections and some staining and look at different cell populations and see what might be changing. Our initial hypothesis was given SHANK3 and SYNGAP's role in the CNS, this should be a pretty clear-cut case of alterations in the enteric nervous system causing digestive problems. So we stained for HUC, which is a basic neural marker, and then we also stained for 5-HT or serotonin. This was to mark an endodermally derived cell line called enteroendocrine cells. This is a really fascinating subset of cells that I could probably spend my entire time talking about. Suffice it to say, they're secretory cells, sensory cells. They interface with the enteric nervous system. They change their morphology based on what you've eaten, so they tend to pull back when you eat high-fat diets and extend, become a little bit more sharply shaped when you're not eating high-fat diets. So a really interesting subset of cells that plays a lot of interesting regulatory roles within the GI tract. So we wanted to look at that. Contrary to our hypothesis, what we found was that there isn't really a significant difference in the number of enteric neurons within the mutants as compared to the wild type. So if you look at this top portion of the graph, the Syngap fish are on the right, and then we're comparing wild type to heterozygous and homozygous SHANK3 mutants. So slight downward trend in the number of enteric neurons in the SHANK3 fish as compared to wild type. Not really that robust a relationship with the Syngap. Where the significant differences did arise were down here in these enteroendocrine cells. Again, these are endodermally derived cells. So it's really interesting and kind of perplexing as to why mutations in genes that are traditionally understood to play roles in the central nervous system or in the nervous system in general might be impacting endodermal non-neural tissue. And we see a reduction in these cells both in SHANK3 fish and Syngap fish. So this is a shared phenotype that is pretty interesting. So this obviously leaves us with a slew of more questions unanswered than answered, particularly what kind of implications this might have for future research for us, and how the CNS might or might not be involved in these GI phenotypes or GI regulation in general. And we're looking to answer these questions through another cool set of experiments, tissue transplantation. So it's another advantage of using zebrafish is you can take cells from developing embryos from a donor embryo, transplant them into a recipient embryo, and have these distinct cell populations grow up. So there's been a ton of work to date done on getting fate maps for various regions. This figure here is from Catherine Wu's paper with Fraser that maps out brain regions in the developing embryo really well. So we're able to take these cells from one embryo and inject them into another and get these nice chimeric animals. So these animals are otherwise mutant, so either SHANK3 or Syngap mutant fish, but they have these fluorescently labeled wild type tissue subsets within various regions of the brain. So with this experiment, we can start to poke at questions about cell autonomy, about how different substructures in the brain might impact other regions of the body. There are a ton of regulatory regions in the hindbrain that play a role in GI regulation. So to be able to go in there and restore the function of either of these two genes, SHANK3 or Syngap, really gives us a powerful tool for figuring out what kind of impact it might have on other organ systems. And this obviously leaves the potential for it to not have any impact, which would still be very telling, meaning that these GI phenotypes were arising independently of any issues in the CNS. So briefly to kind of conclude, I want to bring up a few key points. One, mutations in genes traditionally understood to be CNS specific are having impacts on non-CNS tissue. These changes in endodermally derived tissues, these changes in these enteroendocrine cells, point to a possible non-neural role for genes like SHANK3 or Syngap1, and could certainly be a source for the GI distress that we're seeing really frequently in autism. Future transplantation experiments will obviously be needed to kind of figure out how or if the CNS is involved in these GI phenotypes. It's something I unfortunately didn't get time to talk about in depth, is the use of these model systems, be it rodent or be it zebrafish, are really helpful in determining what kind of medications can be used to not only treat behavioral issues, but not worsen other problematic comorbidities. A lot of current drugs on the market aimed at treating behavioral issues within the autism spectrum disorder kind of sphere really have negative side effects on GI function. So to be able to use these animal models moving forward and to remember to also check for other problematic comorbidities is really an important part to remember and keep in mind. With that, I'd obviously like to thank everyone in my lab and people who have collaborated and contributed to our work thus far, funding sources included, and I'm happy to take any questions.
Great presentation, awesome. I wanted to remind the audience really quick to go ahead and get your phone out and go into Slido and ask your questions, and we will definitely field them to David. David, I want to thank you for being here. Great presentation, fascinating. By the way, I like your little Zoom background. Your little zebrafish back there. No, thanks so much. This is really fascinating. And of course, you know, we're really super excited about working with you guys on the gut study and learning a lot more about how the brain and the gut work and function together, because as you know, with the discussions, of course, that we've had, has been truly fascinating and very relative to our community. So my first question to you is what did you find surprising? What was the one thing that you found surprising about your study that maybe you didn't expect, or kind of the outcome that you had?
Yeah, I think I had briefly talked about it, but just to expand a bit, you know, our first hypothesis was that this definitely had to do with this enteric nervous system, this nervous system that controls your gut. It makes the most sense in terms of what we knew about the genes, be it SHANK3 or Syngap, and their role in the nervous system. Mm-hmm. So to have non-neural effect was really bizarre for us in terms of findings. To see changes in serotonin expression or in the number of these enteroendocrine cells would imply that mutations in these neural genes are having impact on non-neural tissue. That kind of crossover is not the most common thing to see, but I think what it really highlights is just that we don't know a lot about these genes yet. Mm-hmm. So that's kind of exciting to be able to study something that's that new and kind of has that many unknowns.
Yeah, that it is definitely is a fascinating unexplored territory, and I believe personally, and I'm not a scientist. This is just going on a mom gut. Excuse the pun. This is very relevant to especially autism spectrum disorders. Okay, so we've got some questions flooding in. Are you ready? Okay, so Jimmy asked, I assume zebrafish have a gut microbiome. Does the altered gut transit in these fish result in the altered microbiome?
Yeah, I think this is going to be a common trend with my answers is we're looking at that right now. So I don't have answers for that right now. A lot of similar work from other labs outside of the autism sphere. So research on microbiome that doesn't have to do with autism shows that motility rates definitely have an impact on the type of bacterial populations we have in our gut. A lot of these bacteria are really dependent on these kind of peristaltic flushes, so to speak, to remove excess amounts of bacteria. So yeah, we're definitely expecting to find some differences there. We have a new grad that just joined our lab that's looking at the microbiome.
Awesome, awesome. That's going to be really super interesting as you move forward. Okay, so we have a question coming in from Dr. Connie Smith-Hicks. It says, patients with autism are often reported to be intolerant of some foods, and many have tried elimination diet. Have you looked at the impact on nutrients on motility?
We haven't yet. We wanted to get initially early on in my beginning of the study, we wanted to look at different rates of uptake, whether it be carbohydrates or fats or that kind of stuff. We haven't looked at that in depth, unfortunately, yet. One thing that we did notice when we were doing these feeding assays to kind of map out the peristaltic rate is we initially tried to do those with standard fish food, and we found that the fish weren't really eating as much as they should be to get a really robust assay. So we actually switched over to a high-fat chicken yolk based diet, basically. So, and they seem really, really excited about that. So preference to certain food types with these larvae, there seems to be something there, but it's nothing we've looked at in depth yet. So it's all kind of hypotheses at this point.
Awesome, awesome. Well, I mean, these are really some great aspects because just out of curiosity, I know that Dr. Dahlman and I have had this a brief conversation about the zebrafish and actually what they eat, because what's very very interesting even about our the Syngap patient group is that we have children that will be super sensory to like texture and they won't eat. And some of our children are actually on G tubes. I mean, because they just don't have that, either the swallow mechanism to be able to get food down, or they choke. But then we have kids like my child, and we've had this discussion even in our Facebook group about how some of the kids just gorge food. And it's like, if you don't, and I don't, mean to, I guess, say this, but it's almost like a Prader-Willi type of behavior where it's just like they don't know how much food they're actually eating, and it's like they will eat and eat and
Sure.
eat. Do you see that in the zebrafish?
It's not necessarily that we see that, but there is an explanation for that, at least a hypothesis for that. The reduced serotonin staining in these enteroendocrine cells is particularly relevant because these cells are mechanosensory cells in the gut. They're stress receptors. They're what tells the brain that we're full or we're not full. So dysregulation in this particular cell type might be the reason why some kids are really, really apt to overeat, so to speak, whereas others are not. So there's definitely some kind of miscommunication going on between these sensory cells in the gut and the brain. We're just not sure what exactly that means right now.
I see. Now, kind of the same thing that I asked earlier in one of the other presentations about, when you look at these fish, of course, you have the control. It's typically the same mutation that you use. Are these fish coming from the same mama and dad, and do you notice a difference in the spectrum of the fish? I mean, because you think they'd be the same if they're genetically the same, but do you see those uniquenesses in that spectrum as well in the sibling, or their litter mates, or I don't know the school,
Yeah.
the school of fish?
Litter mates works. So yeah, there's always a bit of batch effect. I think Sereni was talking about this briefly as well. There's always variations between individuals. We obviously try to control for that by using multiple batches and by getting averages between those batches. Kind of poking at the idiosyncrasies and the differences between individuals becomes difficult in any animal model, whether it's zebrafish or mice. But there's certainly some aspects that we can look at that we just haven't really touched on, but we try to control for batch effect as much as we can. Yeah.
Okay, and this is going to be an odd question too, but do you see constipation in the zebrafish?
Yeah, absolutely. Yeah. In fact, there's a medical term, at least with the shank 3 fish, there's a clinical term for humans called megacolon, where they become so constipated that their colon actually distends and they get really a lot of buildup, and they can actually get diarrhea from that because they're so clogged up that it's just going around this blockage. So we do see that from time to time in individual fish. Constipation and kind of this reduced digestive transit time are very similar on a biological level. So it's sometimes it's difficult to compare the human case and animal models, but yeah, we definitely see similarities for sure.
Yeah. Well, I mean, for our own patient group, just to add that in, is we've seen that where some of our children have had to go to the hospital to actually have help because it just stops. And so this is absolutely fascinating, and I'm just so excited. And I know our patient community is excited about the Smurf cakes, and we're going to be getting into that on Thursday. So it's just been amazing, and let me check my little Slido here really quick, because see if we have any last minute questions. And we don't have any right now, but if you do have a question that maybe has come up later, please add it to the feedback survey, and then we will give it to David and let him talk to you and exchange information. So David, I just wanted to say thank you so much. It's such a pleasure. I wish it wasn't COVID, like I've said with everybody else, but it has been incredible for you to be with us and learn more, because this is just fascinating about how all of this stuff ties together. So thank you so much, and we'll definitely be in touch soon. Thank you. Awesome.
Bye.
And now I'm going to pass it to Chuck to introduce our next speaker.
All right. I am pleased now to introduce our next speaker, who is Yoichi Araki, and hopefully I pronounced that correctly, PhD Research Associate, Johns Hopkins School of Medicine, the Department of Neuroscience. And he's going to talk to us about Syngap isoforms differentially regulate synaptic plasticity and dendritic development. So with that, I turn it over to Dr. Arachi's presentation.
Okay, let's get started. Hello, my name is Yoichi Araki from Johns Hopkins University, working with Dr. Rick Hugnier. And title of my presentation is Syngap isoform differentially regulate synaptic plasticity and dendritic development. As you know, genes are differentially transcribed to messenger RNA, which we call the alternative splicing, and that messenger RNA is translated to the protein to exert the distinct functions. In Syngap1, it is reported that 4 distinct C-terminal isoform was expressed in the brain. In Syngap1-related intellectual disability, many gene mutations were reported so far in Syngap1 gene. Importantly, these mutations affect all Syngap isoforms, both in messenger RNA and protein, to change its function. So it is critical to understand each isoform expression pattern and functions to understand Syngap1-related intellectual disability. This information will provide the mechanisms of the disease and will lead to the efficient gene therapy by unique one Syngap isoforms. This is showing the protein structure of Syngap1. You can see RAS cap domain in the center And C-tail has a commutative phosphorylation site and coil domain, which is required for phase separation of with PSE95. Near C-terminus, there is alternative splicing junction points. Beta is coming from the alternative splicing of exon 17 and 18. Alpha 1, alpha 2, and gamma isoforms are coming from the alternative splicing of exon 18, 19, 20 junctions. To characterize the Syngap isoform expression and localizations, we raised each individual antibodies to specifically recognize each Syngap1 isoform. We saw most of the isoforms are specifically expressed in the brain, and also this region-specific Western blotting shows almost all isoforms are enriched in the cerebral cortex and the hippocampus in the adult brain. We also characterize the temporal expression of Syngap1 isoforms. As you can see, beta and gamma isoforms were expressed early compared to alpha 1 and alpha 2 isoforms. And this expression is replaced with alpha 1 and alpha 2 expressions later. We calculated the composition of Syngap1 isoforms. As you can see, most of well-studied alpha 1 expression comprise only 25 to 35% of Syngap1, highlighting the importance of study all isoform to elucidate Syngap1-related intellectual disability. Again, beta is well expressed around the birth, and this is replaced with alpha 1 and alpha 2 expressions later. We previously reported that Syngap alpha 1 isoform is interacting with PSE95 to form condensed phase in synapses to provide a physical basis of postsynaptic density. So we investigated if the other Syngap isoforms have similar functions in the cells. We transfected each Syngap 1 isoforms with or without PSE95 in hex cell and fractionated into soluble fraction and a pellet phase separated condensed fractions. This is a graph showing how much protein is going to phase separated condensed phase in the cells. As you can see, when I expressed Syngap alpha 1 with PSE95 here, most of proteins are going to the condensed pellet phase, reconfirming the previous observation in 2016. Interestingly, the other isoforms, especially beta isoforms, are more going to the soluble fractions and less going to the condensed phase, suggesting the distinct biochemical properties of Syngap1 isoform in the cells. Reflecting these distinct biochemical properties in the cultured cells, the in vivo brain localization of Syngap1 isoform are also distinct between Syngap isoforms. We fractionated the mouse brain into S2 cytosolic fraction and postsynaptic density PSD biochemically. As you can see, Syngap isoforms are enriched in PSD fractions. Among these, alpha 1 isoform is going to PSD fractions mostly among Syngap1 isoforms. Interestingly, reflecting the soluble biochemical properties of beta isoform in the cells, beta isoform is going to cytoplasmic soluble fractions in the brain in vivo compared to the other isoforms. This is a quantification. Beta isoform is actually 30 times more going to cytoplasmic fraction compared to the alpha 1 isoform, suggesting the special role of this isoform outside the synapses. We surprised that, in contrast, extremely low existence of Syngap alpha 1 isoforms in cytoplasmic fraction. This extremely low ratio is much lower than any other famous postsynaptic density molecules such as PSE95 and AMPA receptors. Syngap is a last gap protein where this hydrolyzes last bound GTP to become a GDP, and this makes RAS inactive. And it is also reported Syngap has gap activity for other small G proteins such as LAP1 and LAC1. So we wanted to know the specificity of each Syngap isoforms to inactivate these small G proteins. We co-express Syngap and small G proteins and how much active GTP-bound small G proteins are decreased by Syngap isoform expressions. For example, active LAP1, as you can see here, is decreased by alpha-1 expression, but you can see more dramatic decrease in beta expressions. This is a quantification here. You can see the beta has generally maximal gap activity towards all small G proteins we assayed, including RAS and LAP and LAC. It is interesting that alpha-1 isoform has relatively low gap activity but has preference towards RAS gap. We previously reported that Syngap and PSE95 are postsynapses at basal state, as you can see here. But once NMDA receptor and CAMK2 was activated, CAMK2 phosphorylates Syngap, and this dissociates Syngap from PSE95, inducing a rapid Syngap dispersion from the synapses. And we found this dispersion is essential for AMPA receptor insertion and the synaptic enlargement during LTP. So we wanted to know if the other Syngap isoform has similar functions for synaptic plasticity process. We expressed AMPA receptors and EBCHERI, who are the marker for the synaptic morphology. You can see the AMPA receptor is inserted and spines are enlarged after chemo-ATP inductions. For example, you see very small spine here, but this is enlarged after chemo-ATP. You see larger spine here compared to the basal here. And also, you can see rarely AMPA receptors at the basal state, but you see the AMPA receptors after chemo-ATP. And you can see in quantification, spines are enlarged after chemo-ATP and AMPA receptors are getting inserted after LTP. And as previously reported, these spine enlargement and AMPA receptor insertions are both occluded when I knock down Syngap by shRNA shown here, suggesting that the essential role of Syngap1 was synaptic plasticity. And we rescued this phenotype by expressing each individual Syngap1 isoforms. Interestingly, alpha-1 isoform is successfully rescuing this LTP occlusion. Alpha-2 marginally rescued this, but all the other isoforms cannot clearly rescue the plasticity deficits. This indicates the exclusive role of alpha-1 for the synaptic plasticity. We noticed that when we knock down Syngap very early in the development at DIV3, the dendritic development is severely impaired in the culture neuron. So we would like to know which isoform plays this neural morphology role. This is a graph for the show analysis, how much dendritic urbanizations were observed at the distal segment. As you can see, you can partially rescue this phenotype by expressing alpha-1, alpha-2, gamma isoforms, but only beta isoform perfectly rescue the dendritic development deficits upon Syngap1 knockdown, suggesting the exclusive role of Syngap beta on the dendritic urbanization at early development. In conclusion, we characterize a variety of functions and biochemical properties of Syngap isoforms. We see different temporal expression of Syngap isoforms where the beta expressed early, alpha 1 and alpha 2 is replaced when they become adults. Syngap beta has maximal gap activity and expressed early and has exclusive functions on the dendritic development. Syngap alpha 1 is phase separated with PSE95 in postsynaptic density and has exclusive role on the synaptic plasticity. These informations are critical for understanding Syngap-related intellectual disability, how these isoforms contribute to the disturbance of the synaptic connection as well as neural circuit formation. I would like to thank you all the people in the project in Dr. Rick Hugnier's laboratory, as well as the collaborators. Thank you so much for your attention.
Hey everyone, I wanted to welcome you. If you have just joined us, I want to remind you that if you'd like to submit questions, please pull up your phone and grab your camera and scan in the QR code for the Slido, and then you can submit your questions for the panelists now. And I wanted to welcome Yoichi. It's great to see you. Great work. How exciting. And just incredible work you guys are doing. And thank you for being here. And we're going to go ahead and start in on some questions for you. And really quick, I've been asking this question to kind of start everybody off. Was there anything really surprising that caught you off guard, or you found that was unique that you weren't expecting during this research that you did?
So I think I'm really surprised that Syngap has a really variety of splicing variants, and that these splicing variants is very, very distinctly, have a function on the neural, development, such as synaptic plasticity and dendritic development. So I really surprised there is a variety of the function of the Syngap.
Awesome, yeah. This is incredible work, and so I just fielded a question, so I've got a couple more questions for you. This is from Dr. Julia Dahlman. Did you look at axon growth as well as dendrite growth with the B isoform rescue?
I'm not sure what the B isoform. B isoform is generally mentioned N-terminal splicing variants, but I suppose this is beta isoform, which is C-terminal isoform. And I generally don't look at the axon outgrowth phenotype in my experiment, because when look at my neurons in the early stage, the dendritic development is so severely impaired when I knock down Syngap. So I determine I am going to focus on the dendritic development first. But maybe it's a very good point. We should look at also axon stuff in the future.
Awesome. Okay, and I have one other question, and that is from Dr. Jimmy Holder. Is there something about the beta isoform protein sequence that you can pinpoint that targets it to a cytosol versus a PSD?
I think that's very good question. So the other isoform other than beta has a complete coil domain, which is required for the phase separation. So that's why they are going to a PSD, but the beta isoform doesn't have full coil domain sequence. They have very partial coil sequence, so that they cannot effectively join the phase separated phase. So I think that's why they are not going to PSD, and they are going to side plasmic fraction.
Awesome, awesome. Well, I fielded those questions real quick, and so what I'm going to do now is thank you, Yoichi, for sharing with us, and I want to go ahead and we are going to bring everybody back onto a panel that spoke this morning, and I would like to introduce Dr. Jimmy Holder, who is going to be the chair of the panel, and he is a research scientist at Baylor College of Medicine and also practices neurology at Texas Children's. And again, I want you to bring up your Slido on your phone, and you can field the questions, and the panelist and Dr. Holder will answer those questions for you for the panelists. Thank you so much. And welcome to the panel. I'm going to bring them in. Hi, how are you guys? Y'all, thank you so much for joining us. This is incredible. Dr. Jimmy, thank you for chairing this panel. We've had some incredible talks today, and I think we're going to have a great discussion. And I will just hand the floor to you.
Okay, great. Thank you so much, Monica. So I want to thank all of our young investigators for speaking today. You all did a tremendously wonderful job. And this is really an opportunity for everyone that's listening to ask any questions they didn't have a chance to, and certainly for our young investigators to ask each other questions, which I think will be really beneficial for all of you. I have a couple of questions I'm going to bring up. I'll check the Slido. Please, if anyone has a question, don't hesitate to ask. I'm going to put a little bit of a challenge to all of our young investigators. Maybe in about 10 minutes, we can go around and just think about, if you've probably already thought about this, but if you haven't, think about it for the next 10 minutes about not just your work, but everything you've heard today, everything you've read about Syngap, everything every talk you've heard about Syngap. And I'd like to hear what you guys think are the major challenges for understanding Syngap biology. And then, of course, for myself as a child neurologist treating kids with Syngap, and Monica and the other parents that might be listening, how can we get from where we are now to personalized therapies for kids with Syngap? So that's the challenge I'm going to put to all of you young investigators to give us the answers. So you have 10 minutes to do that. So no pressure there. So I'm going to start off by just asking a couple of questions that I thought of during the talks I didn't have a chance to ask. The first one is actually for Brennan. So one thing that just very kind of small detail that I picked up on, maybe you can help clarify. You described the behavioral seizures, behavioral phenotypes associated with seizure in P7 mice in the Syngap het versus wild type mice, and showed that one type of seizure, I think it was lordosis and maybe behavioral arrest or something, was similar between wild type and Syngap hets. So are the wild type mice having a significant number of seizures?
No, so the initial graph that we're talking about is behavior when blinded completely to EEG. So those behaviors of what you would, maybe the behavior classification is based off what you would want to be for neonatal behavior. So neonates have specific like ataxia and some of the things that we said there, like the jerks. So that's just their behavior. So that's not actually that graph doesn't show anything towards seizures. That's just describing the behavior on video, blinded to EEG. But what I did is, so if you took all of those behaviors that you would classify on video, and then you took that and then overlaid it to a completely different seizure score, which is with the EEG, then that graph that was below 50% shows that if you were just to score behavior, you have a really low true positive rate of identification. But that's only looking at the true positive rate. So if I showed the inverse of that, which I think would be, which would really get at your question, it would show that when you just score the behavior, you score a lot of the behavior that has nothing to do with any seizure activity at all. So that was what we were looking at in that specific graph. But none of the wild types have seizures. And but my data is also, we put it up as like a preprint so that people could look at it. And there is like an hour-long trace on that, which may be of interest if you want to look at it. And you can see that the wild types do have like delta brushes, which is something that you can see that's like a high amplitude activity. It looks very different than seizures, but it does show some of the small differences that are in the EEG they're just reality of recording.
Okay, terrific, great. And then my next question is for Yoichi. You mentioned that the beta isoform appears to have maximal gap activity, I guess, compared to the other isoforms.
Yes.
I'm going to ask, similar to my question about distribution of the different isoforms, is there anything in the protein sequence that you can pinpoint to as to why the beta isoform would have more gap activity than the other isoforms?
I think this is mainly the accessibility of Syngap to the small G protein. Both the small G protein is not phase separated. They are mainly at the cytosol or the membrane tethered. They are never phase separated. And Syngap beta is less phase separated. So there is more chance to meet the Syngap beta to small G protein. So that may be the main reason that the Syngap beta has generally high activity to the gap function. So that's our guess.
Okay. Terrific, great.
On the isoform, since we're on the isoform question, I'd like a question for Cerny. So in your experiments, you did like Syngap A and B, the two isoforms that you have in zebrafish. But I was wondering if what happened if you do them like separately, if you look at Syngap A alone and Syngap B alone, do you have insight on like the phenotype and?
Yeah, so it would be really good to like have these, different genotypes and then look at like variations. But I always noted this problem. I still don't have a single double homozygous mutant fish, like female one. So it has been really challenging to like really get these mixed genotypes. And I totally get your question because I am personally interested in that. Because I really want to see, like, so far I have Syngap 1A double homozygous fish, but I don't have Syngap 1B double homozygous female fish. So it's really challenging to like get all these combinations, but I think like if we can, I'm hoping, one day I would get one fish, but it's been almost 4 years now, so I haven't gotten any. But yeah, in order to identify like how severe these different genotypes and like what are the different functions of each different functions of each genotype, it would be really good to look at like in terms of like understanding. But right now my hands are pretty tied because it's really difficult to get hands on of these different genotypes. Did I ask you a question or?
Yeah.
Okay.
Great, thank you. I think Dr. Dahlman had a question. So I'll open this. It's not directed necessarily towards anyone in particular, I don't think. Perhaps Brennan is a good person to answer this, but I'll leave it for everyone. Can absence seizures be distinguished by EEG?
That's a good question. And that question, I assume, is in the mouse, right? That's my assumption going into
It
answering
doesn't
that.
say so. I can talk about human, but
Yeah, I think it's a good talking point to start with the human. Maybe if you want to take a stab at it first, Dr. Holder, and then I can add.
sure.
Yeah.
Sure. So for patients, absence seizures definitely do have sort of a characteristic EEG abnormality, which is so it depends on the type of absence seizure. If we're talking about sort of typical absence seizures of childhood absence epilepsy, we see 3 to 4 hertz spike and wave activity in the frontal regions. For atypical absence seizures, which is in general, I think what we're talking about with Syngap 1 patients, and I'll present some data about that, I guess tomorrow. They often have a bit of a slower spike and wave frequency of 2 to 3 hertz. And then really, it's a combination though of both the EEG finding and the seizure manifestation. In short, to answer your question, in general, yes, we see some specific epileptiform discharges in patients with absence seizures. And then in mice.
Yeah, so in mice, I think if a mouse was having just this maybe 3 to 4 hertz discharge, but maybe no other type of seizure phenotype, right? That would be very difficult. This is from a practical standpoint. The field is obviously split between absence seizures and spike wave discharges and other atypical absence seizures, the array of that. And I would look towards, there was just a preprint put up by the Peter Kynes group, I believe, and they looked at spike wave discharges, and they have a discussion in there, and they also cite a paper on spike wave discharges by Dr. Dan Barth and Dr. Ed Dudek. And I think that I'd rather point to the literature on this for my response, but it is definitely a point of debate in the field for mouse epilepsy research. But this really is a problem when there's only this, there's no other types of seizure phenotypes, right? So when you have other types of seizure phenotypes, this becomes less of a problem because really you might be describing a poly spike, interictal spiking, or something you may see on wild type. But when you have these overt seizures, like in the paper that we published this year out of our lab, and you look at the seizure phenotypes there, you can see that there's an array of seizure phenotypes that get worse as they get older. So I think when you have that kind of data with it, it's easier to really think about seizure phenotype than it is if you just had something they might call absence, if you believe in that as a preclinical researcher.
Okay, great. So just to clarify, so mice can have seizures that are just behavioral arrest. Isn't that correct?
Yes, I mean, they can. The idea is when you start classifying it within the human context, there's a debate there to be had.
Okay, great, thank you. Do any of our panelists have any questions for each other?
Well, just a comment, more a comment. In hearing all these presentations today, I'm sitting here going, oh, are they talking about my daughter? And the reason why I say that is we started with gait, and so she has gait issues. We talked about senses, so she has sensory issues, an obsession with, like if she gets it wet or drop or any water comes on her, she has to dry it off, or she'll dry it off you if she sees you're wet, by the way. She has seizures. Luckily, they're controlled, like Monica's son, Beckett. And it's when the seizures got controlled that we started to see improvement in her behaviors and learning. Mm-hmm. She has behaviors. She has hyperactivity. She has learning and memory issues. And she has gastrointestinal issues. And so you kind of covered the the gamut there. And then from the last presentation, Dr. Araki, obviously she has Syngap. So it's just an observation that all this research that you're doing is all very relevant to the families. Mm-hmm. Obviously, I don't understand some of the science. It's beyond my pay grade, as they would say. And one question I did have for David, though, is that a mutant zebrafish in the background?
That's actually a good question. I'm not sure. I have so many videos on my hard drive that I couldn't tell you. I just grabbed one.
So that was the one thing that was fascinating me. But I just want,
Chances are probably, yeah.
but I just want to say, you've touched on everything that my daughter experiences as a result of, we're assuming, as the result of Syngap. And each area is very, very important. So if you had any questions, I'd be happy to try and give it sort of that clinical view. What's happening in the real world with day-to-day activity.
So I think I had a question on this sense that ties back to what some of Jimmy's question to me in terms of sensory perception. Is it more like avoidance or is it more like seeking? I think there's a spectrum that goes both ways from some of the registries. So I was eager to get a sense of that in your experience, and also I think one of the things that was that I was curious about was the evolution since your daughter is 18-year-old, so you've been able to observe the evolution across a very long period of time. And I think that's a very interesting perspective in my sense, because a lot of time we think of this in the context of like younger patients, right? When at least the family has seen in the past, it's younger patients. So I was interested in if you can touch upon this a little bit as well, Chuck.
So from a sensory perspective, there's both avoidance and seeking. So she tends to drool, and if she drools and she gets wet, she wants to dry that off. She also seems to have this predilection to wear, she's great with masks, by the way, because she likes things on her face. She likes goggles. She'll wear a helmet. She'll wear a helmet all day long if you let her. She goes horseback riding, that therapy. So she seeks that. Hyperactivity, I guess, is a way of she seeks certain videos and certain activities, and it just ramps her up in terms of that. So she seeks that, and then there are certain things that in terms of sensory, she will avoid. But one thing to notice is when she was younger, she had that sort of pain deficit. She'd fall, and I always remember she'd get up and look at her hands like, oh, what's that?
Mm-hmm.
Something just happened to me, right? But now she's much more receptive to pain. Mm-hmm. She seems to recognize it, has an appropriate response, I guess you would say. Same thing with water. She would sit in the pool until your teeth start chattering, your lips turn blue. When she was younger, not so much now. Now she doesn't seem to be as enthralled with water as she used to be in going in and swimming. And then just from an evolutionary perspective of when she was younger till now, what I've noticed is all incremental. Everything is very, very incremental in terms of she was not able to she couldn't dress herself. She can kind of dress herself now, for the most part. Front and back is still an issue for her. But from not being able to do it to actually accomplishing it. And also if there's a reward at the end, sometimes, she seems to get it right. Sounds like children, right? She seems to get it right. But all very, very incremental, which kind of brought me back to the first lecture I ever heard on Syngap was Dr. Hugeneer in his lab, and about how Syngap is kind of full. Maybe I don't know if I'm getting this right, but your one isn't working, the other one is almost like over full or almost to its capacity, and there's only a little bit room left. And so my little hypothesis was, well, that little room, we're slowly filling it up through behavioral modifications, repetition, 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 and she eventually learns to put socks on. She eventually learns to put pants on, and she gets it all down, like that autistic part. Repetition, you keep repeating it and she gets it. So I hope that's helpful.
Yeah, no, that's very helpful. I mean, to tie this back to the research, or like even some of the design, it's like, okay, that's interesting, because for like some behavior, or like even with the reward aspect, we're like performing tasks with mice, and sometimes they get it, like through repetition, but maybe the latency to which criteria is much longer. And to go back to the evolution and what you're describing, I think it's interesting to see, where the phenotype is really important in terms of like pain and sensitivity, and what happens if we do like the experiments in younger animals or older animals and like incorporate this temporal aspect more. I think would provide great insight.
Great, yeah. So one thing that I think from the discussion that I want to bring back to Monica and Chuck, if you could maybe tell our group for your children, and I think this is going to be expanded upon much more in the FDA meeting on Thursday. What would be the one symptom, if you could only improve one symptom, what would it be for your child, for both of you?
And that's changed throughout, I think, the course of their life, because it's almost like it's incremental because it changes over time, and it's been wild. But I have to say, I think I would take intellectual disability, living and coping with the cognitive deficit that our children have, over behaviors any day. It is because the behaviors actually prevent living a more high quality of life, I guess. And cognition, I think I can deal with. It's like I can deal with the 5-year-old. He can be bribed, but I think the behaviors, I think, would be one. But then that's hard to say, because for me, it's the seizures as well. So it's almost like a catch-22 in a way, because I believe that seizures have a lot to do with the behavior. So, but behavior, if we can get the symptom of what's causing the behaviors under control, that would be huge.
Yeah,
That would be my answer.
Yeah, I mean, my answer would be, well, first of all, the seizures. If you don't control the seizures, you got a lot of issues. Forget the behaviors and everything else. So one would be the seizures, and I know some of our parents have more difficulty with their children controlling the seizures, and luckily we just found the right cocktail of medications and seems to keep it under control. But going to the behaviors, that really does impact their quality of life, because when you get down to sort of the nitty-gritty of it, and you're dealing with state agencies, and you're dealing with school districts, and you're dealing with caregivers, if you have a child with, let's say, very aggressive type behaviors, finding the right caregiver is very difficult. School districts are, finding the right aid. Then you're dealing with, the PPT and the IEP. And then when you talk about transitioning out of the school district, no employer is going to hire anyone with behaviors. They're just not eligible. I'm in Connecticut. I work with the Bureau of Rehab Services in some of the capacities. Behaviors just put them into the unemployable category. Mm-hmm. So from a day-to-day practical, and with the behaviors in control, there's a better opportunity of, like with all our children, we expect them to leave the home at some point, right? To strike it out on their own. Independent living or group homes would be a little bit more of a challenge with that. But the one thing that you may find that I probably would say, if I could correct one behavior right now, and looking at all the behaviors, if I could correct just one behavior, it is stuffing. If it's stuffing her face. And the reason I say that is in nursing homes, one of the leading causes of incident of injury or death is people choking. And I can tell you, you can watch her like a hawk, but you turn your head for 1 second, and suddenly she's stuffing, while she's eating, she's stuffing her face. And so looking at it, yes, let's correct the behaviors, but it might be just, if I could correct one behavior, that would be it. Because I'd have to less worry about somebody watching her when she's eating. That's my big concern. It's very minor. It seems like a minor behavior, but that would be one of the main issues.
Well, thank you both, and I believe this is going to be a big topic for the FDA meeting. It's kind of the core of what that's getting at is for, a larger group of families, what is the one or two things that are most impactful on the family, on quality of life. So I'm going to bring it back to my question. I don't think there are any more questions on Slido. But my question is for the panelists. So what is the biggest barrier right now for understanding Syngap pathophysiology, for developing personalized therapies for kids with Syngap? What do you guys think?
I can take a stab at it first. So I think for developing a therapy, which I think if you look at the type of research I've done over the last 2 years, things that I feel that are our biggest limitation are that a lot of what's known about Syngap, we're seeing that there's these different roles in different neuronal cell types, right? And there's isoform differences throughout development. So from the bench, we're learning more about that every day. But one of the things that's interesting, I think it's because Syngap, its story of coming from the bench to the bedside and now back, is that we need, I think my data and some things that we've been looking at is, are these seizures occurring early in life? And yes, and then our data also shows that even in the mouse, they're not really, you wouldn't, people have been breeding these mouse for a long time. No one's been like, hey, I think these pups are having seizures when they're younger. My data supports that. So I think that as a scientist, I want to give the pediatric neurologist the best chance, right? So we need to get the gene on panels. We need to get EEGs so that a pediatric neurologist can have, we can identify as many Syngapians as possible, and then start to develop the standard of care. Because once we know the standard of care, I think we find something, maybe something works better, or we start to really understand the genotype, phenotype. And then we can start in the mouse research, we can then say, okay, if this drug works the best, well then instead of trying to give, I'll call it the right hook, right? Like I think that this is the left-handed jab is your standard of care, but the right hook are the gene therapies. They are the completely new and novel things, but that's really a right hook, right? And I think that right now for what we can do today is we put the panel, we get the gene panel, we get EEGs, we start to get bigger cohorts of patients, we keep the communication going, and then we can compare that standard of care in the mouse, and that will allow us not to find 50 right hooks. That will allow us to say, hey, there's 5 really good ones. There's 4 really good ones. And I think, at least for me, that's what I've been thinking of.
Yeah, just to kind of build off of what Brennan was saying, I think as researchers, we tend to compartmentalize things a lot. So I think taking multidisciplinary approaches to, not just the core behavioral symptoms, but to comorbidities, to things like epilepsy, to things like GI distress, it becomes really important because, as Charles and Monica were saying, a lot of these things impact behavior later on. You get stuff like seizures under control, and suddenly behavioral issues start to disappear. We have a tendency to look at things as distinct pathways, whereas more likely than not, they're all really, really interconnected.
Great.
So one of the challenge I think that I'm thinking about and really stems from Chuck's comment on we found the cocktail that works for us regarding seizure. And my understanding is there's a lot of families and different drugs work for their children, and for some other it doesn't work. And I don't know if the information is out there or if there's more things known depending on where is the point mutation and what type of drug combination works and why does it work for my kid and not the next one. And I think those are really challenging and can provide the good insight into where is this diversity coming from? Is there something else than Syngap that's also been susceptible during the development and that might lead to those differences? So I think this still is very puzzling and remains a big challenge.
Great. So I think I'll unpack a couple of things there that you said. One is it sounds like You're talking about genotype-phenotype correlations. Where is the mutation and how does that impact the phenotype? But then the other thing that I think you're bringing up is two kids, nearly identical or identical mutations. One has intractable epilepsy, one does not. Why does that occur? And I think that's another great question. Lots of hypotheses about that. Second hit hypothesis, right? Another genetic change that either makes seizures worse or maybe something that's protective. And, the child that has the relatively easy to control seizures has a mutation in a gene that actually protects him from seizures. Versus, and this is something I thought quite a bit about, is there something stochastic going on? Is it just something not related to other genetic hits, but either epigenetic or really just stochastic in circuit development that leads to one child having intractable epilepsy and another not? And maybe that stochastic thing includes things like what was the seizure burden the first 3 months of life that it wasn't picked up, and that could be part of it. So I think those are really important questions. Anyone else have some thoughts?
Yes, Jimmy, I have a question for you. So when we look at, sex chromosomes, so do we have like any, similarity between boys versus girls, or not?
Yeah, so that's a great question. I don't think there's anything really published on that. If anyone knows of anything, please clue me in. We have, for most of the work that we've done, not really looked at male versus female. There's one study that hopefully we'll have published soon, which was looking at sleep in kids with Syngap mutations, and we did not see any significant difference between boys and girls. But it's a great question. I mean, what about seizures? I know we haven't looked at that in depth, and I don't think the neurology paper from Ingrid Scheffer looked specifically at male versus female. If anyone has any other knowledge, please let me know.
So just
or maybe in the mice. You can tell more about them.
So there's another question that because this, variability of phenotypes that we are seeing in the symptoms. So when we look at genes along with SYNGAP1, do you see any other mutations associated with these patients, or is it just SYNGAP? Because, if you have another mutation, it would definitely, cause these phenotypes. Any consistency throughout?
Right. So, looking at, genetic reports in patients are actually always interesting to look at, and it depends on which company did the analysis. There are some companies that they will only tell you this is clearly a pathogenic change in this gene, and that's the only information you'll get. There are other companies that will say this is a clearly pathogenic change, and then here are 20 other changes. We don't know what those are. They're usually variants of unknown significance. How do those impact the phenotype? Have no idea. I definitely see some children with clear pathogenic mutations in SYNGAP who have other changes that I look at and I say, that could be something. Have a variant in a channel, a sodium channel, or a GABA receptor. But again, it's hard to say because, this child has these two changes, a SYNGAP mutation and then a GABA receptor change, let's say, I'm making that up, and has difficult to control seizures. But I also have other kids with just all I know that they have are SYNGAP mutations, and they also have difficult to control seizures. So it's hard to say. I think you're on the right track, but I think it's a big challenge. And I do know for other disorders, and the one I know best about is Phelan-McDermid syndrome, there has been some effort to try to identify modifiers with not a lot of success. And I think part of that is just the complexity of the genome. I think it's the rarity of these disorders, right? If you had 10,000 kids, it might be a little bit easier to pick these out. When you start talking about 100 kids or 50 kids or 20 kids, it becomes much more challenging. But I think those are very interesting ideas.
Yeah, one thing I would like to add, as thinking of how do we design experiments that get at the problem, it's easy, right? It's always easy to identify the problems, much harder to design the experiments that could potentially, make an impact on them, is that, if we took just, wild-type mice or any model organism, and you induce seizures early in life, what type of phenotypes can you get? And do we understand these, what is the burden of seizures early in life? And then just with seizures, maybe not all the other genetic interactions and stuff, what are we really looking at? And I think that some of the data for the really early data from Dr. Rumbaugh's lab with Dr. Clement, I believe, was the first author looking at thalamocortical connectivity. And I think that some of those outputs that they were looking at with some really good electrophysiology data, actually, when you look at other epilepsy models early in life, it can be similar, but also different. And so I think that incorporating just early life seizure models, or at least taking your data and looking at it for what is known about the things that have seizures, any model organism that has seizures, and understanding the alterations in the brain, I think it's a great control, I think. It's very complicated control and it's very expensive control, right? But it is a great control, at least, and you can at least use it as a thought experiment, right?
All right, so we're at about a minute left. If there's no other questions, we'd like to wrap this up and throw it into the lunch hour. So once again, I want to thank everybody on the panel. It's actually been fascinating. Like I said, I thought you were talking about my daughter all morning. So I appreciate all the work that you're doing. And in terms of housekeeping at this point, we're going to have lunch until 12:10. And then from 12:10 to 1:10, there'll be a Zoom chat with families and the science panel, and the Zoom chat link with the families, and the link can be found in the registration email. And remember to come back to the live screen at 1:10 Eastern for the NIH panel. And also, another reminder, we're going to remind you throughout the day, please do the feedback survey at the end of the conference. So we're on a lunch break till 12:10. Check your email link for the Zoom chat from 12:10 to 1:10, and we're coming back live at 1:10. With that, I thank everybody for their attendance this morning and look forward to this afternoon.
There are thousands of genetic disorders that contribute to mental health, many of which we don't yet have a handle on. There are many that are rare, one of which is Syngap1.
Mutations in Syngap1 are one of the most recently described disorders, only discovered about 10 years ago. It's a relatively rare disorder, probably affecting around 1% or so of all individuals with intellectual disability. And at this point, there really are not effective treatments for patients with Syngap1 mutations. And really, we need continued research to understand the symptoms associated with Syngap1 mutations and develop personalized treatments for those disorders.
So the clinical features of Syngap1 is so varied that many genetic disorders can present similarly. The way to make the diagnosis really is genetic testing.
I would encourage families to follow their gut instinct. If they truly feel something is different about their child and there's something going on, to ask to speak to a geneticist, to ask to have a comprehensive workup. We were on a 9-year diagnostic odyssey to figure out what was going on, and in retrospect, I know I would have pushed harder for some more comprehensive genetic testing. This is definitely a case where you may not see physical features in a child that would point towards something underlying genetic that's going on, but there in fact is.
So mutations in Syngap1 cause a devastating neurodevelopmental disorder for which we have no treatment at this time. And it's only through research that we can identify targeted treatments for these patients and for other patients with these sorts of devastating neurodevelopmental disorders.
The Syngap1 gene plays an important role in brain function. It's important for us to learn and remember information. And changes in that gene impacts how that protein functions and consequently manifests generally with learning disability, which could vary from mild to severe. Epilepsy is a common clinical feature, behavioral challenges, Some individuals have autism. By far all have sensory processing difficulties. Syngap 1 affects many aspects of brain-based function.
For Syngap 1 patients, there are no targeted treatments. We treat the patients symptomatically with medications that only treat the symptoms, but not the underlying cause.
And without collaboration between researchers, clinicians, and patient groups, it would be extremely difficult to advance our understanding of the disorder.
It's so important for families to participate in research because it's the only way we're going to get data to work towards finding treatments. And the idea of participating in research is overwhelming, especially for working, busy families, but it's not as daunting as it seems once you get your feet wet. We've participated in a few research studies. Sometimes it's just answering some questionnaires, sometimes it's bringing your child in to participate in some activities or some studies, but I know that contributing to the research process makes me feel like I'm doing something and helping me be part of the solution and get closer to having a treatment for my son.
There's often a difficulty in initiating research studies because of the lack of funding due to their rarity. And so it's really important for foundations and individuals to contribute to launching the necessary research for understanding these disorders.
The funding that we are bringing into the organization are supporting these strategic programs. And these programs that we have, like working with Texas Children's, Kennedy Krieger, Stanford, and many other organizations and KOLs that are studying Syngap around the world, give myself, a mother of a child with Syngap, and the families that are supporting our organization, the hope they need to keep going.
Having the Bridge the Gap Foundation has been a lifesaver for me. We finally met our people. I finally see videos and pictures of children that are like my child. I finally am connecting with parents that have the same challenges that I'm having, that someone might post, my child is struggling with this. Does anyone else have this experience? And 10 people write back, and we all are supporting each other and sharing strategies, and that has been the silver lining in this diagnosis.
The funding for our organization will go to our strategic plans to help support our mission, which is education, research, and awareness. Our programs, such as a registry program, will be supported. We will be supporting the families through our family meetups and awareness. And then we have our international Syngap 1 conference that will support our scientific community as a whole, on top of allowing the families to participate into the research to know what is actually going on, to keep them in the loop. Then we have our centers of excellence that are going to provide the care and the needed experts that our families need. And so we need these type of funds to support these programs. And as we bring all of this together, eventually, and hopefully soon in the near future, we're going to be finding the treatment that we've been searching so hard for.
Funding for rare diseases might seem like putting a lot of money towards a very small patient population, but it's very possible that a treatment that could impact a rare disease community can be applied to a much broader population and a wide variety of diseases and illnesses, and something that might seem to only be applicable for a small population could actually impact millions of people.
Hey everybody, welcome back. I hope y'all had a great lunch. I wanted to welcome this afternoon. We're going to start off with the NIH Grant Mechanisms panel, and I wanted to introduce our first panelist, which is Dr. Vicki Whitmore. She is with the National Institutes of Health, with the Neurological Institute of Neurological Disorders and Strokes. She is a program director in channels, synapses, and circuits. And she's going to discuss grant mechanisms today at the NIH. Welcome, Vicki. We're excited to have you here.
Thank you, Monica. So if I could have my slides, please. Great. So first of all, my disclosures, I'm a federal employee. I'm not going to talk about any unapproved uses of medication, foods, or devices, and I have no conflict of interest. Next slide, please. So we're really very happy to be with you here today. So as Monica said, I'm Vicki Whitmore, and I'm a program director in the Division of Neuroscience, and I'm going to come back to how NINDS is organized. But with me here today, I have Becky Roof from the Division of Translational Research and Adam Hartman from the Division of Clinical Research. And in the next slide, to take a step back and just to talk about NIH overall, NIH is divided into 27 different components that are called institutes and centers, and each of them has their own specific research agenda, often focusing on particular diseases or body systems. So for example, this morning I was listening to the talks and heard the very interesting talks about GI dysfunction in the zebrafish models. And a grant like that might not be reviewed and funded by NINDS, but likely would be funded by the National Institute of Diabetes, Digestive, and Kidney Disease. So, you know, there are just many different institutes and centers at NIH that support research across the body systems. So each institute has an intramural and extramural component, and I'll come back to that also. And all but 3 of the NIH components receive funding directly from Congress and administrate their own budgets. So NIH leadership, so Dr. Francis Collins is the director of NINDS. He's been with us for quite some time, and who just showed up on the bottom is not Francis Collins, but the infamous and now famous Dr. Tony Fauci, who's the director of the National Institute of Allergy and Infectious Disease, who's been really leading the charge on all of the COVID vaccine development, diagnostics, and treatment. So in the next slide. So the mission of NINDS is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. So to support this mission, NINDS supports basic translational and clinical research through grants, contracts, meetings, and through research in its own laboratories and clinics. We fund and conduct research training and career development programs, as well as in translational and clinical research, as well as the basic sciences, and promote timely dissemination of these scientific discoveries. So in the next slide. So NINDS and all of the NIH institutes are organized with an office of the director, and the director of NINDS is Dr. Walter Koroshetz. And then there are administrative offices, but then the scientific part of the institutes is organized into what's called the Division of Intramural Research, which are the research labs that for the most part reside on the main NIH campus in Bethesda. Some of the labs are elsewhere. So for example, NIEHS, Environmental Health Sciences, is down in North Carolina. NIDA, National Institutes of Drug Abuse, has some labs up in Baltimore. But for the most part, the labs are focused and are concentrated on the NIH campus. The other division then is the division that Adam, Becky, and I are in, which is the Division of Extramural Research. So this is the division where the research funding goes out of NIH to support research investigators across the United States and around the world as well. And in the next slide. So within the Division of Extramural Research, as I said, there's the 3 divisions, the neuroscience, translational, and clinical research, as well as then the Division of Extramural Activities. And the important piece here for you to note is the Office of Training and Workforce Development, which is the office within NINDS that supports all of our training grants, so fellowships, postdocs, the K Awards, Career Training Awards, as well as the Office of Programs to Enhance Neuroscience Diversity, where there are many programs that are supported through that office to support diversity in neuroscience research. And in the next slide. So within the Division of Neuroscience, where I'm located, so as Monica said, I'm a program director within the Channels, Synapses, and Circuits cluster. And our cluster focuses primarily on grants on epilepsy, and I, in particular, oversee the grants on the genetic epilepsies. But we also have a fair amount of very basic neuroscience research that's supported and have oversight of those grants in our cluster. The other clusters within the Division of Neuroscience are neurodegeneration, neurogenetics, systems and cognitive neurosciences, repair and plasticity, and neural environment. And there's a lot of collaboration and cooperation that goes on between these clusters, but it's a really, as we say, flat structure in that each of the clusters reports up to our division head, who reports to the director. So we have very direct input into the research that's going on in each of the areas that we oversee. And in the next slide. So what is the role of a program director? And I think more importantly, why should you get to know them? So as a program director, I'm here, as are all the program directors at NINDS, to provide information about funding opportunities and grant mechanisms, and most important, the last bullet, answer questions and help you to navigate the NIH system. Because it sometimes is not always straightforward. It may not be clear which mechanism you should be thinking about applying to. And that's what we're here for, is to really help you through the maze of what is NIH. When you're preparing a grant application, we don't do a complete review of your grant. That's not allowed, and that's not our role. But if you send us your specific aims page, we can talk through your specific aims. So what's your hypothesis? How are you proposing to approach the study? How are you proposing to address your research? And we can give you feedback and advice as to that, how that's formulated, because that really is one of the most critical pieces of your grant application. If you don't catch your reviewer's interest on that page, then you're in for a tough haul to really convince them to continue to be interested and really think about your research and the significance it may play. So when a grant is assigned to me, so to sort of back up, a grant comes into the Center for Scientific Review and is assigned first to our institute, then it's assigned to a specific program director. So when a grant is assigned to me, then it's my responsibility to listen to the review of that grant during the study section. And then once the summary statement has been released, to meet with you if your grant is not going to be did not get a fundable score, to meet with you, discuss with you how you can strengthen your application and talk through the strengths and weaknesses that have been identified by the reviewers during that review process. So once your grant is awarded, then I become the NIH administrator of that grant. And so any kind of administrative issues that arise, that come up that you have questions about or need assistance with, I'm your contact person, together then with a grants management specialist who's in our grants management branch, who also oversees your grant. If you are trying to find out who you should contact, so as I said, my portfolio, I cover the neurological and especially epilepsy aspects of the genetic epilepsies. But say you're interested, as I said, in the GI system, if you go to this matchmaker link and type in genetics, epilepsy, or genetics GI, what will come up is a list of grants that fall under that, as well as the names of the program directors who oversee those grants. And so that's a great way to identify who you should be talking to at NIH. Very frequently, someone will contact me and I'll say, wow, this sounds like a really cool project, but I'm really not the right person. Let's figure out who that person is. And we help redirect you to get you to the right program director to help you with your grant application. And in the next slide. So if we now turn to thinking about the whole path from training through training, NIH does support research at the undergraduate and post-baccalaureate level, all the way through to pre-doctoral training, to postdoctoral training, early research career development, and then investigator development and career development. So, and this can be, and this sort of path is available for both individuals who are doing basic research, so be PhD neuroscientists or whatever your background is, as well as for clinical scientists who are interested in doing either bench research as a clinician, also interested in doing basic science, or is interested in doing research that involves human subjects. So in the next slide. So at the bottom, I put the link to the NINDS Training and Career Development website. And I really encourage you to check this out. It has a lot of really helpful information and contacts for people in that office. And the way we work with the Training and Career Development Office is that they will typically handle grant questions, very technical kinds of questions, but when it comes to the science, then they'll transfer you over to the appropriate program director, like myself, to talk about what your actual grant application is and how you're going to proceed with that application. So as I said, we fund research at many different levels, all the way from high school, all the way through to early investigators, early stage investigators. Obviously, a large part of the NIH funding goes to established investigators who are no longer early stage investigators. And in the next slide, this is a list of all of the different career development awards. And if we start at the top, the individual fellowship awards, the F30, F31, 32 are for undergrad and graduate and postdoctoral fellows. The F32 at NINDS is a bit different than at most other institutes. The F32 at NINDS, you're required to submit the grant either before you start your postdoctoral fellowship or during the first year. So we're looking to support really very early stage postdoctoral fellows with the idea that the earlier you start writing grants, start getting into the system, and learning the whole grant writing system, the better. And I think one of the other aspects, the reason we shifted this a bit at NINDS, is that we were finding in review of NIH fellowship grants overall, that reviewers were really expecting that the F32 early postdocs to have a lot of preliminary data. And at NINDS, we really look much more at the candidate. Is it a good candidate, a good fit with the lab you're going into, and a great project and a good career training plan? So that's a thing to keep note of, especially if you're thinking about applying for an F32 at NINDS versus another NIH institute. The F99K00 is an interesting grant where you apply for that as a graduate student and you get the F99 funding during your graduate years, but then when you transition into a postdoc, you transition to the K00, which then becomes funding for your postdoctoral training. The Career Development Awards, there's a variety of them here. The K01 is, there's a couple of different K01s that NINDS supports. One that we really would like to see more grants coming in is for later stage postdoctoral fellows. So you're not in your first year anymore, but you're in a lab, you're doing a lot of research, and you really just need to get that research done. You've got a great project. So that K01 grant is really the kind of grant mechanism that you should be looking at. The K99R00 is similar to the F99K00, except that this is a grant that is awarded to senior, sort of more established postdocs who are nearing the end of their postdoctoral training, but not yet done. So you get 2 years of additional training on the K99. And then when you transition into a faculty position, you submit an application and get approved to transition into the R00 phase, which really is great for a lot of people because it gives you that research support going into your first faculty position. The other awards, I won't go into specifically. I think maybe Adam might later, but many of these are used by the clinical fellows for either additional training support, either to do research while they're finishing their clinical work or their clinical fellowship to do some lab work and or to do work doing research utilizing human subjects. And in the next slide. Next slide, please. So just to define what an early stage investigator is. So an early stage investigator is an individual or an investigator who's completed your terminal research degree or the end of postgraduate clinical training, whichever date is later, within the past 10 years, and you've not previously competed for a substantial NIH award, so meaning an R01 research grant award or an equivalent of that. So the great thing about being an early stage investigator is that across NIH, many of the institutes like NINDS has a set pay line for established investigators. So for example, right now our pay line for established investigators is the 14th percentile, meaning if you score a 14th percentile or better, your grant automatically gets funded. But for early stage investigators, there's an additional bump. And so typically it will be 10 points above, 10 percentile points above what the pay line is for the established investigators. So we really give that bump to early stage investigators to help support more early stage and young investigators coming into the NIH funding realm. Young investigators can also sign up to be a grant reviewer. There's what's called the Early Career Reviewer Program. If you go to the Center for Scientific Review page on the NIH website, you can go to the Early Career Reviewer Program page and submit your information if you'd like to be considered as a reviewer. And I really encourage anyone who is in the position to do this to sign up, because it gives you that experience of participating in an actual study section. You typically are given a few grants to review, never as the first or primary reviewer, but as one of the reviewers. And it really helps, I think, for you to have that experience to see what happens in a study section as a grant is brought up for review and discussion. And in the next slide. So in addition to NIH, there are other agencies and nonprofits that support research. So the American Epilepsy Society and CURE and the Epilepsy Foundation all support early stage investigator research. The Syngap organization resources out there. And I just, as I was preparing for this talk, came across this interesting article by of that might support research. So take a look at that article. It's actually quite interesting and creative, the way he's been able to support his research. And in the next slide. I'll stop there and thank you all. I think hopefully we'll have time for some questions later, but at this point I'll turn it back over to Monica, and thank you for your attention.
Thank you, Vicki. And now we're going to move into our next presenter. And I know I have some questions later. We're now going to have Rebecca Roof, PhD Program Director for NINDS, Division of Translational Research, National Institute. And she's going to talk about funding and support opportunities. Thank you, Rebecca.
Great, thank you. I'm happy to be here. Thank you for the opportunity to talk about this. Like you said, I'm Becky Roof. I'm a program director in the Division of Translational Research here at NINDS. My email address is right here on the front page. Feel free to reach out if you have any questions about anything that I talk about today, or if there's any questions we don't get to during the chat. Also, I'm not sure if you'll be getting these slides, but if not, I'm happy to share them as well. So first of all, the Division of Translational Research at NINDS, our mission is to accelerate basic research findings towards patients for use in neurological disorders and stroke. And we do this by providing not just funding, but also expertise and resources, as well as training opportunities to the research community. And we support small molecules, biologics, device, therapeutics, biomarkers. And what we consider translation here at NINDS is anything from, assay development all the way through early clinical trials. So typically, if you have some target that you're interested in developing a therapeutic towards, that's where we take people in as an entry point. And then once we finish with early clinical trial, then we hand things over to our Division of Clinical Research, and Adam will tell you about that in a little bit. So that's the zone we're talking about today. Now, before I get into all of our funding opportunity announcements, I just have this slide here to kind of illustrate that you need to have a little bit different mindset when you're applying to translational research than maybe when you're doing basic science. So people who've come to us from the basic science space, we tell them, okay, you're not exploring anymore. You're not doing, hypothesis-driven research. Now you're on a path. Okay, there's a goal, and what steps do you need to take to get to that goal? So it's a little bit different mindset, and that's really the point of this slide here, is just to kind of illustrate that. So you put your translational hat on when you come to us. Because you're thinking farther ahead and you're on a path, you also need to be including additional expertise on your team. You might be needing to think about, regulatory or tox studies that you're not used to thinking about if you're in the basic science space. So the point here is just a little bit different mindset. And if this is new to you, again, as Vicki mentioned, it's always good to talk to us in advance. Because this is, like I said, it's a different hat you got to put on when you're doing translational research compared to your basic science that maybe you've done before. I also have a slide here about milestones, because this is something that's new to a lot of folks. If you're used to doing basic science, I don't want you to be scared when you see a milestone mechanism. So the way these typically work is you come in and you do your first phase, and the first phase is just to demonstrate feasibility, to show that you get your ducks in a row, that you're ready for the next stage. And then there'll be a milestone, and then you'll go on to the bigger event. So for example, maybe the first phase you're setting up an assay, and then the second phase you're going to use that assay to screen for potential therapeutics, or maybe the first phase you're finding your lead candidate, and then in the second phase you're doing your IND enabling studies, right? So you can see where the first phase sets you up for the second phase. And the reason I put this in here is because some people get a little nervous about milestones, but it allows you to do those dependent aims, right? That maybe you can't do in other mechanisms. So that's why we use them, because like I said, the translational research, it's more, you're on a path, you're taking steps. Step 1 leads to step 2, leads to step 3. And these mechanisms really lend themselves well to that kind of activity. So I just put that out there to say, don't be worried when you see a milestone mechanism. It's there for a reason, and it really opens up doors to you. It allows you to do these things that you might not get to do in other mechanisms. All right, so now I'm going to talk about our programs that we have in the Division of Translational Research. We have a whole pile of different funding opportunity announcements, and we break it down by stage in the process, as well as therapeutic approaches. And the reason we do that is so that we can be very directed and focused, like you need to be for translation, but at the same time, we can be flexible because we have a lot of options for people. Okay, so I'm not going to get into a lot of detail on any of these today because, I don't have a ton of time, but I do want to touch on each of these and kind of tell you what we have and where to go if you want more information. So first, I'm going to start with the early drug discovery and drug development tools opportunities that we have for you. And then I'll get into the later stage. So the first one I'm going to touch on is the IGNITE program, Innovation Grants to Nurture Initial Translational Efforts. So this is the early stage therapy development program. This supports, coming in with a target all the way up through, now you've got a compound that you need to develop further. So I'm the point of contact for this program. If you have questions, I'm happy to talk to you. I'm happy to talk to you about a particular application that you're thinking about, or whether this is even something to consider. We have 3 FOAs here. The one supports pharmacodynamic and in vivo efficacy studies. The second supports assay development and therapeutic agent identification. A lot of people use this for high throughput screening, but it doesn't have to be. Both of these support both small molecule and biologic development. The third one is for animal model development. I saw a lot of zebrafish this morning. So for example, that might be something you would be interested in looking at. These all use a phased milestone-driven grant mechanism. They're all 3-year grants. So again, if you're interested, I'm the one to talk to about this one. The second program I'd like to touch on is the biomarker program. We have 3 FOAs here as well. Here, your point of contact is Marianne Pellimonter. The 3 FOAs are discovery for biomarker program, first for identification, development of the detection method, attain proof of principle. The second is analytical validation of your candidate biomarker. This one you can use to determine the precision, sensitivity, stability of the detection method, whatever in the real world setting. And then the third one is the clinical validation of the candidate biomarker. This one you would use maybe to determine sensitivity, specificity, predictive value, etc. Again, in your real world setting. So again, Marianne Pellimonter is your point of contact if you want to pursue that mechanism. We have 2 different screening programs. The ETSP might be of interest to this audience for epilepsy therapy screening program. This program has actually been around for quite a long time and has contributed to a lot of currently available anti-epileptic drugs, which is really quite exciting. We're pretty pleased with that. But the way this works is no cost to the participant. If you have compounds that you think might be interesting as anti-epileptic drugs, you can send them to our contract resource, and we will have them screened for you for free. It costs you nothing, and you maintain your intellectual property. And we do this as a service to the community because we want to make, to help the field. So it's open to academic and industry, and it's open for small molecules, biologics, and natural products. If this is something that sounds of interest to you, your point of contact is Brian Klein. Now, because this program has been so successful over the years, we've also developed a similar program for pain. I'm not going to go into a lot of detail on the pain programs today, but know that that's also available if it's something that's of interest to you. Your contact there would be Smriti Iyengar, and her information is here. Okay, so I'm going to move on to the further later stage drug development programs. But first, I am going to mention there's a whole suite of HEAL initiatives that I'm not talking about today, but I do want you to know they exist if you're interested. HEAL is helping end addiction long term, and NINDS's part in this is we're interested in developing pain therapies that would be non-addictive. There's a whole series of FOAs around that I'm not getting into, but know that they're there. And if you want more information, feel free to reach out, and I'll send you some information. All right, but now moving on to the drug development programs. The first one I'm going to talk about is the Blueprint Neurotherapeutics Program. We've actually supported quite a bit of epilepsy projects here, which is quite exciting. This is actually quite a cool program, actually. So a while back, it was identified that there's a lot of really great ideas coming out of basic science by people who sometimes don't have the expertise or the know-how or the connections to bring it all the way forward to to a therapeutic. So this program was designed with that in mind. So we don't just give you a grant and walk away and say good luck. We also give you, in addition to that grant, we give you access to consultants. We give you access to a whole series of resources, contract resources to do things like MedChem, to do PK, to do the data management, to do the formulation, to do the toxicology studies, all the way up through the first in human clinical trials. So that is provided to help people bring their ideas forward. And the hope is that by the time you've gotten your first in human clinical data, you're either ready for a later stage program like Adam will talk about, or you're ready for industry investment. And we've actually had quite a few projects get to that stage, which is quite exciting. A key point is that each project is unique, and we will build a team around your project that involves these consultants and the NIH staff and the contract resource organization. So it's really quite a cool holistic program that really gives you everything you need. And along the way, even though we have a contractor making chemicals for you and doing your PK tox and stuff, you maintain intellectual property the whole time. So that's really quite an exciting program. And also, because it's been so successful with the small molecules, we've expanded it to, or we will be expanding it, hasn't happened yet, to include biologics and devices. So that is coming in the future. Now, meanwhile, since that hasn't happened yet, we do have a Create Bio program, which supports later stage development of biologics. This program fills a similar space as the Blueprint, except that it does not have all the contract resource organizations available. It does have some consultants available. But again, it's this later stage. So what I mean by later stage is you've got a compound of interest and you've proven, or you have evidence to suggest that modulating the target you are interested in causes some positive biological effect in vivo. So if you're at that stage and you're developing biologics, this is the program that you should be looking into. The point of contact here is Chris Boshoff. Now, while we're talking about biologics, this is coming soon. It's not here yet, but we are working on a program for ultra-rare disease gene therapy network. This will also be a program where you can apply for a grant, but also get access to contract resources and consultants and a network. So I don't have a lot of details on this one, but it is coming in the future, and that we're quite excited about it. So stay tuned on that. And the last, I've talked about small molecules, I've talked about biologics, and now I'm going to talk about devices. We have a whole series of funding opportunities for device therapeutic development for different stages along the pipeline, starting from concept all the way up through clinical trials. And the point of contact here is Nick Langles at the bottom. Now, the programs I've talked about so far focus on either stage or modality or type of therapeutic you're trying to develop. The SBIR program is broader. It supports early stage and late stage. It supports any kind of therapy development, as well as diagnostics or research tools or anything that might be of interest to you, anything that would have commercial potential and is a fit for NINDS submission, we can support through this mechanism for small businesses. Now, the cool thing about this mechanism is that we're required to set aside funds for this program. So if you're eligible to apply, I encourage you to use the SBIR program so that you can take advantage of those set aside funds. And your point of contact for that program is Emily Caparello. Okay, Another program I'm just going to touch on, I don't know if this is a huge interest to this audience or not, but just so that you know it's there, we have a Countermeasures Against Chemical Threats program. If you're developing or hoping to develop therapeutic countermeasures against chemical threat agents, this is also a program where you have access to not just the grant, but also contract research organizations who will help you with various steps along the way. And this program is actually in collaboration, not just with NIH, but other federal agencies, as you can imagine, are also interested in this. So the point of contact for that program is Dave Jett, if that's something that's of interest to you. All right, so that's it for those programs, but I do want to touch on some training, since I think that might be interesting to this group. Most of our training at NINDS is through the programs that Vicki talked about, which I'm not going to go over again, but we do have a couple that are very unique to translation, so I thought I'd share those with you. We have a T32 program, which is for an institution to be a training site. The FOA is here, and Natalie Trzynski is your point of contact. Another cool resource that we have is we have this free 3.5-day class on how to do drug discovery and development. It's free. It's intended for academic scientists who maybe aren't trained therapy development, trained in therapy development, but it is freely available. We've missed the deadline for this year for applying, but you can see previous year's classes, slides, videos are all freely available. Like I said, if you want to ask for this slide set, I'll send it to you, and these links are clickable. You'll be able to go there. You might be able to Google it as well, but it's freely available class just to learn how translation works and what steps are involved. So that's a cool resource. All right, and before I close, I'm just going to give you some general advice. Vicki has already told you to contact us in advance. I'm going to reiterate that, especially with the DTR programs, because a lot of them are a little bit nuanced. There's entry criteria, there's specific spaces. Some of them will only support certain things. So please, please, please talk to us before you put together the effort of putting in an application. I hate to see somebody submit something and then it's not a fit for the FOA, and that effort is wasted. So don't do that. I've told you at the beginning where some of these are milestone driven, especially if this is a new thing for you, writing milestones. You can talk to us, but it's very important that they be clear, go, no-go decision-making points. I talked about how this is important for how these mechanisms work. Include rigorous study design and supporting data. I don't think this is new to anybody. I think we've been talking about this for a long time, but I can tell you when you apply, reviewers will look at it. So this is a preventable problem. Please pay attention. A multidisciplinary team is really important for a lot of these mechanisms. You'll have a multidisciplinary review. I talked at the beginning about all the different steps you need, not just the disease expertise, but you're also going to need to have, clinical expertise. Maybe you need regulatory expertise. Maybe you need a med chemist. Think about what makes sense for you and your project. But keep in mind, your review panels will be very multidisciplinary. So you want to make sure you have that team together. If it's relevant for the FOA, you want to think about intellectual property. If it's relevant to the FOA, you want to think about the therapy development plan. And by that, I mean, not just what happens in this grant, but how is this going to feed into this getting all the way to a patient? And what are the next steps? And I'm, sure that you're thinking about those next steps. And pay attention to clinical research rules. Every once in a while, we get somebody who's surprised to find that they're using human subject tissue, and that means they're doing human subject research. And some FOAs allow that and some don't. So just pay attention. Don't just assume it doesn't apply. Look it up and make sure. And reach out to us if you have questions. All right. I feel like I went kind of fast, but we'll have time at the end for questions, I think. All right. And I think that's it for me. Back to the panel. Thank you.
Hi. Thank you, Rebecca. That was a great presentation and just overview of what you guys have. The next presentation I want to introduce Dr. Adam Hartman, and he is the Program Director for the Division of Clinical Research at NIH at the NINDS. And he's going to be discussing the NIH divisions of neuroscience. And so thank you for being here, Dr. Adam, and you can take it away. Thanks.
Thanks a million, Monica, and thanks a million for the invitation. It's really fun to be here today. My name is Adam Hartman. I'm one of the program directors in the Division of Clinical Research at NINDS. And it's a lot of fun. I always love talking to the young investigator group. A lot of what you've seen already sort of rolls right into what I'm going to discuss now. So if I think, both Vicki and Becky have talked about the continuum. So this is, the next stop. I don't like to say it's the end of the road. It's the next step in the process. So some brief disclosures. I'm a federal employee. I also do second opinion consultations. My wife works for Baltimore County and earns income from Johns Hopkins Hospital. And I will not be talking about unapproved uses of medications, food, or devices. So the mission of our division is to provide oversight for clinical trials to test the safety and efficacy of innovative treatments of neurological disorders and stroke. And so the key words there are clinical trials. We also oversee epidemiological studies of natural history and biomarker studies and studies designed to elucidate the causes of neurological disorders. We also try to ensure the proper level of patient safety monitoring, supporting the other divisions as well, and support compliance with human subjects policies across the federal government. And finally, we provide expertise in statistics and clinical trial design, both to the Institute and to clinical investigators. And so what I'm trying to show here is that our division funds and oversees human subjects studies that really run across the spectrum of, so we're sort of laying out the different divisions here. Vicki is from the Division of Neuroscience, and she's already talked with you about the different clusters within that division. Our division spans those interests, as does the Division of Translational Research. So we have a number of programs that are used to support this, and I hope you can see some of the finer type. We go all the way from clinical trial readiness, which you see on the far left, including a funding mechanism for clinical trial readiness, all the way through dissemination and implementation, as well as comparative effectiveness research on the far right, where we have, we're part of specific funding announcements for dissemination and implementation, as well as our own funding announcement for comparative effectiveness. We also have, for phase 1 first in human studies, let me go back, sorry. Okay, we also have a number of funding announcements that where we provide advice. We're not really the primary division for some of the biomarker discovery and validation studies, as well as outcome discoveries and validations. We are involved in the clinical trials that are embedded in natural history studies. We also are involved in studies for first and target population studies. We have an exploratory clinical research funding announcement, which I'll talk about a little more in a minute, as well as phase 2A and phase 2B studies. And we have a funding announcement that specifically addresses phase 2B and phase 3 efficacy. We also have a number of networks, clinical trials networks, that I'm going to talk about in just a minute. So let's talk a little bit about some of these funding opportunities. We have an exploratory clinical trials funding announcement. So this is where you're just starting to figure out whether or not the treatment or intervention works. We also have an SBIR for small businesses that Becky alluded to. We have an efficacy clinical trial. So this is where you've already done the exploratory work, and now you're going for the more so-called pivotal trial type of a funding announcement. The exploratory clinical trials and efficacy trials are funded under what's called a cooperative grant mechanism. And what that means is that after the grant has been approved for funding, once you get that, the magical notice of award, a member of NINDS becomes a part of the management team and helps with oversight of the study. This is generally perceived to be a real advantage because it really helps to move the studies along and gives the investigators an opportunity, sort of an avenue to communicate more directly with NINDS. We also have a number of clinical trials networks, which I'll talk about a little bit later, including NeuroNext, StrokeNet, and Siren. We'll talk about that. We're also a part of the dissemination and implementation research and health funding announcements at different funding levels. We also, as I mentioned before, have a clinical trial readiness for rare neurological and neuromuscular diseases. So this is a study, the type of a study you would do apply for here would be something where you need sort of that last step for the validation of an outcomes measure for a clinical trial that you're going to do. So it's already been pretty well developed, but there's that little gap in between having it pretty well developed and being ready to use it in an actual clinical trial. And so that's meant to focus on rare neurological and neuromuscular disorders. And so that's relevant here. We also have a comparative effectiveness funding opportunity that is, it's agnostic to whether the disease of interest is a rare disease. We've had rare disease applicants and we've had more common disease applicants as well. Let me just expand on that for a minute. So this is one that was published back in 2019, and this is meant to support pragmatic study designs using cost-effective means of prospectively collecting observational data. So this is where it's care as usual, but there may be more than one type of care as usual for a specific target, for example, seizures or epilepsy within a given disease. And so we're only taking prospective observational studies for this. If you want to do a comparative effectiveness trial, where you're actually trying 2 or more interventions that have already been approved or in common practice, we actually have a separate funding announcement for that. This is, Becky's already made reference to the staged funding opportunities. And so this is a staged funding opportunities. And the one thing that we want to emphasize, particularly for all the diseases, but it's particularly relevant for the rare diseases, is that we really want strong engagement with patient advocacy groups throughout all the way from the design through the implementation of this particular type of study. So some of the goals of our clinical trials program, number one, obviously, is to provide funding opportunities and some infrastructure for clinical trials. We also create some disease-specific initiatives for specific mandates. We've talked a little bit about some of the rare diseases. There are some that are a little more focused. Some of the examples for disease-specific initiatives include stroke. And so the stroke studies are for any type of a cerebrovascular abnormality. It's not necessarily for just older adults who have hypertension, diabetes, and have a stroke as a result of that. We also have studies for neurological emergencies, which isn't really necessarily relevant here. We support training in clinical trials and research methods. We're going to talk about training in a couple of minutes. We provide scientific oversight of ongoing research, as well as administrative stewardship of clinical trials budgets, and everyone knows those can be very expensive, and the promotion of sharing and reporting of data. So what I'm going to show you here, we've talked a little bit about the spectrum from exploratory all the way through phase 3 trials. And so we have a number of funding announcements. I've already made reference to the acute care Siren network and the NET network, which isn't necessarily something I think would be that relevant here. But we are going to talk for about a minute or two about the NeuroNext study. So NeuroNext is designed to do phase 2 studies and biomarker exploration, biomarker studies. And we're going to talk about what sorts of studies have been done in that. We have a separate slide on that because that really does, most of the studies that have been funded through NeuroNext have actually been rare disease studies. We have StrokeNet, as I've already mentioned, and now the Siren network for more acute care. And then we also have, it says coming 2019, it's already here, the HEAL initiative, which is for pain. And this is, pain across the board. It's not just a specific type of pain. So let's talk a little bit more about NeuroNext. And so NeuroNext has multiple sites as well as a coordinating center. We have 25 regional sites and data and clinical coordinating centers, as I mentioned, and it really is designed to centralize the clinical trials organization because there's a lot of infrastructure that goes into clinical trials. So there's a number of studies that have been done, completed already, as well as some ongoing studies. And when you look at this, you'll recognize there's a number of rare diseases here. So super baby was the very first one. That was a spinal muscular atrophy biomarker study. We'll talk a little bit more about super baby in a minute. We funded studies on multiple sclerosis, myasthenia gravis, acute stroke, so that's not a rare disease, Huntington disease. We have ongoing studies in glioblastoma, form of brain cancer, learning in Fragile X, the AFQ056 Novartis study, cryptogenic peripheral neuropathy, and GNE myopathy. So as you can see, most of these are rare diseases. Just a little more about Neuronext. We're really trying to change the landscape of clinical trials, trying to empower academic investigators. This is really meant for academia, not for industry. And it's really trying to provide a real world perspective natural history data set to accelerate public and private development, particularly with the biomarker studies. And then I mentioned the super baby study already. Really setting those biomarkers has helped to not necessarily with the first drug that was approved for spinal muscular atrophy, but for subsequent drugs. The data set's been, we're told, very useful. We also, you may have noticed in one of the prior slides, we talked a little bit about common data elements. And so just to give you an idea of what common data elements are, it's something that really, where NINDS is a leader in implementing this across NIH. The goal is to develop common definitions and harmonized data collection so that we can develop more efficient trial designs and promote data sharing and pooled analyses. There's a number of disease areas, which I'll show you in just a minute. This is the result of collaboration with industry, other federal agencies, and patient advocacy groups. And as I mentioned, it's a trans-NIH effort. We don't have one for every specific disease. I'm going to show you a list in just a minute of the ones that we've developed thus far. But there are some generic common data elements where that you can use. You can sort of, if you think of it, just sort of picking and choosing off a buffet. Not that anyone's going to buffets anymore, but you can pick and choose off the menu, if you will, which ones may be most useful for your study. That can facilitate research by other groups that may have related diseases and disorders. It can also be very helpful for you if you're going to be doing a comparative study. So here are some of the areas. You may notice that epilepsy has pediatric specific recommendations, as well as a variety of other diseases and disorders that you see listed there. This isn't a requirement, but it is something that's strongly encouraged, and they're actually easier to use than you might imagine. The way the process goes, just to give you a little bit of background, they're developed and vetted by experts in the scientific community, and then we turn to our partners and other federal agencies like the National Library of Medicine, which is a part of NIH, CDC and Department of Defense. They're aligned with the ISO international data standards whenever possible. We also lobby for patient advocacy group input and industry input, and then we provide continuous guidance, review, and oversight. Just to talk a little bit about training within our network. So training is a big part of this. Within Neuronext, we have pilot trials, but we also have, we're going to talk a little bit more specifically about it, all of our networks have educational webinars, journal clubs, education committees. They span the US, but also parts of the international community. One of their goals is to train new investigators because the people who are doing them now are not going to be around forever. And they also have working groups for education as well. One thing within Neuronext, part of the Neuronext network is the clinical trials methodology course. This is an ongoing collaborative course that includes faculty from several of our networks, as well as a number, it's mostly outside faculty. The American Academy of Neurology helps support it with education grants. There is a series of comprehensive webinars. Actually, the last one of the season was earlier today at noon. If you caught it, probably not. But there are webinars on a whole variety of clinical trial subjects. And you can, these are available, they're recorded sessions, so you can look at the archive sessions. There also, in addition to that, is a year of small group mentoring, where you'll be, in a group of, there'll be 2 or 3 faculty either from universities and or from NIH, mentoring two or three trainees who typically are people who are junior faculty. Sometimes they're senior clinical fellows in that, but it's really fantastic. And the idea is to develop a really coherent research proposal. It's really the protocol that we help you develop over the course of a few months, usually 3-4 months. And from that protocol, you will usually pull parts of that protocol out for an application. There also is a mock study section at the end of the course, which is, and it's people who sit on study sections who are on that mock study section. And we've been told that the reviews are a little bit more challenging, a little harsher than they are in the real world. But you'd rather hear it from friends where the decision isn't being made, and then come up with a much stronger proposal later on where hopefully you'll get funded. Vicki already showed this slide, the researchtraining.nih.gov career path. This is really helpful. You can put in what type of research investigator you are, and it can give you some suggested funding announcements that may be relevant to your career path. So I've noticed, unfortunately, some of the background on some of my slides isn't showing up. This is a slide that's meant to show our diversity program. So diversity is very, very important at NINDS. We have a number of programs ranging from T32s and K awards of different levels, as well as diversity programs in the R25 space. Career stages really ranging anywhere from high school all the way through new faculty. So you can see at the bottom on the NINDS website, the Training Career Awards, Diversity Awards URL. And hopefully these slides will be made available so you can see them. Here's some hopefully helpful websites that may be of interest to you. There's the NINDS website. There's also the NIH Reporter. This is where you can look to see what's been funded. You can see what people are working on. You certainly don't want to propose something that's exactly the same as what's being funded right now. You can look for potential collaborators or possibly competition. And you can also, there's a feature called Matchmaker, which Vicki mentioned, where you can look up, you can put in some keywords or put in a set of specific aims, and it'll give you an idea of which study sections might be looking at your proposal, give you an idea of what types of funding mechanisms have been used, which types of institutes have been the ones to fund that kind of work. And it actually will spell out a list of the actual grants for you. So you have an idea of who's doing it and what specifically they're looking at. There's also the NINDS funding opportunity announcement URL that I've listed for you to review. So I think I'll end there. Vicki had made mention earlier about some of the K award mechanisms that can be used by clinicians, people who are either late in fellowship or early in their faculty career. And I guess I'll just expand on that. I'll take that as a question from Vicki, and I'll expand on that for just a couple of minutes. And she mentioned there are 3 of them. There's the K08, which is for a clinician who, so these guarantee 75% protected time for you to do research. That's a requirement, actually. And the other 25% typically is made up of a combination of clinical work or teaching or possibly administering a small program or something like that. So the K08 is used for people who are interested in doing basic science research, and sometimes a lot of the translational work might go there too. There's a K23, which is for people who are doing patient-oriented research, human subjects work as a part of their research. So for example, if you're doing imaging studies, you're probably going to be, in humans, you'll probably be coming in under a K23. So those are for people who are typically starting out. There also is, if you are a child neurologist, there also is the Child Neurology Career Development Program, which is a national program that's funded under a K-12 mechanism that's awarded to the Kennedy Krieger Institute that oversees it. That's limited to child neurologists. That's up to 3 years of support. Typically, that would come before that's to get child neurologists enough preliminary data to really prepare for a K08 or a K23. If you're a neurosurgeon, we have a similar and analogous program for neurosurgeons. It's a national program. And then finally, there's the K02, which is for people who either are almost ready to move on. It's technically a mentored award, but it's really for people who are just about ready to move on to their own independent funding, an R01 or something comparable to that. That may be something that comes on the tail end of after you have completed a K08 or a K23, you're not quite ready for the R01 yet. Or it may be something that, for example, if you're part of one of those K12s, you may be able to use that as a springboard onto an R01. So there's really a wide variety of funding mechanisms, and like Vicki and Becky have both said, the training, the NINDS training office oversees some of the more mechanical training aspects of that type of award, and we provide guidance on the scientific aspects of those awards. So with that, I turn it back to Monica and Charles. Thank you.
Thank you, guys. This was incredible information, and we do have questions coming in. So what I would like the audience to do is, if you go to your phone and go to your camera function, we're going to display the Slido QR code. All you have to do is aim and click, and it will bring you into the questions that the audience has. And I will ask the panel to come back, and we will go in, because we do have a little bit of time for some question answer. And thank you all, because this is great. I mean, this is a great resource for scientists. And I've actually always wondered myself, how do you go about getting these grants? How do you go about finding money for the research? And it's of course, especially in rare diseases. So I want to thank everybody for taking the time this afternoon to doing that, because it is really super beneficial.
I did have one question for all of you as you did it. I've never applied for a grant. I wouldn't know how to apply for a grant, etc. But are there any particular underused funding or grants that are like begging for proposals? Or is everything kind of like saturated, so to speak? You have more proposals than funding?
Anybody want to take that?
Whoever wants to take it.
Well, I think we always have more awesome science than we can fund, unfortunately.
Exactly.
The SBIR program is a program where there's set aside funds. So we're always encouraging people to apply there, because there's less competition for those funds. But I wouldn't say it's begging and underused. It's certainly used, but that's the closest I can think of. I don't know about Vicki and Adam, if you have other thoughts.
So as I mentioned, I think in the basic science world, the K01 postdoc awards, we really do not get many of those grant applications, or not as many we would like to get. So I think that, as I said, that's for not someone that's a brand new postdoc, but someone that's in a lab, has a project, and just wants to submit a grant to get that work done. So that, I think, is an underutilized grant mechanism.
All right. Okay. And I do have my first question. And I guess this is not designated to just one person, so I guess you can jump in. But they're asking, does the NIH, NINDS, or NIH in general, have a good resource for young investigators to learn how to find funding sources, and also how to actually grant write and submit research proposals, making sure, I'm assuming, kind of the example you gave about reviewing aims, actually getting started and understanding the format, really key things to focus on when you're wanting to hammer down a research project. Did we freeze? Okay, I think we back. Okay, I'm going to re-ask that question. The question does the NIH or NINDS have a good resource for young investigators to learn how to find funding resources, and also how to write and submit grant proposals, almost like grant writing workshops on exactly how to structure a grant. What are some of the key some of the key components of aims, and just basically how to structure a grant, because of course, when you're coming right out, you want to make sure you're hitting it, and they are time consuming from what I hear to do. So that's for anybody on the panel. It wasn't directed just to one person.
You want to start, Adam?
Yeah, I'll take a whack at it. So, so we could fill the rest of the time talking about this. So, there are a number of grant writing workshops that are typically offered through universities that can help people who are usually offices of professional development will have courses on grant writing. And I think if you're at a university, that's a really, really good place to start. Number one, because they tend to be more targeted to the level of experience of the attendees, but they also may include components of, different institutions have different rules. So I guess one thing we'll sort of take a big step back, and that is when NIH makes an award, the award's actually to the institution, it's not to the individual. And so the institution plays a major role in managing the financial and sort of just day-to-day logistical management of that award. So if you are able to take a grant, be part of a grant writing workshop, which most institutions offer, multiple times a year, if you can do that, you'll learn not only about how to write a good NIH grant, but you'll also learn about what some of the individual institutions' rules about when to get. I mean, one of the things that drives everybody crazy is deadlines. And so you need to know when to submit, when it needs to be into your office of sponsored projects or its equivalent, and how long it takes them to process it, where you stand in the, in the pecking order, that kind of thing. So I think that's one piece of advice. Another piece of advice is that there are sample grant applications that you can find from at different institutes. I don't think NIH has them, NINDS has them, but I know that NIAID, the Infectious Disease Institute, has some sample applications. If you want to see what a mock study section looks like, then the Center for Scientific Review has some great videos. Actually, one of them featured one of my old professors in college. That was a little bit freaky when I saw him on there. I didn't realize still doing it. So, so they have a mock study section, which it's pretty close. I mean, each study section is a little bit different. It has its own personality, but it's a very good generic one. So, there's a lot of ways, Monica, to jump into this, but I think if you're already at an academic institution, the Office of Professional Development is really a great place to start. In terms of the different types of funding mechanisms, it's not always clear what someone should go for. So I think probably the most useful thing would be to scratch down some ideas, contact, you can put them into that matchmaker feature that we mentioned in the NIH report, or see which institute or institutes, typically it's almost always more than one, has funded that kind of work, and see who the program directors are, people like Vicki and Becky and me, who's been involved in that before, and contact those people. I think that's probably I'm oversimplifying it a little bit, but I think that may be the most, hopefully, a pretty accessible way to do things. We could, like I said, we could fill up the whole 2 hours talking just about how to do this. I don't know if Becky or Vicki have additional.
Yeah, I'll add, NIH has regional seminars. That's just general how grants work and how to write a grant and where to go for information, a little bit more comprehensively than what we've scratched the surface on here. But that's something to consider too.
And one thing I'll add is all of the nonprofits have come together in the epilepsy space to form what's called the Epilepsy Leadership Council. And together they have a website that's called Epilepsy Research Connections, where any of the organizations that support research can post their funding announcements there. So that's also a really great place to look, especially for trainees who are looking for, postdoc or early training awards.
Awesome. And we do have a couple more minutes, and I do have a couple more questions that came through. And this, of course, is just for the entire panel. The question asks is, what do you think has been the biggest benefit of researchers to include patient engagement as a part of their study design? Basically, I guess, I don't know if they're referring to just data or actually involving the patients in the design study itself.
Yeah, I'll start off with that one. So there's a number of different ways in which patient advocacy groups can be involved. And I think it depends a little bit on the funding mechanism that you're going for, what kind of work you're trying to do. In the clinical trial space, we really are looking to see that it's not a requirement at this point, but when our clinical trial study section looks at, an application, I think it only can be of benefit to have a patient advisory group involved. And you really, I think ideally, get the group involved at the design stage. Mm-hmm. I know what's going to be practical. Can your child come in to get 4 sedated MRIs in a week? No one's going to do that. What if the families have to travel, hundreds of miles? That happens. And so I think that if you get a letter of support from a patient advocacy group saying we were involved from square one, and if the design of the clinical trial looks like it really did have meaningful input, the study section members can see that. And they do see that. And it's not a requirement for most of our funding opportunity announcements, but it is something that we strongly advocate. For our comparative effectiveness FOA, it is a requirement. We have to see evidence. And again, if you just say that we showed it to the group at the very end as we put this application in, the applications look very different than one where they were involved from the very beginning. Again, as program officials, we can see it. The study section members are smart people. They can see it. We haven't mentioned PCORI, the Patient-Centered Outcomes Research Institute, which is not a part of HHS, the Health and Human Services Department, but we consider them to be one of our federal partners. And for them, patient advocacy group involvement is it's really one of their keystones. And so they have to be a part of it. When you look at natural history, I'm going to sort of march back from clinical trials and some of the later stage studies. As you march back, natural history studies, if you're going to propose to do something without input from the community that's most affected, no trials without me, right? No studies without my input. So as you start to move in the translational space, I guess I'll let Becky and Vicky comment on it in their spaces. If they weren't here, I would comment on it.
And I can add, it's certainly more critical at the clinical stage, but if you're coming in with a translational grant and we don't always see that, we don't often see it, but if you came in and said, occasionally we do get somebody comes in, we've worked with the advocacy group and they're on board and they think that we're treating something that's really meaningful, that's much stronger than somebody just saying, oh, this is a problem that I'm trying to solve. But it means more coming, with that backing, with that letter of support.
Yeah, and I'll just comment from my perspective, just a couple of examples. So I typically talk to investigators who maybe have identified a potential treatment using an animal model, and they now want to move that forward. But say they've done this in an animal model of temporal lobe epilepsy. I'm making this up. But they want to move it forward as a treatment in infantile spasms in humans. But they say, oh, we're going to formulate this in a big horse pill. Well, how is an infant going to take a big horse pill? I mean, any mother that, anybody knows that that's not going to happen. And another example, soon after I joined NINDS, I was asked to participate in a discussion about a clinical trial proposal that came into us. And the investigators were proposing this was in adolescents with epilepsy, and they were proposing to put the individuals on and off drug and look at how many seizures how much the drug reduced seizures. And then when they took them off, how many it increased, and essentially put them on and off and on and off. And I was, as a mother, I was like, if I had a child with epilepsy, I'm not enrolling my child in this trial. And so there's things like that that may make sense, or the investigators may think they make sense. Scientifically, when you think about implementing that in a real person, make absolutely no sense. So that's why it's really critically important to have that patient involvement up front.
That's good. And I do, well, as you know me, I am a huge advocate for the patient having a voice in every piece. And it's really critical. It's because it's helped our group, our community just move, at an accelerated pace, really over a short period of time to get us where we are today. Because without our community and a very active community, I don't think we would have the attention that we're getting currently on trying to find treatment. So I think it's really important, even at the basic science level. I mean, I think it's just critical.
Monica, I have
Yes.
one question. You have this matchmaker function, which sounds very fascinating. So I'm an investigator. I go in and I try and match. I wonder, does it work the other way? That is, can they find advocacy groups? Is there a matchmaker function for an advocacy group to look and say, hey, that's something we're interested in, or the investigator saying, hey, I need that patient involvement early on. Let me find a group that might be interested.
Not through Matchmaker, but on the NINDS website, if you type in a specific type of epilepsy, you'll get information about that type of epilepsy. And at the bottom, there will be a list of the associated patient advocacy groups. The other place to do that would be through the American Epilepsy Society if it's an epilepsy-related neurological disorder, who help match up. Very often so an example is there's a type of epilepsy called hypothalamic hamartoma, which involves a very specific part of the hypothalamus. And some independent researchers identified the signaling pathway that they believe is involved in the neurons that are causing epilepsy in that type of epilepsy. They knew nothing about the human clinical condition. And so I was able to bring the patient advocacy group together with those very basic neuroscientists to move that research forward. So that's a role that we play as well, because we know the advocacy groups that are out there and can match the basic scientists who maybe have a very interesting finding, but haven't really thought about how that could be applied to any kind of human condition.
Thank you.
Yeah, that's awesome. And I think we are running up on break time here pretty soon. And I wanted to thank all of you for joining us this afternoon. I think this is awesome. I think the young investigators and even some of the our seasoned scientists, can use this information. So I really appreciate you giving us your time today and the information. And we will definitely be in touch, and we will definitely share the resources that you've provided. So we want to thank you. And if our audience has more questions, go ahead and put them in our feedback survey at the end, and we will definitely filter them out to the participants so that they can answer your question if you did not get to answer it today. And I know we had a couple more questions that we had, but we're running quick on time. So please add that to your comments. And thank you guys again for everything. I mean, this is huge, and I appreciate everything. So
thank you
for the opportunity. Thank you.
Thank you. Thank you.
Thank you. Thank you. Okay, guys, well, we are gonna prepare to go on a quick break. But before you go to break, I want you to look at the slide, because when we come back from the break, we have 2 breakout sessions, and we have those 2 sessions are gonna be on 2 different links, or a link, and we have our basic scientist. If you are signed up to participate in the basic science group, then you're going to stay right here on the live feed. If you have signed up for the breakout group A with the clinical research panel, there is a separate Zoom link, and that was emailed out to you in your registration pack. And go to that Zoom link and you can log in, and it will be open with the panel there. After the 90 minutes, we're going to come back and we are going to close out, and we are going to introduce to you the end of the conference survey. We would love your feedback. So make sure that you don't get lost and click on that Zoom link, but then keep open your live hub link, because you'll just come right back there after you finish. And again, if you're in the basic science panel, you're going to stay right here. So we're going to take a 20-minute break, and we will see you back here at 3 p.m. Thank you.
Welcome back, and it's my pleasure to introduce the Basic Science panel. And today we have Richard Huganier, Bloomberg Distinguished Professor of Neuroscience and Psychological and Brain Science Disorder at Johns Hopkins School of Medicine. A quick personal note, I attended my first parent meetup at Johns Hopkins and was very impressed. Dr. Huganier came in on a Saturday and was explaining Syngap to a group of parents shoved into this small room. And I actually understood what he said. So I really appreciated that he was able to do it in, I would say, layman's language. And kind of opened my eyes a little bit, too, about what was going on with my daughter. We also have Shilpa Kadam, so forgive me if I mispronounced that. Research scientist, neuroscience, associate professor of neurology, Kennedy Krieger, and also Julia Dahlman, the associate professor at University of Miami. And they're going to have a conversation. And take it away, panel. Kadam, so forgive me if I mispronounced that.
Welcome, everybody.
Research scientist, neuroscience, associate professor of neurology, Kennedy Krieger, and also Julia Dahlman, the associate professor at University of Miami. And they're going to have a conversation. And take it away,
open it up
panel.
to questions. So,
Kadam, so forgive me if I mispronounced that.
I'll start and
Welcome back. We apologize for the technical difficulties, but as I'm sure we've all experienced in these days of COVID and everyone Zooming, inevitably there's a hiccup in some of these presentations. Once again, it's my pleasure to introduce our basic science panel. We have Dr. Richard Huganier, Bloomberg Distinguished Professor of Neuroscience and Psychological and Brain Science Disorder at Johns Hopkins School of Medicine. With him is Shilpa Kadam, Research Scientist, Neuroscience, Associate Professor of Neurology, Kennedy Krieger, and Julia Dahlman, Associate Professor, University of Miami. So without further ado, and hopefully no further technical difficulties, off to the panel.
Great, thank you. So welcome everybody, and we're going to talk about careers. And what we thought we'd do is start out by talking about our careers and the travels we've had. I think we have people at different career stages here, so that's great. And then open up for discussion. Maybe talk about the signature events in our career, grant writing, whatever you want to talk about. And certainly a signature event for me was Syngap and the discovery in 2009 of the mutations in Syngap leading to intellectual disability, and that's changed my lab. But I'll start from the beginning. And so we have an hour and a half, so I'll start at kindergarten. No, I'm joking. But I will start in high school. So in high school, I was really interested in the brain, and actually my senior research, senior project was actually learning and memory and protein synthesis, where I trained goldfish to learn a certain fear response. And so I've been interested in this a very long time. High school, I went to Vassar College, a liberal arts college, but had a biochemistry major, and so majored in biochemistry. But I always wanted to do neuroscience. And so after Vassar, I graduated and actually did 2 years of a gap year at Harvard University, working on RNA polymerase, so very molecularly oriented. And then went to graduate school at Cornell University in Ithaca, New York, where I worked with a famous biochemist, Ephraim Racker. And I went to work with Racker because he was an expert in purifying membrane proteins. And so I was really interested in neurotransmitter receptors, and decided to go there to learn how to work with receptors. And so my thesis was actually on the nicotinic acetylcholine receptor and purifying it and reconstituting it. And this was a prelude to my studies that I continue today. So from Racker's laboratory, then I went to Paul Greengard's laboratory at Yale. And many of you know Paul Greengard is a Nobel laureate, and there continued to work on receptors, nicotinic receptor, and then moved with him 2 years later to Rockefeller University in New York, where I was assistant professor for 5 years. And there I continued my work on nicotinic receptor and trying to understand how receptors are regulated by phosphorylation. And so this, I extended that to other receptors when I moved to Johns Hopkins, and this was 1988, so this is a long time ago. And so we started working on other receptor systems, including GABA receptors and eventually glutamate receptors. And that's what led me to Syngap. So I've been at Hopkins now 32 years, long time, and worked my way up through the ranks, and now I've been chair of the department for 14 years now. And so my lab still continues to work on receptors, really focusing on glutamate receptors, but also synapses and how synapses are involved in synaptic plasticity. And that's how we got to Syngap. We were characterizing proteins back in the 1980s and trying to identify proteins that regulated receptors and isolated Syngap due to its interaction with a major PSD scaffolding protein. And this is 1998, and this is at the same time that Mary Kennedy also cloned Syngap at the same time using proteomic techniques. And since then, we've worked on how Syngap is involved in plasticity and learning. And then the 2009 paper came out showing mutations of Syngap in intellectual disability. And so that's changed the way we do science in the lab, and we've become very translationally oriented. And working with people at Kennedy Krieger Institute and setting up the clinic there, and interacting with and collaborating with Shilpa. And actually now interacting with biotech companies and trying to develop therapies, potential therapies for the kids. So I've had relatively long career at this stage, and been through many different levels of going up through the ranks, but also serving as leadership levels. For example, I was Society for Neuroscience president for a year. I would not recommend doing that. It's a rather intense experience, but it certainly was interesting. So why don't we move to Shilpa? Why don't you tell us your story?
I didn't realize Dr. Hugener was going to start from his school days, so I've not even prepped for thinking that way. But the first story of my lab getting involved, which is I run an epilepsy research lab at the Kennedy Krieger Institute, and we're basically doing Syngap research because Dr. Hugener got us involved. I think we had our first meeting at the end of 2017, and he encouraged that we start looking at the EEG of these mice. And so that's where our Syngap story started. But my personal story, so all my pre-grad school education was done in India. So maybe I add the international perspective if anyone who's joined is an international student or postdoc. And that is a kind of slightly different perspective and trajectory. And I'm happy to answer any questions related to that. But I was actually working in a neuro ICU in a city called Pune, which is very close to Mumbai, which is the capital of my state back in India. And the neuro ICU, there are two things that we saw that were the biggest thing coming from ER into the ICU. One was, traffic accident, head injury, any kind of trauma, life-threatening. And the other was status epilepticus, young children, babies, young adults, old adults, coming in what is status, which is, continuous, where the EEG is showing continuous seizure activity and the brain never goes down to baseline, which is a life-threatening situation. So those were the, we got thrown into this, or I got thrown into this world where we saw that day in and day out. And as I watched the management, which seemed to me, it was for me, it was not very evidence-based. And everything but the kitchen sink was thrown at situations like that. And then there are well-defined protocols for certain syndromes. And that's where I got interested in neuroscience research. So unlike Rick, who was doing research way early in life, I never did any basic science research until I joined the lab, Ed Dudek's lab, when I joined the PhD program at Colorado State. So I'd never touched a mouse in my life, and it took some adjusting to, but I knew that that's where my interest was. And I was very fortunate to join Ed Dudek's lab, which was an epilepsy research lab. He's well known and well respected around the world for some of the protocols that we have been discussing this morning of when we are doing translational research. What are the kind of guidelines that will help us? Because, we are not trying to cure mice. We're trying to cure children, and we're trying to cure patients. So that translational perspective has always been heavy in my thinking, and also the way I talk to my lab and all the trainees that have, luckily many of them have now ended up in epilepsy research field. I'll hand it over to Julia and answer more questions later. But one, like Rick mentioned, something pivotal related to his Syngap experience. As a junior investigator, as a postdoc, and as an international postdoc, there's limited sources for where I could get funding. And one of the earliest fundings I got was this early career research grant from the Epilepsy Foundation. And that was very critical for me to set up my EEG core, because the equipment to get started in a location where there is no such facility requires funding. And that is the only reason I credit that early funding to me then getting NIH funding and actually having a full running EEG core now with telemetry systems and what have you. And funny enough, so that funding I got in 2011. And now I'm on the council that gives out those grants to early investigators. I've been serving on that council for 2 years now. And I have fought hard to increase the number of postdocs who get that early career grant research. So I feel like I'm trying to give back, because that was the pivotal moment where I knew I could set up my own lab and pursue what I was very interested in as a career that could then get funded by the NIH. So I'll stop there, and Julia.
Okay, so I started off completely basic science. So I went to Swarthmore College, where I actually got interested in biomechanics rather than even more biology, and then had a very influential developmental biology teacher who shifted my focus to more development. Ended up applying for graduate school, went to University of Washington for graduate school, where I found myself completely bewitched by the ability to record from ion channels. And I did that in Bill Moody's lab at University of Washington. I worked on a sea squirt, an Ascidian. And the reason I did that, and this dates me, is that it was a way of doing lineage tracing. So the organism itself marks its muscle cells. It's the cytoplasm that will become muscle is bright orange from the time of fertilization. So you see it sink with fertilization, and then it gets partitioned into the cells that will become muscle. And so we could look at the electrical program that led to the maturation of muscle cells. And I found that there was a period of spontaneous activity that resulted in the maturation of these cells. And I found that exceedingly exciting, but I wanted to understand at the molecular level how that might work. And so I applied to work with Gail Mandel, whose lab was working on transcriptional regulation of sodium channels, and had identified this fascinating mechanism where sodium channels were actually repressed in tissues outside of the nervous system. And I came to her lab, I had worked on Ascidians. I like animal diversity in terms of the lessons it teaches us about the many ways things can work. There are so many different ways to solve a problem, and so in her lab, I was sort of I worked on flies in a mammalian lab. There's no other way to put it. I was the only one working on flies. But basically, I took a transcriptional cofactor because flies didn't have the same transcription factor that the mammals did. And so I learned a lot of molecular biology in that project and a lot of chromatin biology. But at the same time, I was missing my physiology, and across the hall was another PI, Paul Brem. They're actually married to each other. And he had just started working on zebrafish, and they were working on a screen that was done in Germany. They were working on mutants that had come out of a screen in Germany, and a lot of them were in ion channels or the scaffolds that localize ion channels. And for me, that was enormously exciting because while and it would allow me to combine kind of the molecular and the physiology. And so I moved over to Paul Brem's lab. At the same time, I came down with cancer, Hodgkin's lymphoma, and it was grand that I was actually in Long Island because I was really close to Memorial Sloan Kettering, which is a fantastic hospital. And so in between those two, I was treated for Hodgkin's lymphoma, which kind of was more complicated than Hodgkin's lymphoma, ended up getting a stem cell transplant. Paul Brem, bless his soul, said, you're really interested in biology and you're not going to let this stop your career. And there are these career development awards that you can apply for. So he mentored me through the process of applying for a career development award, and which I got. And that funding was really essential. One, for kind of forging my own path, and two, for getting my job at University of Miami. I arrived at University of Miami with a whole group of human geneticists who University of Miami hired away from Duke. And so they saw that I, worked on this model that maybe could be useful in understanding inherited neurological conditions. And it turned out I moved in across the street from the person who is now the chair of human genetics. So despite the fact of being on different campuses, I found myself in this fascinating world of molecular inherited causes
or genetic causes of neurological diseases that related very much to what I'd been working on in Paul Brem's lab. And it was through those collaborations with the human geneticists that I arrived at autism, which was, to me, the disorder that I felt my skills could best inform at a deep mechanistic level. And so I found myself pulled in lots of directions, but that was the one I chose to dive into. And that was in large part because of Margaret Pearchick-Vance, who said, "Hey, do you think you can model autism in zebrafish?" And so then we had to decide which genes to look at. And her lab had identified a Syngap variant, and that's one of the reasons that we looked at Syngap. And we also looked at SHANK3, in part because I had a lot of experience in synaptic physiology. And again, yeah, you can ask informed questions based on your background. So, so since arriving at Miami, my lab has really taken that deep dive into zebrafish models of autism. And I like to say that I've never met an excitable cell that I didn't like. And so one of the things that was quite apparent to me, like the pancreatic beta cell is a beautiful excitable cell. There are lots of excitable cells that function in basic physiology of the body. And we don't know quite as much about how these genes are functioning in those contexts. And I thought that zebrafish, they form these very functional, coordinated organisms within 5 days of fertilization because they have to. They have no parental care. They either do that or they don't survive. And so, so you've got this small, self-sufficient little organism where you can maybe understand things at the systems level. And so, those are the questions that I decided to pursue.
Great. So I forgot to mention, that you have to ask questions. You have to log on to Slido app, and hopefully some of you have done that already. And so we do have one coming in now. I wanted to just to point out an interesting observation. So here, Julie and I are basic scientists. I was always a basic scientist, wanted to understand how the brain works, and but ended up through basic science, at least in my case, studying a molecule, defining a certain molecule as critical for. Here's my dog visiting me, and there's my wife.
Hi.
And
let's go.
Usual Zoom experience. But then this led me to translational opportunities. And so this is really, I think, a basic science dream, and really shows the power of basic science that, if Syngap was found to be mutated and we didn't have any idea what it was or what it did, we'd be 20 years behind. So, so I think that's really an example where basic science has impact on human disease. And I hope NIH is listening right now. So, but then, Shilpa had a very different path. She came from, being a clinician and being in the front lines and really seeing that it needed more evidence-based approaches. And so we do have different paths using different model systems. I want to come back to mentors because that's one thing that Julia brought up, but let's see what the question is. What would you consider the top 3 considerations academia will consider supporting a lab? So I think, funding from NIH is by edict, it really is supporting the development of therapeutics and for the health, increasing the health of the U.S. population, the world population. But they do support basic science, and that's something Story Landis said to me years ago, and she was, of course, the head of NINDS for many years. And she said, we really do support basic science, and so that's what I encourage my faculty and junior faculty, especially. You don't have to make a crazy model for how your basic science fits into a disease that NIH still funds in basic science. So, but obviously they are very disease oriented. So I think if you are really truly interested in disease, that's definitely of value. Story always said they fund. They like to fund translational science, but not fake translational science. So she really wanted, if you're doing basic science, you should say it's basic science and not put it off as translation. So obviously, I think to get good funding, you need do excellent research, and there's a lot going into that, running grants. Having good mentors who can teach you how to do that. So Shilpa and Julie, what would you say?
So I don't know whether I'm understanding the question for what they think, or asking us what are the skills that would get support from your department to have an independent lab. If that is the question, then this conversation I've had with multiple postdocs, and we have these sessions at the American Epilepsy Society mentor-mentee sessions, where clinician scientists who want to do basic research. So that's the other trajectory, right? That they have their residents, but they're very interested in basic research. And this question usually comes from that category. And it's the same formula that Rick just mentioned. You cannot ignore the basic science if you're going to do. So when I said translational, I mean, basic science is the base for all translational research. When I meant translate, that is what drives my brain. But for getting the kind of support you would need to set up an independent lab, my basic story was, I came from my epilepsy research lab. I came to as a fellow in the Department of Neurology, and I joined a group that was doing neurogenesis. And Kennedy Krieger, of course, the mission is developmental disorders, so that is a given. That's the basis for most of the faculty research at Kennedy Krieger. But going into this very different field where it's not different, because neurogenesis is also happening in epileptic brains, and seizures, any kind of synchronous activity affects neurogenesis. So doing my postdoc in that area, and I'd done 2 years of that, published a few papers. And then my department head, who was Dr. Johnson, who's retired now, I was ready to up and leave and do another postdoc. And he was like, if you're confident enough to set up, and surprisingly at Hopkins at that point, there was no one who was doing epilepsy research in the basic science category. And he said, if you want to set it up from scratch, that is something that Kennedy Krieger would be interested in. And that is why I said that, to try and get the funding, that was critical for me to start buying my initial equipment and start doing the initial recordings. But just then to answer that question, it is, like Rick mentioned, there are mentors who guide you, and that is very critical. And if my department had not said to me that, yes, we need somebody like you here, that took my confidence to, yes, I can do this. And also it was in an area very different from what my PhD advisor was doing. His lab was focused on temporal lobe epilepsy. And just the requirement of Kennedy Krieger being focused on developmental disorders was where that trajectory became a novel trajectory for my career. And but it was easy to fall into because everyone around me was doing exactly that, whether it be in Rett syndrome, whether it be in white matter disorders, and all that input from all those people allowed me to hone in exactly what I would do uniquely that would actually benefit the mission of KKI, but also be completely independent. And like I said, the support of the head of department was critical, and him communicating to me that there was a need for that, and that I should stay and try and set it up. So hopefully I answered at least part of that question.
So Julia, what do you think are the 3 key factors?
So I think it helps to make sure that you know how to write about your science. Writing turns out to be super important for success in academia. And another thing that I want to underscore from what Shilpa said is that there was a need, and she had this ability to fill that need. And somehow finding a happy position involves finding a community that appreciates your background and skills, and also valuing that community. So somehow that feedback, finding, if we're going to talk about stem cells, finding your niche, right, where you can grow and incorporate into a circuit is really important for success. And I think as somebody like when I went on the job market, I had been like really beaten up by life, and I had zero confidence. And I got job offers, and I was like, what are they thinking? But this is part of, it's interesting how engaging with different communities changes the way you see yourself. So I would say, and I think that's part of why people emphasize networking. It's partly, getting that feedback is really important, and finding a place where you can, be successful.
Yeah. So I'll go to the next question, but I just want to bring up the issue of mentors. I mean, and mentors are really, really critical, from your graduate thesis mentor to your postdoc mentor, but mentoring throughout your career as well. So, I was mentored as a full professor, still am, by, giants in the field like Saul Snyder and others. And so you need those mentors. And as Julie just said, networking is so important to interact with many, many people, collaborate, of course, obviously, but to have somebody there to help you along the way. So there's several questions coming in now. So what is the average size team you typically start with? It's 1, right? You start out with yourself, but go ahead. You want to start, Julie?
Well, that's a very good question. So you start with yourself. My mentors had very different types of advice on how to get going. And actually, it's an interesting transition, because you're going from being, usually when you start your lab, you are sort of the best experimentalist in your lab, for sure. And so being close to the experiments is really important, and working closely with that team, but also not well, at least the advice I got, and I thought it was good advice, was make sure that the quality is high. Don't overwhelm yourself with people before you really know everything that's going on, because it's a very I found it challenging to go from being the primary person doing the experiments to overseeing experiments. That's an interesting transition. So,
Shilpa, do you have any?
So, this was happenstance that when there was this model of neonatal ischemia, which was heavily studied at KKI long before I got there. But again, the EEG component was novel because they were quantifying everything by behavioral versions. And, hopefully when I talk tomorrow, EEG is the gold standard in the clinic for confirming seizures for a reason. And in the animal models, it is now, it is pretty clear if you're going to do any phenotyping or natural history or seizure burden kind of quantification, that it ends up being, it is the gold standard. And luckily, this was happenstance again, that during my PhD, at that point, Kevin Staley, who's at Harvard now, the head of neurology, he was at Colorado. And he and Ed Dudek at that point, when I joined the program, was setting up this telemetry system, cutting edge, DSI, very expensive. And I just finished my rotations and I joined the lab. And when I entered the animal facility, I remember Kevin Staley sitting on the floor making the connections to set up the initial system, right? And at that point, even he was head of neurology in Colorado. And it was amazing to watch that process being set up. And I said, I want in, I want to learn this. I want to do my project on here. And so the lucky part there was I got to see that system being built up from scratch. And I cannot stress enough, if you're going to be confident about something that you're going to do as a career, you better know how to solve every problem that comes up, okay? Because that is, if you don't know how to do that and you're depending on someone other than yourself, that one thing, I'm not saying multiple things. Yes, you need to be the master of at least one thing. And for me, it was not even a strategy, but it just happened that system was getting set up and they were problem solving and figuring out, and the best minds, because Kevin Staley is very well known for his epilepsy research career, and so is Ed. And that's when I saw that happening and I got interested in it. I designed my PhD project based on that, on telemetric long-term recording. And you will hear a lot in the clinical talks, even in this session, where even in the children, long-term EEG is another gold standard. To catch a seizure, even to figure out whether your child is seizing or not, especially if they're subclinical, you need long-term EEG recording. And telemetry afforded that. We went to the cutting edge of it. We were recording for 5 months continuously. And I had no social life for 3 years because we were just recording. But that kind of, getting down on your knees, connecting everything, figuring out when it crashed, how to correct it, figuring out the protocol with people who were leaders in the field. I think that kind of training is much better. It might be intimidating in the beginning, but when you want to set up something on your own, you have the confidence. Then whatever comes up as a problem, you'll find a way to solve it. And the same thing, any electrophysiologist, they know if you build a rig by yourself, then when the noise comes up or something goes wrong, you know how to fix it. And so sometimes, I have seen people who, a well-oiled lab sometimes, where you come in and everything's already fixed up for you, and you design your perfect experiment and do it. You might have a great project. When you become a PI and you have to set it up from scratch, and trust me, everything goes wrong when you first started. I feel like if you've had that experience, at least for me, it is true, that nothing intimidated me. Everything that could go wrong went wrong, but I figured out a way. And of course, I had, my PhD mentor. I would talk to him on weekends. He would take my call whenever. And setting up something from scratch at a place that does not have a similar core or expertise, you do heavily depend on people who have mentored you. And like I said, getting involved in something when it was just getting started was happenstance for me. And hence now when I design new things, when I just set up telemetry for my own lab, it was as if, that's what I was meant to do kind of stuff. But that is because of the experience I had. And I did not dictate that, that they would be setting up that system right when I joined the program. So that has been my experience.
So, what Julia and Shilpa said is true, right? You start out by yourself, and you're the best experimentalist in the lab, and you will be for the first few years. And so that transition is difficult. And also remember, we are not really trained to manage people. That's a big transition for a lot of people, is how to, as you build a lab, how do you choose the right people? How do you evaluate people? How do you manage them? And so that's a big leap. So in my case, I started out with me and a few, a couple graduate students, and then build up along the way. And my lab is very interdisciplinary. We do everything from proteomics to imaging to physiology. So it's actually, now it's 25 people, so it's relatively large, and a combination of students and postdocs. And so that's grew pretty quickly in the beginning of my career. And but what certain people have certain they prefer to have a small lab and continue to do experiments themselves. I haven't done an experiment in 20 years, so if I walk in the lab and hold up a pipette pen, everything stops and everybody looks at me. But so you have to be able to manage and maintain the productivity of each individual. Remember, you're responsible for the success of all the postdocs and trainees in your lab, so you don't want to fail them. So I think you have to balance the how many people in your lab due to your own personal taste, and also your managerial skills, and also the breadth of your program. So I think for different people it's very different, but obviously everybody starts out at the same spot. So then how do you decide how far you branch out from your postdoctoral and your advisor's research directions as an independent PI? So Shilpa mentioned that already. Julia, do you want to tackle that one?
Yeah, so that's something that for me was solved by, I branched very quickly, in part because of the collaborative opportunities that existed at the institution that I landed at. So by being sort of an honorary member of the human genetics department, I moved away from many of the models that I'd worked on as a postdoc. And so that gave me quite a bit of independence from my postdoc mentor. And that's a tricky transition, figuring out what you're going to work on and what you can take. And, sometimes people are in direct competition with the labs that they leave. And they'll publish back-to-back papers in the same journal, right? I mean, that does happen. And so that's something that's, at least in my experience of watching other people, is very idiosyncratic how that process plays out.
In my case, it was pretty simple because actually I brought my project to the lab in Greengard's lab. And so I was working on the nicotinic receptor, something he wasn't doing, and then I left with that. And so it was relatively simple. My advice that I give, this is always an issue for my postdocs who go on for me, have open discussions about it. Some of them, try to find a niche that are still interested in receptors or synapses, and they work on a different receptor. Steve Moss, actually, who was mentioned earlier today, was a postdoc in my lab. He and I, 25 years ago, decided he would do GABA receptors and I would do glutamate receptors. And so there was that arrangement. And but then I've had other postdocs who have basically competed directly with me, and that was fine. Gavin Rumbau and I are a perfect example where he was a postdoc in the lab and was working on Syngap, and he went off independent career and continued to work in Syngap. And to this day, we still are both working on Syngap, but we talk all the time and collaborate and coordinate our research. And so there's many ways to do that. So I think, sometimes in a crowded field like mine, it's not always good to to work exactly on what I'm doing, because there's 10 other people doing exactly what I'm doing as well. So I do encourage people to find a niche, if they can, because funding in a crowded field and can be difficult. Shilpa, do you have anything to add before we go to the next?
No, I agree. I mean, like Julia mentioned, it is a little bit idiosyncratic. It totally also, depends where you are and what the situation in that department is. If you find a niche, it may sound a little bit starry-eyed, but find the thing that makes you wake up in the morning and want to go to work. And then if that overlaps or does not overlap, or it's a strategy, that's great. But you should be really excited about what you are doing. And if that meets with your strategy, fantastic. But strategizing just for the strategy, I don't know. Maybe don't have enough years to know whether that would just work by itself. If you're very, you are motivated by what you're researching, you have a goal for where you want to take your research, these niches will show up. They will in collaborations, in networks. It's not static. So nothing is static in science. That's the beauty of it. And you will find the collaborator, you will find the niche, maybe not in the first 2 years, but definitely in the first few years. And then you'll figure out from the study section what the NIH is interested in. But if you are interested in it, then you can change NIH's mind and get them interested in what you're doing. That might sound a little bit starry-eyed, but I'm still in that phase.
I'm starry-eyed too. And this actually is related to another question coming up, which when we are interested in many research areas, how do you select which one to pursue? So just exactly, I advise my postdocs, don't try to plan your research plan based on what you think is fundable. Do research that really interests you and will drive you and motivate you, exactly as Shilpa said. So so that's what you need to do. And it's obviously you'll learn how to make that approach more fundable with time, but don't work on something that you're not passionate about, that's for sure. And so if you're a young investigator who stays in science, no, wait a minute. Yeah, well, I lost the question here. But if you have many different research interests, really pick the one that's you're most passionate about, and also obviously you need the technique, so you have to something you can approach with your expertise and the techniques that you have. How about funding opportunities available for international students? So that's a major issue. For postdoctoral, for pre-doc, for PhD students, a lot of universities have funding for PhD students. Postdoctoral fellows is more difficult, as we all know. But there are opportunities, mostly non-NIH opportunities, although the K99, and there are a couple other mechanisms that are open to international students. But a lot of my international postdocs will apply to, for example, the Simons Foundation or to the BBRF. So private foundations that are funding, postdoctoral fellows, international and U.S. citizens. Shilpa, you're international, so.
Yep. So as a pre-doc also, I agreed, I applied. So I'd say write your first pre-doctoral grant, if not on the first day of joining your lab, within the first month. The draft, send it to your mentor, have them put in inputs. For me, I already knew why I was joining the lab after I had done the rotation. So I actually, practice what you preach. I sent in 2 pre-doctoral applications within the 4 months of joining the lab. And both of them got funding scores. One was, again, Epilepsy Foundation. The other was American Heart. So that was totally, guided by the mentor on what fellowships are available for internationals. And the American Heart was a 3-year fellowship. So I got my entire PhD, almost all, and then I also got a 1-year extension competitive renewal. So my entire pre-doctoral research was funded by the American Heart, and it also gave the confidence of writing grants and getting good scores and reviews. I mean, that I've never been able to replicate those kind of, beautiful reviews as a faculty, but that speaks more to the mentorship. They were, the reviewers were very confident that the lab I was in would provide the support I needed to attain. Every grant I've got is this is too ambitious. I don't know what it is. I don't know whether it's a story for everyone who gets that from pre-doctoral, postdoctoral grants, even now, one of the comment is always, this is too ambitious. And, I almost feel like going back to the American Heart person who wrote that this is too ambitious, that I got everything I said done during my PhD. And I also graduated before because my lab was a huge lab, just like Rick's lab. There were 7 to 8 postdocs, and grad students who joined the program with me were 3 other people other than me. So his lab took in 4 grad students, and he almost was like, this is too much. I had to almost twist his arm because I made my decision a little late because I was doing a 4th rotation. Although I knew right from my finishing, so that would be, when you do rotations, one advice I would say that I had not communicated with him that, yes, I think that was the fit. And I went to do a 4th rotation. And by that time, who would have thought that there were 4 people who would want to join the same lab. But we were not short of resources. Rick can speak more to managing huge groups, but I watched and I wonder if Rick I think he will agree with this, that there's not one formula fits all as a mentor and mentee relationship. Every mentee is an individual, and you have to figure out, some people need constant supervision, some do not like it. And I watched in real time how my advisor would spend more time with people that needed it. And he hardly met with me. We met on the weekends. But when we met on the weekends, he knew exactly where I was. He addressed all my questions, and with the funding thing, he encouraged me, go ahead, write. I thought he threw it as a challenge that go ahead, write your grant. And I remember after I told him 2 months after joining the lab that I want to write a grant based on cortical epileptogenesis. He said, go ahead, send me a draft. And I was back in his office 2 months later, and I said, I sent you the file, and he printed it on his printer, expecting 1-page specific aims, and the printer kept spitting out 25 pages. So I had my entire application ready. And that's when he said, okay, fine, you can go send this out. And then it got funded, and then he got more confident that I could do this on my own, and I did not need the kind of, and I saw him, that he treated every individual according to their needs as he figured it out. And I didn't appreciate it then as much, but now as a PI myself, I appreciate it more and more that not all interactions with your mentees cannot be cookie cutter. You have to take into account who the mentee is, what their needs are, what kind of interaction helps them thrive. And I don't have as much experience, obviously, as Rick has under his belt, but that approach has, I realize when I'm interacting with someone that they think that is too much, and they're pretty independent, and letting them be independent actually gets the best out of them. And but you also have to prevent the people who, just like you have to be very strong in certain techniques, especially if you want to run your own lab, because you are the one, like everyone has agreed, it's a lab of one when you start, and you set up the foundation. But you also have to guide people when, intelligent brains have multiple ideas, but you cannot follow through on all ideas. At some point, you need to focus. And sometimes then you have to, that kind of mentoring I find is your job is just to keep their focus on what they are most interested in. And and the golden rule of course is publish and perish. And so if you want to publish, you have to be focused on that immediate project is what I'm finding before you move on to all the other, because as in any science, you go to answer one question and all you get back is 100 more questions, right? But to get funded, you need to give NIH a story and a study section, a path of what you're going to do for the next 5 years. And many times and I'm not even speaking of mentees, I'm speaking of colleagues with me, people who were in grad in the grad program with me who are not faculty position. And I don't know whether we'll talk about industry, but industry is a good alternative. We all don't have to stay in academia. That the only reason I graduated before all the other 4 in the lab was in the second month, I knew exactly what my specific aims were and what I was going to focus my energies on. And the others, and I don't know whether this is good or bad, maybe Rick can speak to this, that they started 2 or 3 different projects and were unable to be masterful in all 4. And those did not end up in publications. And they did not end up being, because we would have these lab discussions that definitely, especially in epilepsy research, the history of publications is 30, 40 years old. So for you to understand the trajectory of where you want to go and what would be a new niche from what is published, you really need to figure out the one thing that drives your interest and focus on it first before, as a pre-doc and even as a postdoc, before you start thinking about, now I have the luxury of thinking about developmental disorders and every disorder that's associated with epilepsy. But if I did it as a pre-doc, I think it would end up with having multiple projects that did not end up in a publication. But that's my experience. I don't know that would be true for everyone, but I prefer for pre-docs and postdoc to be laser focused on at least one major project, and do it all yourself. So my PhD advisor said a beautiful thing that when we used to have discussion, he's like, "This is your project. So here you are the PI, and you're explaining to me as the person who needs to have the most depth of knowledge on that project is you, not me." And he would his mentoring was he would make me tell him everything I was doing, why, and it was like a live study section. And which means you need to be reading up on every possible paper that's ever written on that subject. Because it's epilepsy research, you need to be reading the case reports, what's happening clinically at the bedside. And he was open to all those discussions. And so I think a laser focus early on actually is better than having multiple projects of multiple interests. But so that was my experience.
So I have a question here from the moderator, maybe Monica. Could each of the panelists talk about what most excites them about the direction of current basic science research in Syngap? So I'll take a shot at that because I've been at it the longest. And what really excites me is actually how many people are getting interested in Syngap. And for many, many years, it was basically me, Mary Kendi's lab, and Seth Grant's lab, and for many years. And then as our trainees went out, a few of them, Gavin, branched out. And but now, especially because of the disease relevance, many, many more people are getting involved. And you started seeing people working with zebrafish, and we're going to hear C. Elgin's talk later in the week, and all the different model systems. So it's really exciting to see so many more people being involved in understanding Syngap. And I think Monica said this earlier that we know a lot about Syngap, but there's really, it surprises me what we don't know, and what we're finding out is we're learning something new every day. It's quite complex, and Syngap function, as we'll talk more about in the week, it's not what we thought it was. We thought it was a RAS gap, but now it's it has other gap activities. It has a structural role at the synapse. So it's involved in dendritic development, not just in synapses. So I think it's we're finding out new things every day. So I'm excited for the number of people, but also the new discoveries. And obviously, from a translational point of view, how do we develop therapies? And I'm really excited about developing genetic methods for increasing Syngap expression, and then we'll talk about this later. But I think it's pretty clear that most Syngap kids are haploinsufficient, that they really only have 50% of Syngap, and so we really need to increase Syngap expression for the majority of Syngaping kids. So whether that's an antisense oligo approach or a gene rescue approach, it's not clear yet, but it's really exciting to think about. So what excites you guys?
Julia?
Julia, you want to go first?
So, I mean, this brings me back to what Rick was saying, that if we didn't know because of the basic science, if we didn't, and all the work that labs like his have done, if we didn't have a basic understanding of what Syngap1 was doing in the brain to begin with, then people like me who have this mindset about the systems of neuroscience approach and the circuits approach would not, that there we are standing on their shoulders so that when we look at the EEG output, we have some understanding of what that might mean. And this morning, the young investigators were talking about, a focus on comorbidities is essential. For me, because at Kennedy Krieger, most of the neurological syndromes that get looked at in the children who visit KKI, whether it's acquired or genetic, many of those neurological syndromes are associated with seizures. And as an epilepsy researcher, I have this, this basic understanding. The circuit that is driving the seizure is also the circuit that's underlying the cognitive disability, the intellectual disability. So there is a mechanistic way, if you understand the mechanism, because the seizure is the output, the mechanisms underlying the seizure, and hopefully I'll address this a little bit in my talk tomorrow, could be very individualistic in some ways, and there's always a Venn diagram overlap that can afford some insights, because as there's not going to be for me, there's not a perfect deadline by which we are going to end up having the perfect cure, because I work with the International League Against Epilepsy, and we are trying to steer, in the mindset for epilepsy, steering away from this word cure. Because even if anything you do in basic or translational science that improves a child or their family's life by 5%, you still have done a considerable difference in that family's life, in that child's life, and in the trajectory that child might take down the line. And so with the, all of this is not, these insights are only gained from everything that's already published and known about what Syngap1 may be doing in the brain. And as the data we saw this morning, as is anything in neuroscience, the devil is in the details. And as the details come along, because EEGs is a circuit output. And I keep telling this, and we were having this discussion last week, the brain is very plastic, especially immature brain. It will adjust for multiple insults. And most of us turn out to be normal. And that's amazing, which means that, for all the kind of insult, maybe high-grade fever for every child that's below 5 years old, we don't all throw seizures and our brain adapts. When there's injury, we all know, the younger you can have a massive resection surgery at ages younger than 6 years, and the brain will adapt. That child will not lose function, which is not true after the critical period. And so for me, the data that comes from basic science, when we apply it to the circuit, which becomes way complex, and simulation studies try to get at it. But what we have no clue, which is what is exciting to me, is during the developmental stage, the brain is also maturing. And that process in itself is very complex, and we don't understand all of it. It's a jigsaw puzzle. And so for my lab and my group, when we see different papers, we get excited because we think we have made some connection, and we have those excited conversations. But these pieces of excellent work by multiple groups is all out there. And I feel like we are sitting at, or just because of our mindset, we are sitting at at least at the 10,000 feet view, where we imagine we are seeing these pieces, and most of it is based on solid basic science. And then all the other labs doing stuff where we try and make connections of what might be a good readout for EEG. And our first paper is a perfect example of that. Because we're looking at quantitative EEG biomarkers underlying just a 24-hour EEG. And there we've already met the gold standard of a long continuous EEG in a developmental disorder. But then other than just looking at seizures, we're looking at the background EEG, which is now actually becoming a thing in the clinic too. The funny part is we are looking at it at a resolution that nobody is looking at it regularly in the clinic. So one of our interests now is looking at, and Monica knows this very well because we've been hounding them to get overnight EEGs of kids who have been diagnosed so that we can look at the background EEG. And the reason we think we know what we want to look at and the reasons why is coming from the basic science. And we apply it to what that might mean for a circuit and what the circuit biomarker might mean to function. And that's the only reason in our pilot study we felt confident to try a low dose of a drug that was an EMPA antagonist. Most of the work came from Rick's lab. And the fact that that modulator can change that biomarker acutely provides tremendous insights for someone with our mindset that hopefully we think will be an addition to add another jigsaw puzzle that will help us figure this thing out.
Great. So we have 5 minutes until we take a break at 4:15 so we can go to the final panel at 4:30, the wrap up. Julie, do you want to address that question? Or?
So I'm going to do it in a big basket. But one of the things that I found very exciting about this morning's set of talks was, despite the fact that we know a lot about Syngap, Syngap is a protein that functions in many different contexts and in different circuits is doing different things. And is clearly expressed early in development before there are synapses. And so there is more that we don't know about Syngap than that we do. And I think that we will learn some really interesting pieces to symptomology from those new contexts.
So, I think that we are moving to more systems level, looking at circuits, and that's one thing Gavin's doing a lot, and others are doing it. And I think Syngap is expressed in most excitatory synapses in the brain. So, why is it giving this unique phenotype? And so understanding what are the relevant synapses that lead to the phenotype, I think is very important. And using all these different techniques, all the different model systems, I think is going to be incredibly helpful. So we have 3 minutes. The last question is, what is your biggest aha moment? And have you ever proposed a project that was seen as crazy, but it worked? So do it in 1 minute. I'll tell you real quick. So, actually that's one thing I miss is that as a PI of a big lab, you don't look down the microscope and have that aha moment that I used to have, when you see the result for the first time. But I've suggested lots of crazy experiments, and the most recent one is that I convinced I have a great technician, and I said, well, I want you to make a GFP knock-in in AMPA receptors so that every excitatory synapse in the brain will be green. And the reason it was crazy is because the brain will turn green, basically, right? So it would be useless. And so we did that, and nobody wanted to work on it in the lab just because we started imaging, and it was just it turned out to be incredibly complex. We were actually imaging millions and millions of synapses at the same time. So, but finally I got a postdoc who was willing to take it on, and he took it on, and it's turned out to be an incredible resource that we can, we're still having trouble analyzing the data, but we can actually image millions of synapses in real time in a live animal. So that's going to, I think, be a very powerful tool going forward. All right, I see Monica's coming back, so I think we've run out of time. So I'm sorry if I took the last word, but thank you, Julia and Shilpa. Really appreciate it. So we're signing off now for the break.
Thank you.
Thank you so much. That was an incredible discussion, and it is just really fascinating what you can learn just by listening. And so I want to thank the panelists, and I want to thank the audience for throwing out some great questions. And what we're gonna do now is we're gonna take a short 15-minute break, and then we're gonna come back for closing. And we would really appreciate you also filling out the feedback survey and tell us what you thought and any questions you might still have. So we'll see you back here in 15 minutes.
Welcome back, everybody. And here to close out today's meeting, we saved the best for last, as one usually does with meetings. Pleased to introduce Dr. Connie Smith-Hicks, PhD, MD, research clinician at Johns Hopkins School of Medicine, and the Kennedy Krieger Institute to deliver some closing remarks for us. Take it away, Connie.
Thank you. Good evening, everyone. We've come to the end of the more formal part of our Young Investigators meeting, and it's my pleasure to thank, on behalf of the organizing committees, Drs. Holder, Rumbaugh, Monica Weldon from BTG, and myself. We want to thank the NINS NIH committee and all the funding partners for supporting this meeting. Certainly the young investigators, without whom we wouldn't have a meeting, and all our speakers. That includes our young investigators, the NINS program directors, and mentors for sharing their experience and expertise. In addition, we want to thank Charles and Monica for moderating all the sessions today, and all the behind the scenes crew, Leslie, Drew, Connor, and Dudley Media, who worked tirelessly to make this day the success that it was. And finally, I want to thank the patients with Syngap 1 and their parents. They are a source of inspiration and the reason why we do this work. Their passion and the obvious can-do attitude that we heard and saw today has certainly encouraged us to do more and to do better. Now, last year when we began to plan for this meeting, no one knew that we would have a pandemic on our hands and that we would need to pivot from in-person meeting to a virtual program. But I think the team did a phenomenal job, and I just want to thank them for that. I just want to leave with a few parting words to our young investigators. When we wrote the grant, one of our primary goals for the meeting was to increase the number of researchers in the Syngap field. So whether your interests are clinical, translational, or basic science, there is room enough for you. As you heard today, Syngap is a complex multi-system disorder. And despite all the progress that's been made, there's still more questions than we have answered so far. Another goal of the meeting was to foster collaboration between research groups across disciplines to advance our understanding of the disorder and to bring targeted treatments to clinic. Now, several of the young investigators touched on this exact topic. They talked about the need for cross-collaboration. And so it is our hope that you will join us for the remaining 2 days of the conference, and that this will be an opportunity for ongoing exchange of ideas and the development of new partnerships and collaboration. Please remember to complete your survey of today's meeting. I think Monica may tell us a little bit more about that, but this is really an opportunity for you to tell us how well we did and the areas that we need to improve in. Now, there will be a Syngap happy hour later today. It's an opportunity to continue to meet and mingle with Syngap families, young investigators, clinicians, and researchers. And the main conference will start tomorrow at 8 a.m. Eastern Time. So thank you again and have a wonderful night. Back to you, Monica and Charles.
Thank you so much, Dr. Connie. And I want to thank everybody today for being with us. This has just been a phenomenal day. And, being the third young investigator workshop and meeting and hearing all the great research, I have to say on a personal note, I never dreamed that a community like this and all the researchers, when we started out with really just 3. And it is such an incredible team. And I want to thank everybody, the committee. It has just, I can't thank you enough. And really quick, you can find the post-conference survey In the email that you received and also on the website where you're watching the live stream now, we will be sending up a follow-up email to remind you if you did not get a chance to do that today, but we would like you to give us your feedback and questions that you still may have. Just as Dr. Connie has invited you to come to our meeting tomorrow. This is going to be phenomenal. And then on Thursday, we invite you all to come and join us for the externally led patient-focused drug development meeting, which is going to give an incredible overview of what our families and what our children are having to live with on a daily basis, in the hopes of working all together with the Food and Drug Administration, with the families, most importantly too, and all the researchers and industry to come together to forward the movement towards treatment. So thank you, everyone, and I'm going to let Chuck end with his little final note and come back and see us tomorrow.
Well, it's been a great day, and the thing I would highlight from the day is if you're a parent of somebody with Syngap, we covered gait, senses, seizures, behaviors, hyperactivity, learning, memory, gastrointestinal issue, and of course the science around Syngap, all of which is very relevant to our children who have Syngap. So with that, I hope you join us for the next couple of days and look forward to it. Thank you all.